His today's topic of the session is discovering biomarkers using high throughput techniques. Sir, we are honored to have you here for this session in this international conference. On behalf of ASP College Devrut, I once again extend my heartiest welcome. Welcome, sir. Now I request Dr. Shom to begin with his presentation. Over to you, Shom, sir.
Okay, thank you very much, Dr. Uday Bhatia and Dr. Pratap Naikwade for inviting me for the talk. So can I now share my screen?
Yes, sir. Yeah, sure.
So today my topic will be, as they have mentioned, that I'll be talking about discovering biomarkers using high throughput techniques. So this is a little bit off from what the other topics has been, but these days, biology and after COVID, a lot and more people have come to know about what are antibodies, what is virus, and what is how they affect us, and what is immunity. So I'm going to talk a little bit about the basic things about what we do in a molecular biology lab, and how we do. So a little introduction about me. Dr. Uday Bhatia already explained. So I'm Ashish, and I'm a postdoctoral researcher at ASU, Arizona State University. And I did my bachelor's and master's degree from Indian Institute of Technology, Dhanbad. And then I came to US and pursued a PhD degree in ASU in proteomics. And my research interest includes proteomics, immunology, and bioinformatics. So with this, I'm going to move ahead, and then I'm going to talk about a little bit about what are antibodies, and like when I say about antibodies and what are their functions. So during the COVID time, we have come to know that when you get infected with a virus, we need to have antibodies in our body. If you don't have antibodies, the virus will invade us. And that is why a lot of people used to get antibody shots in their body if their body is not generating enough antibodies. So on the left, you can see that this is an antibody molecule. And this was first, scientists, when they crystallized this protein, and they found that they thought that it's a Y-shaped molecule, and they thought it's a common thing, and there is all these antibodies are the same. But later in 1900, Paul Ehrlich, he for the first time found that actually these antibodies are very specific, and they have the specific epitopes, like the sequence of proteins, sequence of peptides, which are very specific, and they can bind to specific things, and they don't bind non-specifically to things. So what happens, the function of antibody ranges from different things, like it acts as it provides an immunity to our body. So what it does, suppose this is a virus, for instance, COVID virus. So when it enters our body, these are the antibody molecules, and these are very specific, and they can exactly target that virus and not affect any other thing, like not bind to any other bacteria or any other body parts, proteins in your own body. So this is one of the reasons why these antibodies are so special, and because they are very specific, and they can target the things. Means theoretically they can bind to anything. It's like anything you get invaded with, it's a bacteria or a virus, there will be an antibody which will be generated by your body, which can then bind and destroy that bacteria or virus. So how these antibodies are produced. So what happens when you have a pathogen in your body, for instance, a bacteria or virus. So we have this immune cells called dendritic cells and macrophages. So they produce this antigen, this bacteria or virus, to your immune system. So in the immune system, there is another cell called B cell, and then these B cells tries to find a match with with the peptides of the bacteria or virus. So if it finds a match, when it finds a match, it activates that B cell. So out of billions of different B cells, only one B cell will get activated, which has exactly the same epitope as one found in the bacteria or virus. And then it differentiates into two. One is memory B cell, and one is this plasma B cells, which produces the antibody. And that's how we get the antibodies in our blood. So just a quick note about why this antibodies and these are important. So during this COVID time, in Arizona State University, we did a lot of sampling of both detecting the coronavirus. So ASU was one of the prime center of Enter Arizona State, and this was all done in our lab. So used different, robotics tools to, because we have like so many people coming, and this was all done free of cost because the government of Arizona supported our lab, because we have all these capabilities to detect the antibodies, detect the DNA in the RNA virus in the samples. So we did in a huge lot of PCRs, we did a lot of this antibody screenings, and we studied a lot of things, and then we tried to understand how this affects the population, whether what is the prevalence of the COVID in the society, and how is it decreasing or increasing, and which variant is increasing and all. So this is all possible because of this antibodies detection. And also there is another part of using the PCR, which is detecting through the DNA, the RNA molecule. So with this, I'm going to look into the antibody part more. So how this antibody-based diagnosis compares with other diagnoses. So you know that a lot of there are different types of, when you have a disease, you get infected with a virus or bacteria, the doctor gives you different types of diagnosis. It can be CT scan, where commutative tomography, or it can be ultrasound, using high frequency sound waves. They see what is the shape and tumor size and everything. Or it can be endoscopy, in which they put a camera and then try to see if there is inflammation, if there is any sore, if there is any ulcer in your gut. And then there are other cancer ways in which people do biopsies. They take the samples and they check if the sample is cancerous or not. And so among all this, if you see, like, we have another thing is the lab test, which we do is in our lab, is based on serum, stool, or any body fluid, and we check the antibodies. So the reason these antibodies are important, because they stay in your body for a long time, and it can tell about, it's like a logbook, and it can tell you about if you got infected with a bacteria, or if you get infected with any other autoimmune diseases or cancer. So you'll have that antibody, and based on that, we can exactly say, like, how serious is the disease, and what are the chances of progressing to a more serious type of disease. So if you look here, early detection saves lives. So if we can detect early, most of the people die from cancer, because a lot of people, they don't know about that they have cancer until they are at the third or fourth stage, when the symptoms starts to show, and when it's already out of control, when it has metastatized to other part of the body. And that's when the person comes to know and goes and sees a doctor, and that's already too late. So if you see here, this is for a non-small cell lung cancer, and the survival rate at stage 1 is from 68 to 92%. And as it goes down from stage 3, it's 13 to 36%, and stage 4 is almost no chance of survival. Similarly with breast cancer, and you can see at stage 1, it's like 100% going down, and from stage 2 to stage 4, it's like abruptly drops to 32%. And that shows like why it is important to detect, the early detection is important. And early detection can only be done when we have something which is non-invasive. We don't have to go to a doctor and see something very expensive device. And we sometimes, it's not possible for us to, in poor and rural villages, it's not possible. But if you have a blood-based sampling system, if you have a blood-based testing system, it's much more easier, and you can check whether you have a chance of developing that cancer, or whether you have symptoms of, whether you have antibodies which are correlated with that type of cancer. So these are some of the techniques of antibody. As you can think, like in our body, there will be a lot of antibodies, because there are so many viruses and bacteria, so many different types of antigens we get exposed to. So every day we get exposed to different viruses. When you get out in the road, every time we are getting exposed to different viruses and bacteria and pathogens. So our body is continuously, like saving us from this. And that's why we should have so many antibodies, different types of antibodies in our body. So as you can imagine, it's not easy to just find an antibody and see. So it needs to be something very high throughput, where you can detect so many, because in human proteins we have like 20,000 different proteins, and then we have post-translational modification. So there are so many proteins, and if we check one by one, it's almost it's not feasible, and it will take so much time. So there are some techniques which are used for detection of these antibodies. One is ELISA, which is called enzyme-linked immunosorbent assay. And in ELISA, this is pretty standard in molecular biology labs. We have antibodies, and then we add the antigen, and then we have a detection antibody and the secondary antibody, the fluorophore we check. And Western blot is also very famous, in a very common technique in molecular biology labs. So this is also based on gel. You run the gel, and then you transfer to a membrane, and then add an antibody and check where the proteins are and what are the size of it. And this is pretty much recent, which is the Luminex beads assay, in which you have beads and you functionalize that with antibody and then check. So all of these techniques are good if you want to check only one or few. But we need to check thousands of proteins. And then we have, like, also from person to person, our genome varies a lot, because sometimes you get a medicine, it works on you, but the same medicine doesn't work on another person, because their genome, their body, their proteome, and then is different, and they react differently. Their immune system behaves differently. So it's so different whenever we see the samples, like we have so different samples. So we need to check like hundreds of samples. So when you check more samples, we get more, the statistical, the power of that statistics is much more powerful. And because of this variation, we need more samples, as well as we need more proteins. So what we do is we use protein microarrays. So this is a high throughput technique, as it is high throughput because we can check so many proteins simultaneously. So this is a glass microarray. This is a glass slide, which you conventionally see in biological lab. And then on this glass glass slide, we print the proteins. So these are the proteins. You can see, like, we have different proteins printed on this glass slide. And there, virtually, you can print thousands of proteins in this glass slide. And then what we do is we add the serum from the patients, and then we add a secondary antibody with fluorophore, and then we check the fluorescence in a laser scanner. And using that, we see, like, what are the proteins actually are illuminated. That means that the antibody bound to this protein, and that means that that antibody is present in that person. And then we check it with another person which has, like, no symptoms, and then compare between them how different they are, and is that something which is occurring again and again in the patients with some type of cancer or some type of autoimmune disease. So let me talk about a little bit about the functionalization, how we do, because this is a glass microarray. So what I do is we print the protein on top of it using robots. So the chemistry is, the glass is made up of silica, and then it will have silicon dioxide SiO bonds. So what we add is we add aminosilin. So aminosilin covalently binds to it, and then it has an NH2 group. This NH2 group, then we add a linker so that it has more mobility. It can bind freely. So we add this BS3 linker, and then we add the capture antibody and then the plasmid DNA. So once we have that, this is what is in our protein microarray. We have this cell-free in vitro expression system which expresses the plasmid DNA, it expresses the protein, and this protein then gets captured by the capture antibody. And that's how we have the freshly prepared protein which is printed on this. One of the drawback of this glass microarray is that this is because this is on a plane surface. So what happens, the signals are dispersed, and sometimes, two adjacent proteins, they have the signals which are overlapping with each other. So that's a problem, and we can't, keep the proteins very close to each other. So we have to keep them quite apart. So in order to curb the problem, what we did is we did a silicon microarray. So in silicon microarray, what we did is we actually using Roberts, what we did is on the silicon microarray, we engraved wells, like in ELISA plates, in 96 well plates, we have wells. So we have something like that, a smaller, very small wells inside this glass array. So now what is the advantage is that because all the proteins are printed and all the reactions are occurring inside that well, so there is no dispersion into the adjacent well, and there is no dispersion, so we can really concentrate the proteins very closely, and we can increase the number of proteins we can actually print on the microarray. So that's a really good advantage, and we have done that. But with that, there is another drawback also with this technique also, because as you can see here, this protein is bound to this capture antibody, and as this protein is bound, it's surface immobilized, and as it is surface immobilized, it cannot rotate. So if there is an epitope which is this part, which is like close to the bottom, then the antibodies doesn't have that freedom to bind to that location because it's hindered, it's hidden. So that's why we thought of something new where the protein is not bounded anywhere and it's free in solution. So that's how we came to the next idea, is multiplexed in solution protein assay, which we called in short Mispart. So what is Mispart is basically it stands for that what we have is we have this plasmid DNA, and in the plasmid DNA we have the protein of interest, what protein suppose P53, and then we have two tags, tag protein, flag tag, and halo tag. These are the tag proteins which are commonly found, which is used to tag something so that we identify that, as the name suggests. And then we express in vitro protein expression, so we get the protein and then flag tag protein and then halo tag. And then we add a barcode. So a barcode is a DNA sequence which is very unique. So like barcode, what we have in our real life is it's a straight lines in adjacent straight lines. So the straight lines identifies as a unique thing. So in our biological experiments, we call this as a DNA barcode because the nucleotide identifies as a unique protein. So we can identify. We can barcode with different proteins. We can barcode with different sequence, and by that way, when we run the final PCR, we can identify, okay, this is our sample, this is this protein. So what we do is we add a DNA barcode to it, and then we bind this DNA barcode. And then what we add is we add anti-flag magnetic beads. These beads then bind to these beads having the anti-flag then binds to the flag tag. And then we add a magnet because these are magnetic beads. So we add a magnet and we wash away everything, all the unbounded DNA barcodes. And then we add a 3x flag tag type because this is high in concentration. So this removes this already bound thing. And now we have this DNA barcoded protein. So we have this protein which is DNA barcoded, and it is free in the solution with no hindrance or no blockage. So after that, once we have that, we add the serum samples, and from the serum samples, we have the same, the antibodies will bind to it. And then in the same way we do using protein G beads, this protein G beads actually binds to any immunoglobulins. And when we add that, it binds to the immunoglobulins. And similarly, we add magnetic beads, and then this magnet, we use the magnet to isolate this from the unbounded antibody in the solution. And this is how we found that at the end we run a PCR, or we do next gen sequencing to exactly identify which are the proteins which have antibodies and how much strong signals are there, those have. So based on that, we say, okay, so one person have this many antibodies, and we quantify that. So these are the different techniques, as I have told you, how we do using protein, protein microarrays and in solution protein assays. Both have their own advantage and disadvantages, but they are all very high throughput. And when we do that, we add both disease serum. So we add a serum from a person who has the disease, like suppose type 1 diabetes, and a person who is healthy, who doesn't have any disease. And then we check the antibody difference between these two, and which are the antibodies which are elevated in the disease serum. And then we do all the statistical analysis to check power analysis and other analysis to see whether there is a difference, in sensitivity and specificity, and then we come to the conclusion, okay, this is the antibody which can be which have a biological role. So with this, I'm going to talk a little bit in over slide through one of my recent paper I've been working. So this is more of a bioinformatics paper, and this is a meta-analysis of serum autoantibodium reveal common autoantibodies in healthy individuals. So I used the protein microarrays data, and what we found that this paper is now in Cell Reports for peer review. So what we found is actually these autoantibodies are a type of antibodies which affect our own body. So there are a lot of autoimmune diseases, and these days a lot of people are in India also, I know, with getting infected. More and more people are. Earlier, autoimmune diseases were specifically for Western countries. There was not much incidence in India, but nowadays with rheumatoid arthritis, type 1 diabetes, inflammatory bowel disease, to name a few, there are a lot of diseases coming up with higher incidence. People are getting more affected with these autoimmune diseases. And this is one of the main part of this autoimmune diseases, the autoantibodies. So autoantibodies, as the name suggests, is the antibodies which kills yourself, which targets your own body proteins. So that's the problem, because your immune system doesn't recognize that it's your own body, and it shouldn't attack that. And due to this mistake, the antibodies are generated which will affect your own body, and because of that reason, it leads to autoimmune diseases. So one of the common biological concept is clonal deletion, which all of you might have studied in your undergrad and grad school courses. So I'm going to go through this a little bit. So autoantibodies which target the immunoglobulins are called autoantibodies which target self proteins. So it is earlier believed that healthy individuals don't have autoantibodies due to clonal deletion. So what happens when your immune system matures, it tries to find the different proteins which belongs to your own body. And if there is a clone which matches with your self antigen, then that clone is deleted, that B cell clone is removed. And only the clones which doesn't have any match, which doesn't have any match with any human proteins, so those are secreted into your main body bloodstream. So, but then it has been, now it is found that it's not like black and white, it's more like a gray region is there. There are more antibodies which are actually targeting our own body, and they are found subsequently, they are found quite prevalently in our human body, which are who doesn't have any disease as well. So their presence can complicate the discovery of biomarkers, because if we find an antibody and it's actually one of the antibodies which is commonly found in our body, then that can complicate our discovery. So suppose we are trying to find an COVID coronavirus antibody, and we happen to find that this antibody is very common in human being without like any infection, then we have to keep in mind that that's a false positive, and we have to remove that from our analysis. So that is the reason why it's so important to understand which are the autoantibodies which are commonly found in human body. So as I have told you that this human genome varies so much as how we behave to medicines, how we behave as at a proteomics level. So that's why we need a huge study from different parts of the different subjects from different part of the globe. So what we did is we did a meta-analysis. So a meta-analysis is where we accumulate studies from different groups, and then we see what are the common things. So we had 9 studies we accumulated. These are mostly the samples are from different parts of USA and United Kingdom. And what we got is we got the samples of 272 healthy individuals, and we studied a total of 8282 unique proteins. So using this 8282 unique proteins and using 272 healthy individuals, we tried to see which are the antibodies, whether they occur commonly or they just occur randomly. And if they occur commonly, what is the mechanism behind it? And if they occur randomly, what is the mechanism? Why are they there? So we did a statistical model using weighted prevalence, and because these, as you can see, that some of these have a more proteins are studied in some of the studies, because this is a problem of meta-analysis, because you are not controlling these studies. So some people will have different protein sets, some people will have different sample size, so you have to find a way. So we used a statistical model of weighted prevalence. We gave weight to different studies based on like how many proteins they studied. So the number, if the proteins and the number of protein studies is higher and number of samples are higher, then we give more weight to that study. And using that we found, and in fact, we found that they are not random. So they actually occur very commonly in human body. So then we try to see what are the prevalence of these proteins and why they are there. So first of all, these are some of the antibodies we found to these proteins like STM, stathmin 4, ODF2, RBPJ, MY2. And we call this, and the weighted prevalence of like 0.47, 0.43. These are pretty high, like almost half of the population has antibodies to stathmin 4. And which is interesting, why do we have these antibodies even though we don't have any disease? So we looked into this. Anything higher than 10% weighted prevalence, we call them as common autoantigens. And we have a total of 77 common autoantibodies in all this meta-analysis. And what it did is we tried to understand if there is an effect of age and gender in this autoantibodies. So usually what happens, when we have our immune system develops something like that. So we have, when a kid is born, they get antibodies from their mother. So initially we don't have antibodies. Our immune system is very weak. So we get antibodies from our mother through placenta. And once we have those antibody, it declines very like in your early infancy. And then your antibodies, the antibody starts to, your immune system starts to develop. And then as we grow to adulthood, like adolescence, at the end of adolescence, our immune system fully matures and we have like fully matured immune system. And until the age of 60, 65, then again, our immune system starts to deplete. And that's when, because as you get older, you get more infected with diseases and you get more viruses and bacteria. So that's the reason. So we wanted to see how this autoantibodies also, does it get affected or not? And we also wanted to look into the gender bias, is there was a gender bias, because usually autoimmune diseases affect more females than males. The incidence is much more higher in females. That's because females have more hormonal changes. And that's because that is the reason why it has been found that females have more autoimmune diseases in their lifetime compared to males. So when we looked into our samples, we actually found that with age, we actually found similar trend as our immune system develops. So from 0 to 6, it was much lesser. This all dots represents an individual and the number of autoantibodies they had. And as you increase to 6 to 12 age, it increased. And then after that, there was no significant difference between 12 to 18 and 18 plus. So that shows that, well, we don't have autoantibodies during our birth at childhood, but this common autoantibodies grows as we grow older. But at 6 to 12, it almost gets matured, and we have something which stays constant throughout. And in literature also, it has been found that this common autoantibodies are very, it's like a fingerprinting. It's like we have very specific set of this common autoantibodies, and they don't change throughout our lifetime. It was done in some of the longitudinal studies, and they found that this is true. And then when you check the genders, males and females, it was surprising because we thought like females will have more autoantibodies, common autoantibodies, but actually found that both of them had very similar, and there was no significant difference. And this was also a bar chart population chart, and we found similar signals in both male and female. So one more thing we thought, because these antibodies are occurring in like both healthy and diseased cohort without any disease. So in healthy it's occurring, so they should occur at similar frequency in diseased cohort as well. And what I suspected, it was actually true, and we found that both in healthy cohorts as well as in diseased cohort, they had similar frequencies of this antibodies, and we had like an R value of 0.97 when we checked the correlation. So next, the question which intrigued us was what initiated these autoantibodies, why are they there, and what is the reason for that for their presence. So we hypothesize that they can occur because of two different reasons. It can be molecular mimicry, or it can be an intrinsic property. So by molecular mimicry, what I mean is it's as the name suggests, it's kind of a mimicry. So you mimic something, and that's what leads to this antibodies. So suppose we have in our body, there is a protein which is very similar to a protein in a virus. So our body, our immune system fails to detect if this is the same protein, or if this is a protein from a foreign antigen. And that's when an antibody which was originally created by our immune system for a foreign antigen, suppose for a virus, is actually now affecting our own human body. And we have now in literature, it has been found that there are so many diseases which occur because of this cross-reactive antibodies, which are due to molecular mimicry. And we also looked into the intrinsic properties. So an intrinsic property is basically something which is inbuilt in the protein. So something like if it is hydrophilicity, or is something, some property of the protein which makes it more antigenic, more immunogenic, and that leads to more antibody secretion in the body. So we looked into hydrophilicity, if it is more hydrophilic, and we looked into aromaticity, isoelectric point, flexibility, if there is more flexible or not, and then beta turns, beta sheet. These are all the properties of proteins, and we looked into all these different properties to see if there is anything going on. So what it did is, first of all, we checked using sequence similarity. The first one was, sorry. So the first one we checked was the sequence similarity. So the sequence similarity is what we did is we wanted to check, as we said, that we wanted to check the molecular mimicry. So the molecular mimicry is if there is some similarity between the virus protein and the human protein. So we took the virus proteome, the human virus proteome, and then we used a tool called CD-HIT. This tool actually removes all the duplicate proteins. So if there is more duplicate proteins, we have to run this huge bioinformatics queries. And if we can reduce the number of sequence, that helps a lot in processing the sequence analysis faster, as well as it helps us to like reduce the computational power. So we use the CD-HIT to, suppose there are two sequences, there are some variants, like for COVID, if you have two variants which are very similar in sequence, and there some proteins might be very similar, there is no difference. But while there is some like the spike protein might have few mutations which are different. So we don't want to have duplicate proteins. So we just remove those using CD-HIT. And then we had this viral proteome, and then we ran a Python script. I wrote a Python script to chop these sequences into 14 more peptides. So we have this 14 peptide sequence. So in a way that, because this long proteins can sometime, when you run the sequence similarity analysis, that can, the algorithms are not very good in checking all the sequences. So that's why if we chop this into different smaller sequences, it gives much more higher incidence of true positive and not false positive. And also on all the human proteins we divided into two different databases. One is common autoantigens, which we call common autoantigens, and then something which didn't have any reactivity, we divided into unreactive proteins. And then this is one of the inbuilt tool, which is Sigmasker, which tries to remove the low complexity regions, which is like amino acid repeats or the sequence which are commonly found in signal peptides. So those are removed, and then we ran a blast. And when we ran a blast, and then we checked the significance of difference between unreactive proteins and common autoantigens. So ideally, what should happen is that it should react more. There should be more matches with the virus protein with the common autoantigens, and very less with the unreactive. And we did a chi-square test to check the difference between the frequency of the matches between these two. And this pipeline actually was designed by me, and we tried to reduce the false positive, because when you have like huge databases with this human virus proteome, there is always chances of false positive, as you increase, because after all, it's all matter of chance. So as you increase the size of the database, so that increases the chance of false positive. So that's why we use this tool to to reduce the false positives. And what I found is actually the Chisquare returned a very good result, and we saw that actually there was significant difference. And what I found among our 77 common autoantigens, we had like 21 of them had one or more sequence similarity with one of the common human viruses. And as you can see here, these are like 7 or 8 amino acid matches. So this is not just with random chances. It might be, but there are very less chances because these sequences are very different from one another. There is no common sequences, and they also have very like 6 to 7 the antibody epitopes are usually varies from 6 amino acid to 12 amino acid. So a 7 or 8 amino acid match is likely to elicit an antibody response. So this is something is interesting, and it shows that, well, viruses can indeed can lead to autoantibody secretion, and it can lead to autoimmune diseases. The next thing we checked is the intrinsic properties, and for that I used an BioPython, the package of BioPython. And what I did is I calculated the values of all the different values of the different physical properties of a protein. So I had like protein 1 to 8,200, 8,282 proteins. I checked all the properties like aromaticity. I calculated the hydrophobicity, everything. And then I ran gene set enrichment analysis. And this is a software which is developed by UC San Diego and Broad Institute at Harvard. So this software, what it does is it tries to check if there is an enrichment. And this software is very useful. It's not just in like in biology or bioinformatics. It can be used in chemistry or any other like botany or any other field. So it basically checks for enrichment, if there is any enrichment or not. So what it does is it actually arranges all the values based on decreasing order, and it tries to see if the proteins you are trying to look, whether they occur at one end or not. So if they occur at one site, so that means they are enriched. If they occur at different parts of the different parts of this order, a different part of this decreasing order, that means they are like all dispersed everywhere. So that means they are not enriched. So if you have like something which is all are high values or all are low values, that shows that there has been an enrichment. And if it's all dispersed, means there is no enrichment. So we wanted to see if there is an enrichment of any properties or not. So what we got is we got some of the good results for different physical properties. So for instance, this is the aromaticity. So this graph, these are generated from the GSEA. So if you see here, the normalized enrichment score is 1.7, and then the p-value is less than 0.01, which is significant. So it shows that I have put here all the significant values here, and then there were some like protein length and like beta sheets. Those didn't lead to any enrichment, so I didn't put it here. So you can see here, it can be explained that a protein which is more aromatic, which is less aromatic, and then which is I can see it. So a protein which is more isoelectric point, and then number of amino acid in beta turns, and then flexibility, hydrophilicity, and beta turns lead to more immunogenic protein. So it's logical also, because if a protein has beta turns, so in beta turns the protein takes a turn, so that part is more immunogenic, that leads to more antibody binding. So also like hydrophilicity is one of the things which is very important, because the protein needs to be it's in the blood, so it needs to be hydrophilic. If it is hydrophobic, it can't stay in the blood, so it needs to be a good antigen, or the good immunogen needs to be hydrophilic in nature. And flexibility is something is also very important, because if we have more flexible protein, that is more likely to elicit an antibody response, because the antibody can bind to it. But if it is very stiff, the antibody match will not, it will be very difficult, and it might not find a match. And then same, this is another technique, which is Chow and Fussman beta turn, and this is also, we found some of the good signals here also. So this shows that, with this I want to conclude this this chapter, is that we have these antibodies, which are autoantibodies, which are commonly found in healthy individuals. You, me, everybody in like 50% of the population have it, and we don't know why is it. So we did this exploratory study and we tried to find why are they there, and then we postulated two different hypotheses. That one is either it's because of molecular mimicry with the virus, or it's because of like intrinsic property of the protein itself. And we found the evidence of both of them might be true. There are evidence of both of them. So there can be both molecular mimicry playing as well as the intrinsic property playing the role, which leads to the elicitation of these antibodies. So with this, I'd like to conclude that these are commonly found in healthy individuals, and age and gender has effect on this autoantibodies. In age, we see as we grow older, it increases, but then it plateaus, it doesn't go any high. And then molecular mimicry might play a role in this elicitation. And these autoantigens were enriched for various intrinsic properties. So with this, I'd like to thank my team. I'd like to introduce to my team Dr. Joshua Lever. He is the director of Biodesign Institute. So he's my PI. And then Dr. Ji Chiu, he is a proteomics specialist. And Dr. Yinru Chung is the statistician. And Dr. Jean Park is bioinformatician. And Dr. Ellison, she is our collaborator at University of Florida. And Dr. Luxing Song, he is our research scientist at our lab. And especially this project was funded by NIH, and we had samples from Mayo Clinic. So I'd like to thank NIH, Mayo Clinic, and AAC for giving the opportunity, and University of Florida for the collaboration with us and giving all the input and insight into it. And finally, I am, if you want to collaborate or if you have any questions and want to have more, anything you want to discuss, you can get me over this email and also on Twitter. So with this, I'd like to complete my presentation, and I don't know how much time is left. So I'm ready to take questions from the audience.
Yeah, thank you very much, Dr. Shom, for a nice presentation. It was really wonderful, and it was also very useful to all the participants, because now we all are talking about antibodies due to the COVID pandemic and all of that.
Yeah, that's what I thought. It will be interesting because nowadays more and more people are getting educating with, like, people know what is like antibody, what is virus, what is immunity, and this will give a little bit understanding to the people.
Yeah. Thank you, Shom, Dr. Shom. So in his lecture, Dr. Shem have you research which was regarding the factors affecting antibodies, as well as he has also discussed meta-analysis of different studies. He has also given information regarding the antibodies as well as the autoantibodies and the clonal deletion and all other research, which was very much interesting. Dear participants, now the session is open for the discussion. You can ask one or two questions to Dr. Shom. So you can directly ask by unmuting yourself, or you can directly write in the chat box.
Nice presentation, Dr. Umashish.
Yeah, I can't. Okay, I'll just stop share. Yeah. Thank you.
So I have a question. Nice presentation, well done. It's
Yeah.
good research, and you're directly involved in the global cause these days is COVID. So congratulations. I wonder whether the COVID virus has more capacity to mimic. Is it true, or is still under investigation?
So COVID has more, it has more mutation rate. So because so we are seeing like a lot of variants coming, because it's trying to find different variants. Because as we are targeting this, like the vaccines we are producing, like the mRNA vaccines by here in US we have Pfizer, Johnson & Johnson, and then Moderna. All of them have this mRNA vaccines. So these vaccines are actually targeting the spike protein. And what this COVID virus is doing is it's trying to mutate this this spike protein. So the spike protein is basically where it binds to our cells and that injects its genetic material into our human cells. So the spike protein has it's very specific, and it's very very strong binding with our ACE2 receptor. So that's why they are like, this virus is very intelligent, and it's trying to, like, as we are finding more and more vaccines, like it's trying to evade it, and it's trying to find its more different mutations to like evade this vaccine. So with this, if you see, like even I was double vaccinated, but during this Omicron variant, I got infected and I was positive. So this Omicron was like, it found a way to, you know, like mutate itself in a way that it didn't affected by the vaccine we had already. So it's a more like, it will be continuously going on mutating, like we have influenza virus, like we get a virus shot, influenza shot, flu shot every season. So it will be more like that. But the death rate and also the the seriousness of the disease has gone down because we have more people vaccinated.
I see. Great. Thank you.
Thank you, Suhas, sir, for asking a nice question. It was in the mind of all the participants. And thank you, Mahashish, for giving a brilliant answer.
Thank you, Dr.
Thank you.
Pratap. Yeah. Thank you, Dr. Rupali Shinde.
No,
Yeah. Sorry.
Actually, sorry, I was using my wife's account.
Yeah. I saw that it
her
was
laptop.
flickering, I think. Yeah.
Yeah. No problem. Thank you.
So, any other question from other participants? Okay. Thank you, Mahashish. It is late night at USA, so
yeah, it's already.
Thank you very much, Mahashish, for giving time for the conference, as well as for giving a very brilliant session in the conference. Thank you.
Thank you.
Okay, so dear participants, now it is time for the third session, and this session will be conducted by Dr. Yakubu Bobe. He is working at Federal College in Zaria, Africa, and it is very early in the morning in Africa. So therefore, he woke up early in the morning for our conference. So I would like to invite my colleague, Chandaya sir, to give a brief introduction of Yakut, Dr. Yakut, and then Yakut will start his presentation. Chandaya sir, over to you.
Yes, thank you, sir. Thank you, sir. It gives us an image to introduce Yakub Bobai. Currently, he is working as a head of the Department of Geography in Zaria. So, sir has a lot of Sir has published many research papers, around 11 research papers in various journals. Sir has completed his PhD degree in physical geography in 2019. We have very grateful to sir. Also, he wrote some chapters in published books. He attended many conferences and seminars around the world. He also, apart from this, he is member of some community service cells. He is a member of Nigerian Red Cross Society. He is also a member of academic staff. He is also a treasurer of College of Education Staff Sports Association Nigeria. He is a coordinator of Youth Wing Kagaru Development Association. He is also a member of Red Dress Code Committee. He is also a member of Ceremonial Committee FCE Zaria, member of Welfare Committee School of Arts and Social Sciences Zaria. He is also a patron of National Association of Geography Students FCE Zaria. He is also a member of School Patron Secondary of School of Secondary Arts and Science Students Association FCE Zaria. He is also a member of Kadua State University Students Zaria, member of College Research Committee. Currently, he is working as a HOD of Geography in Zaria College. So, it is our honor to have us, Sir Yakubu Bobai. We very much thankful to Sir, and I request Dr. Naikwade sir to please continue. Thank you, sir.
Thank you, Chandaya sir. So, Dr. Yakut, can you hear me? Dr. Yakut? Yeah, please unmute yourself. Yeah. Yeah, very good to see you, Dr. Yakut. Dr. Yakut is also my good friend, and today he will going to discuss challenges of environment sustainability in the developing world. So, Dr. Yakut, you have 35 minutes for the session, and yeah, we are very eager to learn from you the environment sustainability challenges as well as your research regarding that. So, Dr. Yakut, would you please unmute yourself and then start your session? We cannot hear you. You have to unmute first. Yeah. Yeah.
Okay, are you with me now?
Yeah. Yes. Pretty good.
Okay. Thank you, Doctor, and good morning. I'm glad to join you from Zaria, Kaduna State, Nigeria. And like you have actually noted, the title of my paper is "The Challenges to Environmental Sustainability in the Developing World." Of course, I take Nigeria as a major developing nation. India, incidentally, is one of the developing nations of the world. So we probably share similar challenges. And I tied my title to the theme, the major theme of the conference. And let me state clearly here that my paper is actually a review paper. It's not an empirical paper. But then by way of introduction, the environment provides us with many resources people use in their daily lives. And the environment is used to grow crops, obtain water, harvest wood, mine for minerals, and produce energy. However, when we overuse the environment, the resources become depleted. It limits our ability to use them in the future. Now, I would start by defining quite a number of concepts. I would want to talk about the environment, define development, sustainable development, and environmental sustainability. Now, by way of definition, the environment refers to both physical and social circumstances which surround people and have influence on them. It includes water bodies and the life within it, the landmass, forests, grasslands, deserts, animals, and the interactions that take place within them. That is the environment. Now, development. Development is a process that creates growth, progress, positive change, or the addition of physical, economic, environmental, social, and demographic components. That definition is by the Society for International Development, 2021. Now, the purpose of development is a rise in the level and quality of life of the population and the creation or expansion of local regional income and employment opportunities without damaging the resources of the environment. Development is visible and useful, not necessarily immediately. And it includes an aspect of quality change and the creation of conditions for continuation of that change. Now, in development, there needs to be a positive change. And the positive change needs to be sustained. We need to do anything, whatever we can, to sustain, to maintain the positive change. That is when development now comes into being. Development is visible and useful, not necessarily immediately. And in line with the current global agenda, development is about sustainability. That is, development that meets the needs of the present without compromising the ability of future generations to meet their own needs. Of course, that's a very popular definition, quoted from our common future, the report of the Borderland Commission by the United Nations in 1987. Now, we would not, I would also want to talk about sustainable development. There's a definition by the World Commission on Environment and Development, which defines sustainable development as a development that meets the needs of the present without compromising the ability of future generations to meet their own needs. That is sustainable development. Now, theoretically, the long-term result of environmental degradation would result in local environments that are no longer able to sustain human populations to any degree. Such degradation on a global scale would, if not addressed, mean extinction for humanity. In the short term, environmental degradation leads to declining standards of living, extinction of large numbers of species, health problems in the human population, conflicts. Sometimes they could be violent between groups, fighting for dwindling resources, water scarcity, and all of this, they are evident in some parts of Nigeria, particularly the Niger Delta region of Nigeria. Environmental sustainability. When we look at our natural environment, we see that it has a rather remarkable ability to rejuvenate itself. For instance, when a tree falls, it decomposes, it adds value or nutrients to the soil. And these nutrients that are added to the soil help sustain suitable conditions for future saplings to grow. Now, when nature is left alone, it has a tremendous ability to care for itself. Nature has the ability to take care of itself. However, when man enters into the picture and uses many of the natural resources provided by the environment, things change. Man coming into the environment changes the settings. Nature left on its own has the ability to cater for itself, to take care of itself. Human actions can deplete natural resources, and without the application of environmental sustainability methods, long-term viability can be compromised. If we do not do anything now to use the environmental sustainability, then long-term viability of the environment is what is being compromised. Environmental sustainability has three pillars. There are three pillars of when we're talking about environmental sustainability. They are social sustainability, economic sustainability, and ecological sustainability. The economic sustainability aims at ensuring economic prosperity and the preservation of environmental assets. From an economic standpoint, sustainability requires that current economic activity not disproportionately burden future generations. Whatever we would do now for economic gains, it should not be disproportionate to the burden of future generations. We should be mindful of whatever economic activities we embark upon. Mining is an economic activity. Fuel harvesting is an economic activity. The extractive industry, industrialization, they are all economic activities. The extent to which we carry them out determines what becomes of our future. Economic sustainability should involve analysis to minimize the social cost of meeting standards for protecting environmental assets, but not for determining what those standards should be. That is economic sustainability. Social sustainability aims at ensuring all people, now and in the future, have their basic needs met. All people, now and in the future, they should have their basic needs met. So in social responsibility, a positive condition within communities and a process within communities that can achieve that condition. The definition is supplemented with a list of corresponding principles. What are the principles guiding social sustainability? They include equity of access to key services. We should have access to key services, all people, not some people. Equity between generations. Equity, no generation should be shortchanged. Now, a system of relations valuing diverse cultures. Okay, diverse cultures should be considered, their needs, their livelihoods should all be valued. Political participation of citizens, particularly at local level, community ownership, a system for transmitting awareness of social sustainability. Okay, all of this, they are some of the guiding principles for social sustainability. Now, for environmental sustainability, it ensures that non-human elements of nature, the non-human elements of nature like air, water, trees, and wildlife remain healthy over a long period of time. Environmental sustainability, therefore, means meeting the resource and services needs of current and future generations without compromising the health of the ecosystems that provide them. That definition also is from our common future, from the Borderland Commission in 1987. Now, I have here listed some of the benefits of environmental sustainability. By the time we are able to attain or we're able to maintain environmental sustainability, some of the benefits include our ability to protect our ecosystems. We will continue to enjoy nature as it is. We can protect endangered species. There will be less pollution in our towns, in our cities, and that will result in better respiratory health conditions for all. It could halt or reverse the effects of global warming. We won't be creating huge landfills all over the world that will take hundreds of years to break down. Now, these are some of the benefits of environmental sustainability. So what I have done so far is to look at the major concepts of this theme, which has to do with the environment. I've defined the environment. I've also been able to talk about sustainability, development, and environmental sustainability. Now, the major crux of this paper today is for us to see the challenges of environmental sustainability in the developing nation, with emphasis on my dear country, Nigeria. And I'm sure some of the issues raised here, they are very similar to some of the challenges to environmental sustainability in India. India is equally a developing nation. And one of the challenges of environmental sustainability in Nigeria is the extractive industry or mineral exploitation. Nigeria is endowed, Nigeria is blessed with abundant natural resources, minerals. Basically, our major source of income is from the oil industry. Nigeria is one of the major exporters of petroleum products worldwide. The removal of a non-renewable resource, such as crude oil, causes some environmental problems. For indigenous people, it has effects on the local flora and fauna. And their subsistence depends on this, on the water bodies, on the fishes, on the vegetation around them. Oil exploration, oil exploitation damages, it has negative impact on these communities. The extraction of oil is bound to have some negative impacts on the Niger Delta environment. Many communities in the Niger Delta believe that local gas flares causes acid rain. A lot of gas flaring is taking place in the Niger Delta region of Nigeria. We flare gas. We do not have the capacity, the ability to tap the natural gas, liquefy it, and use it as a source of domestic energy. Rather, that gas is being burnt off. It is being flared. Now, it pumps tons and tons of carbon dioxide into the atmosphere. It affects the vegetation of the area. It causes acid rain. During the rainy season, you find atmospheric carbon dioxide, atmospheric oxides combining with water, falling down as acid rain. It has effect on the vegetation around them. It has effects on the roofing sheets, and it contaminates the water bodies. The toxic substances which have been emitted into the flares for over 40 years, including benzene and particulates, have exposed Niger Delta communities to health risks and property damage. And this violates their fundamental human rights. The flares affect their livelihood and expose them to an increased risk of premature deaths, child respiratory illnesses, asthma, cancer, as well as acid rain. Okay, now these are the issues I have raised. The Niger Delta, and that is why they have been agitating. That is why there have been unrest there. Not only does gas flaring affect them, oil spillage has tremendous effect on the water bodies, on the vegetation around them. It is a delta. Okay, the River Niger Delta. The area is swampy. There's a lot of water around, but because of the oil, they do not have access to potable water. And even though oil mining corporations there do not help matters, they do not clean up the environment. They do not provide infrastructure to the locals, do not provide hospitals, do not provide them with potable water, boreholes, or any other alternative sources of water. And that causes a lot of unrest, a lot of unrest. Corruption. Corruption is one other challenge we are facing. And how does corruption affect sustainable development? Corruption impedes development. It minimizes the capability of government to reduce poverty. In Nigeria, corruption has been at the center of development, and it is an impediment to true and authentic development in our society. Corruption has automatically ravaged the entire Nigerian system, thereby making other nations of the world to perceive us as the most corrupt nation in the world. Transparency International, in its 2021 report, actually, out of 189 countries, Nigeria is ranked as the 154th in terms of level of corruption. So you could see that Nigeria, the level of corruption is very, very high in our country. And corruption in the country's political and bureaucratic cycle often takes the form of corruption manifests itself in various forms in our country. One, gratifications, where you asked to grease a person's palm. If you need a contract, you need to give a percentage to the contractor, to the person issuing the contract. You need to issue a percentage, gratification, inducements, embezzlement of funds, conflict of interest, influence peddling, padding of budget. Imagine lawmakers padding budgets, inflating budgetary allocations, and what the funds are being budgeted for would not even be executed. They will not be implemented. Padding of budget, extortion, fraud, bribery, misappropriation, falsification of financial records. All these are acts perpetrated by some public office holders, and they are inimical to the implementation of what? To the attainment of environmental sustainability. Now, overt and covert corruption in government institutions and amongst key bureaucrats has contributed greatly to hunger and deprivation due to failure in the implementation of key policies on poverty alleviation. Now, what are we talking about? Corruption has prevented us from implementing various policies. When it comes to policies, Nigeria is endowed with the intellectual. A lot of policies that are not being implemented. Why? Corruption. Okay.
Dr. Yakut?
Yes.
Yeah, sorry to interrupt you. Yeah, it is very thought-provoking. You are discussing about all the issues which are regarding the sustainable development and sustainability of the environment. Yeah, but due to time constraint, you have 5 minutes more. So you have to conclude within 5 minutes.
Okay, sorry. So thank you very much. I will try to round up in 5 minutes. Now, so we have the issue of corruption there affecting environmental sustainability. Now, poverty. Poverty is also factor, is another factor that affects environmental sustainability. It is the bane of growth in our country. And how does poverty affect? Poor people are being forced to depend on the environment because they cannot afford major sources of energy, electricity, gas, petrol. They are being forced to go for alternative sources of energy. And these alternative sources of energy are coal, fuel wood, animal dung. Now they rely solely on the environment. Because they are poor, there's pressure on the environment. And the environment in turn is not being sustained. So it affects them, making them poorer. So it is a vicious cycle between environmental degradation and poverty. So poverty is one other factor affecting environmental degradation. Insecurity. Nigeria is facing a lot of security challenges. You over there in India, you're very much aware of the Boko Haram issues we're having in the northeastern part of this country. Boko Haram is a religious sect. They are armed, they are linked with ISIS, and they affect farmers. You do not go to the farm. We also have the issue of bandits around us. Bandits are armed Fulani herdsmen. They are all over the place. Farmers can no longer go to the farm. People who are depending on the extractive industry cannot go to, do not have access to their farms. They cannot go for fuel wood. They cannot go for mining. So they are that makes them poorer. It makes them to depend more on the environment, weakening its ability to revive, to resuscitate itself, thereby affecting sustainability. There are different ways to use the environment to attain sustainability. We can switch to renewable energy, such as solar, wind, hydroelectricity, and biomass. We can begin to recycle metals, such as iron and steel, and minerals. We can raise the fuel standards of vehicles, reduce greenhouse gas emission, and environmental education. All that should be circled around sustainability. Conclusion: sustainable development is called to improve the quality of life for global citizenry without increasing the use of our natural resources beyond the Earth's carrying capacity. The interdependencies of the economic, social, and environmental elements of our planet requires us to think critically and strategically about issues and taking actions that will truly create a future where human society and nature coexist with mutual benefits, and where the suffering caused by poverty and natural resources abuse is eliminated. I want to thank you for your time. Thank you for listening.
Yeah, thank you very much, Dr. Yakut. You have discussed very related things which are related to the environment as well as the sustainability, as well as you have discussed the problems not of the developing countries like Nigeria, but there are the problems of most of the other developing countries like India also. So in generally in the world, globally, we can see that, we can observe that generally the voice of people from the developing country, it is ignored. And so it is a must, or it is a mandatory to give voice to all the people from the developing as well as the developed countries. And that is the reason that in our conference, we have given a representation of all 5 continents. So the resource person as well as the participants, they are from Europe, America, that North America, then Asia, Australia, and of course Africa. So thank you very much, Dr. Yakut, for your thought-provoking lecture. And now session is open for the discussion. So dear participants, if you want to ask any question regarding the topic of Dr. Yakut, then you can directly ask him by unmuting yourself, or you can write it in the chat session. Yeah. Thank you, Dr. Yakut. Your lecture, it was so self-explanatory that I think our participants don't have any queries regarding that. So thank you very much, Dr. Yakut, for accepting our invitation as well as for such a nice lecture. Thank you.
Thank you for making me part of this conference, and I will always be available for future conferences. Thank you.
Yes. So dear participants, let's move to our next session, that is poster as well as the oral presentation of abstract number 51 to 134. So earlier we have given one thing that what happened that yesterday, we start at around 1:30 and we have to complete nearly 50 abstracts presentation, and it took nearly 5:45 to complete. And due to that, we have decided to have breakout room, use of the breakout room for the presentation of today's papers. I'm not sure that whether this breakout room feature will be useful to you, or whether you are familiar to this feature, because some of the participants, they have told me that they have not used breakout room feature earlier. Yeah, but let's try, and if it is not going to work, then we will start as regular as we have completed in the yesterday's session. In yesterday's, that is very first day of our conference. So dear participants, what we have to do that our host Tanmay Manjrekar from Srishti will made 2 breakout rooms. One will be for the presentations of 51 to 90. And then the second breakout room will be for the presentation of 91 to 134. And dear participants, the actual schedule as well as your abstract number has already been shared to you in WhatsApp as well as Telegram group and on email. So you know very well that which one is your presentation number, and then you have to join your respective breakout room. And then we have two chairman at both the session. In breakout room first, my friend Dr. Mandar Zaki will be the chairman for the session, and for the breakout room, that means from abstract number 91 to 134, my friend Dr. Ranjit Bansode will be the chairman of the session, and they will continue with the presentation. Then another thing, the most important thing, dear participant, it is that as we have to complete today nearly 100 presentations, and so therefore we have time constraint, and so therefore you all are requested to complete your presentation within 5 or 6 minutes. So dear participants, let's start. Tanmay. Hello, Tanmay, are you here?
Yes, sir. Yes, sir.
So you can give options.
Yeah, I'm preparing breakout rooms. Once I prepared, I will open for all to get in.
Yeah.
Excuse me, sir. Means you are providing the separate link for that.
No. The link here, the link is the same. Here, all the participants have already joined. They will have the option to choose the breakout room. So within 2 minutes, we will have option.
Okay.
Yeah, and then we will be there. And if anyone want to leave the breakout room, then he can leave the breakout room, and then he will be in the main session where I am there. Yeah. Just a minute, Tanmay will.
Sir, what about the YouTube link? Will it be same for both, or how does it work? Because there are few teachers who would like to see your presentation as well. So
yeah, but this is the first time that we are trying this one, so maybe.
Okay, sir. No, just confirming, sir, because like I'm sharing my paper with my guide. So she is obviously at home and she would like to see the presentation. So confirming. If possible, do let me know. Otherwise, I'll inform her it's not possible. Okay, sir.
Yeah,
That's okay.
definitely, the YouTube live will be there, but maybe sometimes it will be for the breakout room 1, and for sometimes it will be for the breakout.
Okay, sir. No problem, sir.
Just a minute. So
Yeah, sir, I prepared the breakout rooms. So according to the abstract numbers, everyone can able to join the breakout rooms.
dear friends, you can see 2 breakout room here. So you have to click on the breakout room and see all rooms. So you can see 2 breakout room. One it is for the group number 1, and another one it is for the group number 2 actually, but here it is written by mistaken. Tanmay, can you correct it? So it would be the group number 2 with the abstract 91 to 134. So those who have not joined their respective breakout room, so they can join. Yeah, if you have any query, then you can directly ask me.
Sir, my presentation number is 62. I can stay here in this group, sir.
Yeah, dear friends, you can join yourself, because we cannot see. Yeah, just a minute. You can see on the screen, you can see the different logos, and here one it is participant, second chart, share screen, record, live transcript, and breakout room. So in the breakout room, when you have to click on the breakout room, as soon as you can see the breakout room, you have to choose. So yeah, then you can see room, and then you have to join the room.
Sir, is it compulsory to join in the breakout room, sir?
Yes, it is. Yeah, let's try, because many of the participants, they are not joining. Maybe they are not familiar to this session breakout. So we will, dear participants, let's try. If it is not going to work, then we will have our regular session. Just a minute. Now I can see that in group number 1, 15 participants have joined, and in group number 2, 7 participants have joined. Hurry up, because all breakout rooms will be closed within 2 minutes. I can see now 14 participants have joined group number 1, and 7 participants have joined group number 2.
Good morning, sir.
Yeah.
ब्रेकआउट रूम कसं जॉईन करायचं?
बघा, खाली ब्रेकआउट रूमचे 4 दिसतात आपल्याला चौकोन, तर त्याला क्लिक करा. तुम्ही ज्या वेळेला क्लिक कराल, तर त्या वेळेला क्लिक केल्यानंतर तुम्हाला दिसतील 2 रूम. एक आहे ग्रुप नंबर 1 आणि दुसरा आहे ग्रुप नंबर 2. तर तुम्हाला ज्या ग्रुपला जॉईन करायचं,
क्लिक कुठे करायचं सर?
बघा, आता ज्या वेळेला तुम्ही जाल, तर त्यावेळेला ग्रुप नंबर 1 ला क्लिक केलं की तुम्ही तिथे जॉईन व्हाल.
तिथे खाली काहीच ऑप्शन दिसत नाही. पार्टिसिपंट, चॅट, शेअर स्क्रीन, रेकॉर्ड, लाईव्ह ट्रान्सक्रिप्ट, रिॲक्शन्स अँड ऍप्स.
नाही, ब्रेकआउट रूमला क्लिक केल्यानंतर एक दुसरी विंडो ओपन होते मॅम, तिथे जायचं आहे.
ब्रेकआउट रूम ऑप्शनच दिसत नाही.
So there is no option regarding the breakout room.
Yes, sir.
On the present screen.
Yeah.
ऑप्शनच नाहीये सर.
Ma'am, whether you are using your mobile or laptop.
मैडम खाली है, खाली ऑप्शन है.
No.
Till now only 14 and 7. That means nearly about 25 have joined, and 77 they are unassigned. So I am a little bit worried that whether this will be useful to all the participants, because only 21 have joined the 2 respective breakout rooms. So Tanmay, can you hear me?
Yeah, yes, sir.
Yeah, so I think it is good to drop the use of idea of the breakout room.
Okay. So many participants are not familiar to this technology.
And as we are living in a democratic country, most of the majority carries the law. So let's drop the idea. So you can.
I will close all the rooms.
Then we close all.
Yeah, it's closing. It's closing.
Mandar sir, are you there? Mandar sir.
Excuse me, sir. I think that breakout rule is not working here because after 120 seconds that returns to the main menu.
Yes, sir. Yeah, as a majority of the participants, they are not familiar to breakout room, so we will start with the regular move. So just 5 minutes more, so that all the participants who have already joined the breakout room can come to the main session.
Sir, it would be better to post the another link of the Zoom and you can say to join the remaining part.
Yeah, it will be one idea, but yeah, it is very difficult to rejoin, isn't it? Yeah, so let's wait for a few more minutes, couple of minutes, and then we will try the another option also. Just a minute. Huh? We will wait for 5 minutes. So Mandar sir, Dr. Mandar as well as Dr. Ranjit has joined the session, and they will be chairman for the span of hashtag number 51 to 90. Dr. Mandar will be the chairman, and from there, that means from 91 to 134, Dr. Ranjit will be chairman for the next part of the paper presentation. So still, what happened that some of the participants, they are not able to join the breakout room, and now the feature is some of the participants have already joined. They don't know how to come or how to leave the breakout room. So yeah, let's wait for 2 minutes more, and then we'll start with our regular session. Tanmay, would you go to the breakout room and tell the participants to leave the room?
I closed both the breakout rooms.
Okay.
Please check whether it's closed or not.
Sir, we are going to breakout room, but it is again taking us to main. This.
Yeah, leave it, sir. Leave it, sir. We are not using breakout room now. It is not needed to go to the breakout room. So I will also share this message in the chat room.
Good afternoon, sir.
A very good afternoon, sir.
Is it the same link or another link, sir?
No. Yeah, we are not using breakout room. Stay here in the main.
Okay, sir. Sir, which time I will present my paper?
Yeah, you have to be
90, 94, 93. Sorry.
Yeah, maybe after 1 hour.
Okay.
I'm not sure about that. Yeah, because some of the participants, they are not able to present, and yeah, so let's be here and let's also. It is a very good thing that we are a good listener, and we are also learning many things from the other researchers also.
Okay, sir.
Okay, dear friends, welcome you again, and then Dr. Manda, can you hear me?
Yes, sir. Very clearly.
Yeah.
I'm audible?
Yes. Dear participants, Dr. Mandar has expertise in various fields, as well as he has a thorough knowledge of all the fields which are related to the environment, sustainability, and all other issues also. So I would like to request Dr. Mandar to work as a chairman for the session, and now he will call the abstract numbers as well as the presenters, and then you have to present your work here. But very strict instruction to all the participants is that you have to complete your presentation within 5 minutes, and 1 more minute will be reserved for any question answer session. So Mandar sir, it is over to you.
Yes, sir. Thank you, Dr. Pratap. Without wasting time, I will directly go to the presentations. I will call the numbers, or I will read the title of the presentation one by one, and whoever will be there attending should go for the presentation. After 5 minutes, I will not actually ring the bell, but I will tell the to stop. And after that, 1 minute will be reserved for, as Dr. Naikar said, will be reserved for the question and answer session. So we'll start from the 51 in the row. Role of earthworms in soil fertility from Satana region, district Nashik, a preliminary study. Bawa R. Mayuri and Siddhartha D. Pagare. Is there anyone? Bawa
Yes,
R.
sir.
Mayuri and
Yes, sir, I'm present.
yeah. Please go on presenting.
Sir, screen is visible.
Mayuri,
Yeah.
your voice is breaking. Maybe you are joined from one from maybe mobile or maybe one from laptop. So you have to mute voice of one instrument and then you can speak. Yeah.
Am I audible, sir?
Yeah, clear.
Screen is visible, sir.
Yeah. We can see your screen.
Hello.
It would be better if we can see your full screen.
Yes, sir, full screen is present. Hello, good afternoon, everyone. Myself, Mayuri Ramanlal Bawa, a postgraduate student of Department of Zoology from Science College, Satana, District Nashik. Today, I am here to present you about my research topic, that is the role of earthworm in soil fertility. I have obtained the guidance for this study from the head of our department, Dr. Siddharth Pagare, sir. Now I will explain you what is the main aim of our study. That means to study the physical and chemical parameters, primary nutrients of red soils and black soil with the earthworm node. Then next, why I choose this project? Because in today's climate change era, the food security is very much concerned, and to eradicate this problem, a healthy and fertile soil is required to fetch more yields and the production in crop fields, and to achieve this in a sustainable way. One of main solution is use of the earthworm is soil fertility. Next slide shows the figure one shows the physical parameters of red and black soil. The physical parameters are namely density and water holding capacity. Density shows the gram per ml and water holding capacity shows the percentage. The density of the black soil and red soil is near about both these values, and the water holding capacity red soil is greater than the black soil. Figure 2 shows the chemical parameters in both red and black soil. In pH in black soil and the pH in red soil, both the soils are near about this equal. Electric conductivity in millimoles per centimeter in the black soil is higher greatly than the red soil. Percentage in organic carbon, the black soil and red soil, both these soils are near about equal, and the calcium carbonate, the black soil is higherly greater than the red soil. Figure 3 shows the primary nutrients of black and red soil. The main primary nutrients are nitrogen, phosphorus, potassium. Now, in the nitrogen in black soil is highly greater than the red soil. Then next, phosphorus in both the soils are near about the equal, and the sulfur and the nitrogen, potassium, black soil is highly greater than the red soil. Then next as the results obtained from the secondary nutrients in black and red soil. Secondary nutrients are namely percentage in calcium, magnesium, and the sulfur. Calcium in black soil, both the soils near about black soil is highly greater than the red soil. In magnesium, both the soils near about the equal, but in sulfur, the black soil is lower than the red soil. The next slide shows the micronutrients of red and black soil. The micronutrients are ferrous, manganese, zinc, copper. The micronutrients ferrous, manganese, zinc are greater in black soil than the red soil, while copper is more red soil than the black soil. Conclusion of our study: from above observation, From above observation, the results obtained from the analysis of both the soils, we can conclude that the earthworm significantly increases the fertility of soils in the both soils. Earthworms are important part of soil ecosystem and they improve soil structure by enhancing its chemical and biological properties. Acknowledgement of our study. Authors are thankful to our college principal R.D. Patil sir and our head of Department of Zoology for providing me necessary facilities during the research work. I am also thankful to lab assistant Mr. Sachin Aire for providing me earthworms, and I am also thankful to my brother for moral support during this research work. Thank you all of you.
Thank you, Ms. Mayuri. The session is open for the question. For 1 minute, please ask anyone if anyone want to ask a question or raise a question. Is there anyone?
Sir, please repeat the conclusion.
Ma'am, please repeat the conclusion. Repeat your conclusion.
Yes, sir. From above observation, sir, may I visible? May I audible, sir?
Yeah.
From
Yeah,
above,
Yeah. Please continue.
From above observation
ahead,
and the results obtained from analysis of both the soils, it is evident and we can conclude that the earthworm significantly increases the fertility of both the soils. From above study, it can be concluded that the earthworms are important part of our soil ecosystem and they can improve soil structure by enhancing its chemical and biological properties.
Okay, which species of earthworm you have used?
What, sir?
Sir, your voice is breaking.
Which species of earthworm you have used for the
So, species name is a Ferentima postuma. It is easily available in Southeast Asia.
Okay.
Okay. Are you satisfied with the answer, sir?
Yes, sir. Yes.
Yeah. Okay.
Thank you, sir. Thank you for my
Yes.
listening my
Thank you, ma'am.
presentation patiently.
Yes. Thank you. Next one is 52. Shefali Chavan, Megha Devgare, Anushri Pawar, Ashish Gele, Selina Fernandez. Is there anyone of the
Mandar sir, they have submitted video because due to some technical problem, it was not possible for them. So I will play the video. For that, I will share the screen, and then you can watch the video. So just a minute, I am going to share the screen here.
More, Swapnajat Devre, and Sudhin?
Yeah, Mandar sir.
Yeah.
Mandar sir, I am video of Shefali and group.
Formulation and evaluation of mouth dissolving amlodipine benzoylate tablet IP-10H. Introduction of a therapeutic substance into the body to improve its efficacy and safety are known as a drug delivery system. Drug introduced into the body by the various routes like oral route, most popular route used for an conventional as well as novel drug delivery system. However, the traditional tablets and capsules leads to the difficulty in geriatric patients, while the solaric and other difficulties too. That's why we have prepared fast disintegrating tablets which disintegrates rapidly and increase the rate of absorption. Advantages of the fast disintegrating tablet. Easy for geriatric patients. Then increased bioavailability and rapid absorption. There are some disadvantages also regarding to the fast disintegrating tablet, integration and dissolution of mouth dissolving tablet. These are the materials which have been prepared amyloidectin mouse dissolving tablets. This is the method that we have been used for the preparation of amyloidectin mouse dissolving tablets, that is direct concentration method. We have been also performed the pre-formulation studies that are followed. Physiochemical evaluation of a drug molecule, where pH, solubility, and solubility criteria. Also, we have performed the pH analysis there, and also we performed the melting point. We have also carried out the chemical nature of an amyloidectin and the hygroscopicity of an amyloidectin. Also, the particle size analysis, flow properties, and bulk density. Density, compressibility index. This is about the flow compressional physiochemical parameters of an amyloidectin granule. There, the F5 and F6 formulation shows the good flow as indicated by the angle of release. We have also performed the post-compressional parameters of an amyloidectin tablet, where the formulation F5 and F6 shows the least disintegration time, which is the most satisfactory method. Drug release value of an amyloidectin tablet. This is about the conclusion about our research project. The current studies are aimed at a successful development and optimization of a mouse dissolving tablet of amyloidectin designed and to enhance the bioavailability of the drug by avoiding the first pass metabolism. From this study, it may be concluded that the amyloidectin IP tablet 10 mg can be prepared as an immediate release formulation compared to the conventional dosage form. These are the references we have referred for our research project.
Yeah.
We would really thank to our institute, Indira Institute of Pharmacy, startup for giving us such a
Yeah, thank you, Shefali and group for sending the video. So that was screened by me. And Mandar sir, now over to you. So you can directly ask abstract number and then the presenter.
Am I audible, sir?
Yeah.
Yes, yes. Go ahead, please.
Yeah, all other participants, they are requested because to prepare themselves so that they can directly start this.
Am I audible, sir?
Yeah, yes, you are.
Good afternoon, everyone. I am Ankar Anaji More, PG Department of Botany, SNJP's Arts Science College, Angad. My presentation topic is in vitro regeneration of valuable medicinal plant, Centella asiatica, under the guidance of Dr. Sudhin Darve, sir.
Hello, ma'am. Yeah, we cannot see your screen. We can see. Yeah, please share the screen. The screen shared by you was, I think, the mobile first page. So can you maximize that PPT? Yeah, pretty good. So now we can see. Yeah, you can start, Priyanka. Priyanka ma'am, you can start.
नहीं कॉन्फ्रेंस तो
Mita ma'am, please mute yourself. Mita ma'am. Priyanka ma'am, we cannot hear you. Priyanka ma'am, we cannot hear you. Please unmute yourself.
Yes. Now it's
Sudhin sir, what is problem? Sudhin sir.
Yes, sir. Now it's everything okay.
Yeah, what happened that it was not audible earlier.
My presentation topic is in vitro regeneration of valuable medicinal plant Centella asiatica under the guidance of Dr. Sudhin Dave sir. Introduction. Centella asiatica, vernacularly known as Brahmi and Mandakpurni, is a small creeping perennial herb. It belongs to the family Umbelliferae. It is found throughout India. It is used as a brain tonic and in the treatment of leprosy, varicose veins, ulcers, lupus, and certain eczemas, stomachache, carminative, diuretic, and laryngitis. The medicinal values of Centella is ascribed to its triterpenoid glycosides, Centella asponine, and medicacic acid. In the present of investigation, attempts were made to propagate the plant by tissue culture technique. These plants can be propagated by means of vegetative propagation, but still there are certain limitations. Medicinal properties of the plant has led to its over-exploration. Therefore, application of tissue culture approaches for rapid multiplication of elite clones and germplasm conservation is of vital importance. The work is started with an objectives. First one is propagation by tissue culture technique of the authenticated plant. Second one is micropropagation. Third one is selection of somaclonal variants. And fourth one is hardening of the propagated plant. Materials and methods. For present investigation, plants were procured from Dhanvantari Medicinal Garden, Mahatma Phule Agriculture University, Rahuri, and cultivated in our College of Botanical Garden. Plant material were used, nodal region of the stem, runner on leaf of Centella asiatica, used as eggs plant, media were used, MS vessel media, Burashik and Scoop, 1963. Various media composition of hormones in full strength and half strength MS media were studied that includes first one is half MS 1 milligram per liter BAP to 6 milligram per liter BAP. Second is half MS media plus 2 milligram per liter B plus 1 milligram per liter to 4D. Third one is media plus 1 milligram per liter P to 6 milligram per liter BAP. Fourth one is MS media plus 4 milligram per liter BAP plus 1 milligram per liter to 4D. Fifth one is
Hello, Sudhin sir, what happened? Sudhin sir.
Hello.
Yeah, we have already tackled.
and fifth one is MS media plus 4 milligram per liter BAP plus 2 milligram per liter to 4D. And sixth one is MS media plus 4 milligram per liter BAP plus 1 milligram per liter NA and so on. And the results are first image showing half MS basal showing only elongation of shoot in supplementation with 1 milligram per liter BAP, and it shows only one shoot for the period of 3 months. It is found that there is very slow growth. And the second image showing half MS basal showing only elongation of shoot in supplementation with 2 milligram per liter BAP, but there is no induction of new shoots for the period of 3 months. And there is multiple shoot initiation, multiple shoot regeneration from single node in MS media plus 4 milligram per liter BAP plus 2 milligram per liter to 4D. Then subculture of the multiple shoots on MS media plus 2 milligram per liter BAP plus 2 milligram per liter to 4D for further growth. And the improving 4 milligram liter BAP showing fast growth of the roots, and showing half ml basal supplemented with 1 milligram per liter Na is found good for root initiation, and this callus in MS plus 4 milligram per liter 4D plus 1 milligram per liter BAP and 2 milligram per liter and 1 milligram per liter BAP found slow in inducing callus compared to plus 4 milligram to 4D plus 1 milligram per liter BAP. In conclusion, first one is it is the best material to inject from tissue.
Okay, Mandar sir. So,
Yes, sir.
Yeah, you can call the next participant.
Yeah. Is there anyone want to ask a question? Or we'll go directly to the next presentation. Hello. Okay, next one is Mansi Verma and Ruchi Yadav. Hello. I'm audible?
Yes, sir. Yeah.
Yeah. Mansi Verma and Ruchi Yadav.
Yes, sir. Shall I start my PPT?
Mansi, yeah. You can continue.
Am I visible and audible?
Can you increase your volume?
Sir, am I visible and audible? Hello.
Mansi, safe drinking. Yeah, which one is your screen?
Your voice is so low. Mansi, ma'am. Your voice is so low.
Sir, is my screen visible?
Yeah, just a minute. Sudhin sir. Sudhin sir, please stop sharing the screen. Sudhin sir. Sudhin sir and Priyanka ma'am. Sudhin sir and Priyanka ma'am, please stop sharing screen. Tanmay. Hello, Tanmay. Yeah, Mansi ma'am, you can directly start to share the screen.
Sir, the host can control also. You can control also. Just only share it to the co-host.
Mansi ma'am, you can see, yeah, we can see your screen. You can start. You have to complete within 5 minutes, Mansi ma'am.
Okay, okay. So good afternoon, everyone. My name is Mansi Verma.
Ma'am, your voice is so low. Can you increase your volume? Otherwise, you can continue, ma'am. Yeah.
My voice is not properly audible. Hello.
Yeah. You can continue, ma'am.
Very,
It is too low, but
very
yeah,
low. Your voice is so
we will
low.
consider.
Okay. So proteomics is the study of proteins on large scale. It is a branch used to identify expressions of different proteins and their functions. So first of all, my topic is computational proteomic analysis of bacterial proteins. What is proteomics? Proteomics is a branch. It is an emerging field which detects proteins involved in human disease and understand its expression, functions, causing the disease and then next is proteomics.
Mansi ma'am, we cannot hear you. Let it be. You can go directly to the conclusion slide.
Now am I audible?
Yeah. Now it is audible.
Okay. So I was discussing about proteomics and then the disease producers. So producers whooping cough, it is a highly infectious bacterial disease of respiratory tract, more common to infants. The symptoms of the disease include like sneezing, runny nose, vomiting, nasal congestion, and fatigueness. During breathing, a whoop sound. During coughing, a whoop sound is produced. Thus, the disease produces getting its name as whooping cough. Now, the bacteria Bordetella pertussis, I have studied about this bacteria. It is a gram-negative bacteria, which is the sole agent responsible for this disease pertussis, which is more common in infants. So, Next, the materials and methodology. So firstly, through UniProt. So UniProt is a freely accessible database having a collection of all the proteins and sequences. In this database, I have searched all the proteins which are related to this bacteria, Bordetella pertussis, and I have predicted the structure of the proteins which are not available on UniProt database. So this was my work. These are the proteins which I have studied. Total 9 proteins I have studied. I have predicted their structures through Swiss model, homology modeling through Swiss model database, and then the functions. So these are the predicted structure, model structure for all the 5 proteins. This is the Ramachandran plot. Ramachandran plot is used for validation, like how much the perfect protein structure you have predicted. And then this is the string network. Now further, I have done in my research paper, I have predicted the functions of all the proteins, like what is the role of that protein in that particular bacteria. So FHA is the protein which is mainly involved in causing pertussis disease. Conclusion, my paper signifies, as I said, all the 9 key proteins that are associated with the bacteria, Bordetella pertussis, and having their function in the pertussis disease. The structure of all the unannotated proteins on the UniProt was identified through homology modeling, and the structure was built using the Swiss model database using the template. Ramachandran plot analysis was done for the structure validation, which depicts the good quality model. Through string database, we have created the protein-protein interaction mapping to see like what all other proteins are associated or are near, what are the roles of other proteins in the bacteria, and which proteins are involved in the pertussis disease. And finally, the KET pathway was made, drawn, this shows that the protein network is involved in the pertussis disease. FHA protein was involved in the pertussis disease. These are the differences. Thank you, sir.
Your voice again went low. Now. Okay. Is there anyone to ask question?
My PPT was shared.
Hello.
Hello.
Ma'am, please close your presentation.
Can you please unshare? I am like from.
Hello. Ma'am, your voice is too low.
Yes.
Is there anyone to ask question? Okay, we'll go to the next presentation. Presentation number 55. Siddhi Sani, Shivani Hatiskar, Prachita Sute, Tanmay Patwardhan, Amol Khade. Hello.
Sir, please play the video.
Yeah, just a minute. I will share the screen and then just now I am going to share the screen and here is the video.
Student Bachelor of Pharmacy studying at Indira Institute of Pharmacy, Sadar. Today, the topic of my presentation is about design and explore triclosan and triclosan mimicking anti-proliferative agents. The contents of my presentation same such as chemotherapy is generally unaffordable and non-compliant to the patients, and also has many side effects which makes it non-compliant to the most of the patients. Considering the above points, we are working on triclosan, which is a popular antimicrobial agent and also cheaper than other chemotherapies. In view of this, we are repurposing its activity for an cancer treatment as an anti-cancer. We have used many softwares for the showing interaction between ligand and protein. So these are the softwares which we have used. ChemDraw3D, Swiss ADNE, NGL Tools Autodoc Vina, BioVia Discovery Studio, and Protein Drug Bank to obtain the protein molecule for the docking. So these are the chemicals which we have used to perform the identification test for the triclosan, which was obtained from the industry known as Dev Inflex, Gandhinagar, Gujarat. The methodology for our experiment is molecular docking. The molecular docking, the Swiss ADNE is a software for ADNE prediction, which we have used for the triclosan and its derivatives. Autodoc Vina software. It is used for ligand protein docking to predict the binding of ligand with the protein. BioVia Discovery Studio 2016 was used for visualization of interactions which were performed by Autodoc Vina. And here are the amino acid residues which are forming interactions with this derivative. The results from the docking which we have performed was the docking score for triclosan was -6.3. For T3, which is a derivative of triclosan, it was -8. For T4, it was -8.2. So here we can see that the docking score for the derivatives was much higher than the triclosan. From the values, we can say that it is more it can be or it could be more potent drug than triclosan to be used as an anti-cancer agent. From the study, we have concluded that in the present study, we have done in silico molecular modeling of triclosan for its anti-cancer activity using open source docking software, Autodoc Vina. We have also designed rationally two novel triclosan molecules, which we found to be better in ADNE prediction and gave more docking score than that of triclosan, clearly suggesting it could prove to be more potent anti-cancer agent. So we are also looking forward for screening of this triclosan in cell lines, for all possible cell lines, and also for the synthesis and characterization, evaluation of design novel triclosan mimicking derivatives as an anti-cancer activity.
Yeah, so this was all about the presentation of Shefali and group. So if you have any question, then you can ask, or Mandar sir will call to the next participant.
I think we will call. Nobody is asking. Yes, Rajendra Mani. Mani sir, please start your presentation. Mane sir. Mike, what is sir?
Mani sir, can you hear me? Mani sir, Rajendra sir.
Yes, sir. It is audible for us.
Yeah, but Rajendra Mani has started screen sharing, but yeah, no voice as well as we cannot see the screen of Rajendra Mani. Rajendra sir.
Yes, yes.
Yeah, you can start your presentation. We cannot see your screen.
Please go on presenting. Go on talking.
My screen is visible.
Oh, no, but yeah, you can start. Maybe within 1 or 2 seconds it will be made visible. Yeah, you can talk. And please try to conclude within 5 minutes.
Yes. It is not moving forward.
No problem. You can talk.
Yeah, actually, basically, my presentation is about the cost-effective and green air purifiers plants. So I have gathered some information regarding plants as a indoor or outdoor pollutants. What are the indoor and outdoor pollutants, and what are the plants which can be utilized to overcome the pollution to the indoor and outdoor pollution to the plants. So I have made a library of these indoor and outdoor plants, and actually, this presentation is not doing.
Yeah, let it be, Rajendra sir. You can directly talk within 2 minutes.
So yeah, I will just talk about my presentation. So it is not there. So the idea was that, so I am developing an app where one can get all information regarding the what are the pollutants, what are the indoor pollutants, what are the outdoor pollutants, and what are the plants that that can be worked on the this as an pollutants. And so I have gathered this information and I will put it in an app. So the idea was to develop a pollution hotspots into green hotspots. So a lot of this traffic or theaters or toll nakas are there where we can implement these plants, where we can implant these plants and we can slowly overcome the pollution. So that was the idea, and to get the information regarding all these plants. So I am
Rajendra sir, please continue. Rajendra, please stop sharing the screen. Rajendra sir, can you hear us? Can you hear us?
We might have lost
Yeah.
connectivity, sir.
Yeah. Okay.
So, shall we call next?
Yeah. Yes.
Yeah. Next 57. Saeed SV and Sonule MD. Saeed SV and Sonule MD. Next one is G. Vedanta Desikan. G. Vedanta Desikan. Next one is Supriya Naik and Chaya Panse. Are you there, ma'am?
Sir, can you please take the abstract number as well? The names is becoming a little confusing.
Abstract number is 59. Supriya Naik and Chaya Panse.
Yes, sir. So is my screen visible?
Yeah, very much. Please go on.
Good afternoon, everyone. I am presenting my topic, study of antibacterial property for biosynthesized silver nanoparticles using chitin. Nanoparticles are particles which range from size of 1 to 100 nanometer. They exhibit large surface area to volume ratio, which is one of the very unique identity showed up by the nanoparticles. Production of nanoparticles need high amount of toxic and harmful and hazardous chemical, which is employed in conventional methodology, which doesn't hold to be environmental friendly. And thus it requires, it requires high amount of energy for its production, expensive, it is highly expensive and time consuming. Thus, due to the usage of toxic chemicals, it limits its application into the clinical fields and in biomedicinal field. The alternative to such conventional methods is to use biological molecules. These biological molecules are not just very fine reducing agents of silver nanoparticles, but they can also act as capping agents. They will bind over the surface of the atom and thus will also increase the stability of nanoparticles. When we talk about biomolecules, here in this my experiment, I have been taking the biomolecule that is chitin, which is a form of primary component which is present in the exoskeletons of arthropod. These are the shells. Arthropod animals are like crustacean animals such as prawns and crabs are one of the most easily available animal source, and their skeletons form the biological waste component which has just been thrown off in the environment. So here we are trying to utilize these components, these exoskeleton of these animals, and to produce chitin. We try to extract chitin from these shells, and it is said that these animal shells have around 15 to 40% of chitin in pure form. Chitin is a modified linear polysaccharide containing acetylamine group, and it has all the required necessary, it is necessary for reduction and catalysis of metal precursors. So when we talk about nanoparticles where it is required, we require a high amount of toxic chemicals for its reduction to the nanoparticle size. Here the chitin material will be acting as one, which will help in reduction and catalysis of these nanoparticles. Silver is one of the metal which we are assuming to always being excellent as an antimicrobial agent. So here in this my experiment, I have taken silver metals to produce the nanoparticle. Silver had been used from very long years as it forms metallic silver and sulfur-diazine in the form of antiseptic creams and many more. So silver nanoparticles are involved as because of the formation of reactive oxygen species, thereby inhibiting respiratory enzymes and protein, leading to physiological malfunctioning and responsibly these metal silver are responsible for the mortality of gram-positive and gram-negative bacteria. The ionic silver strongly reacts with thiol group of vital enzymes, and it will inactivate the enzyme activity of these bacteria. Earlier researchers have reported that silver inhibits phosphate uptake and exchange in E. coli bacteria and causes efflux of accumulation of phosphate as well as mannitol and hexacenate in these bacteria, leading to death of these bacteria in the given culture. So when I talk about chitin-based nanoparticle, silver nanoparticle, as we know, antibiotic resistance of microorganisms is the major problem which is being faced by.
Sukriti ma'am, try
Yes,
to
sir.
within a minute, ma'am.
Yeah, sorry. So antibiotic resistance is one of the important criteria that we are looking into. So chitin is a chemical which is biocompatible, biodegradable, and has a rapid dermal regeneration property. So on basis of the rapid degeneration property, it gives a high amount of application of the silver nanoparticle which has been synthesized using chitin. So the objective of this experiment was to develop a new protocol for extraction of chitin with using minimal amount of chemicals to synthesize the silver nanoparticles using chitin and to analyze its antibacterial property. The sample used here are the prawn shells. The dried prawn shells are being used here in this experiment. The extraction process included the alkaline method where the fractionized of prawn shells biomass involves demineralization, deproteinization, and deacetylation process. The parameters which were worked on during this experiment included the concentration of the chemical, the temperature, time, and solution to solid ratio used for the extraction process. And the metal used for preparing of silver preparation of nanoparticle is silver. Further, we have tried to analyze the biological activity by using the in vitro antibacterial testing method, that is this diffusion method, which is also called as Bayes test. And the bacteria culture which I have used here in my experiment is E. coli. So by using a mother culture on the LB broth, I have tried to create a culture, and once the spreading is done on the petri dishes having the nutrient medium, antibiotic filled small Watchman paper which have been treated into small size circles have been implemented on these bacterial cultures, and they are allowed to stay in the incubator for a while. So the result here, what I received was that these chitin were not only stabilizing the metal nanoparticles, but also they were forming a caping agent. And because of this combined structure, we could find the change. This is the visual observation of chitin. Sorry, sir.
Sum up. Okay. Is there anyone to ask any question? Hello. Okay, thank you, ma'am. Thank you.
Thank you, sir.
Next one is Hrishikesh Sansare, Nitesh Tokre, Kanaya Pakhre, Tanmay Patwardhan, and Amol Khade.
So I am playing the video. Sir.
Okay.
I'm going to present a presentation on our project repurposing of triclosan for the treatment of cancer or prevent bacterial contamination. Initially, triclosan was considered well tolerated and safe. However, triclosan was recently found to affect endocrine and impair muscle contraction. However, the exact role of triclosan in NCF7 regulation of treatment is still largely unknown. In the present work, triclosan is shown to induce the expression of drug-resistant genes and promote the clearance of triclosan MCF7 cells. The conditional study to determine anti-cancer activity and to conclude the results. The chemical used in this project were triclosan API, DMSO, acetone, neutral KMNO4, nitric acid, silver nitrate solution, and NaOH. Whereas the software used is ChemDot3D, ADA, Autodoc Vina, BioWare Discovery Studio, and PD. The methodology used for this project was molecular docking, which is a key tool in structural, microbiological, molecular biology, and computer-assisted drug design. With the project, BioWare Discovery Studio of the system was used for visualization of receptor ligand interaction. These are the images of triclosan which were captured with help of BioWare Discovery Studio. In first image, ligand protein interaction is shown, whereas second image, triclosan deciding YAG package is shown. These are the images of T1 ligand which were captured with the help of BioWare Studio. These are the images of T2 ligand which were captured with help of BioWare Studio. This project resulted in two stages. In the first, ADME prediction. Various parameters were found, like molecular weight, log P, HPD, and HPA. In each of them, T1 and T2 showed better results than that of 5%. Both T1 and T2 obeyed Lipinski's rule of 5. Second parameter was in silico molecular and the top score of T1 and T2 were much better than that of 5%. T1 with top score of minus 8.7 show the interaction like hydrogen bonding interaction of nitrogen of isoxazole with ASN 1/17. Multiple pi stacking interaction of 3 phenyl rings of E1. Multiple van der Waals interaction of 6 AA residues. E2 show dot score of minus 7.9, where no H bonding interaction ratio and pi stack interaction ratio. In the present study, we have tried to explore virtually calculation for anti-cancer potential. Additionally, we have designed 2 novel calculation limiting agents which were found to be better than calculation in ADME and molecular predictions. We are in the process of doing an in vitro screening of calculation by FTP assay on various cancer segments so that it can be repurposed as an anti-cancer. As an output, we are undertaking the screening of calculation for all possible segments. We are also conducting synthesis characterization and evaluation of design novel calculation limiting for anti-cancer activity.
So this was all about the presentation of Chisquare, Ansari and group. So Mandar sir, over to you.
Yes, sir. Next one is 61 in a row. Sunil Sunavane and Shiva Padme. Okay. Next one 62. Durgesh Patil and Uday Singh Bhosle. Next is 63. Y Savitri and P Ravi Sekhar.
Yes, sir. I am presenting, sir.
Yeah, yeah. Ma'am.
1 second.
Please continue.
1 second. One second, sir. I'm opening my PPT.
Other participants, they are requested to ready with their presentation.
Sir, is my screen is visible for you, sir?
Yeah.
Good afternoon to everyone. This is Dr. Y. Savitri, lecturer in zoology, Government College for Men, Autonomous, Kadapa. Today I am going to present my oral presentation on anthropogenic activities and their impacts on biodiversity on behalf of the International Conference on Environment Development and Sustainability. So before going to present my topic, we all know environment means literally the surrounding and everything that affects an organism is called as environment. And biodiversity means bios mean life and diversity means diversification of animals in the earth. Today, biodiversity facing so many challenges because of human advancement as well as development. So these are the different important issues regarding to the environment as well as biodiversity. We had to sustain the development, otherwise we may face so many challenges in coming days. So these are the importance of biodiversity. Biodiversity consisting of various values like commercial values, like oil, fertilizers extracted from species of plants as well as animals. And we are also having the biological values like pollination, soil formation, nutrient enrichment. And recreational values, we are enjoying a lot with the biodiversity. And we are also having the aesthetic value. And even the animals and biodiversity also we are using in scientific advancement also. In gene pool experiments, evolution, human welfare, etc. And nowadays, the human, because of the human and anthropogenic activities, biodiversity is affected in many ways. And these are the main effects of human beings: habitat destruction and fragmentation, poaching and hunting of flesh, fur, skin, hair, nails, and recreation, and forest fires, including smoking in the woods, over exploitation, and natural calamities like floods, high wind, volcanoes, and earthquakes, etc. Pollution of various kinds and release of toxic substances because of industries and urbanization and various human activities. And the soil erosion and loss of soil nutrients also one of the important factors for the biodiversity loss. And population pressure, agriculture expansion, spreading urbanization and establishment of industries, construction of hydroelectric power projects, and climatic changes, mining activities and quarrying, and lack of awareness. So these are the different anthropogenic activities that affects directly or indirectly by the biodiversity. And these are the different conservation methods as we follow the legislation of government and in situ, ex situ conservation, maintenance of hotspots, and following the IUCN categories, cryopreservation, tissue culture techniques. So these are the different conservation methods of biodiversity. And what is the need of conservation of biodiversity? And we have to create the awareness among the people. So by taking quality of environment and creating awareness among the people by conducting rallies and interaction with the people, and giving information about the atmosphere and the changes in the atmosphere, and the human activities, as we previously discussed, deforestation and everything, they affect the environment as well as the biodiversity also. And these are the key areas of human activities that affect directly the human biodiversity. The first one is deforestation, agriculture, industries, mining, transportation, and ozone depletion. So these are the major things, and some other factors are also influencing the biodiversity loss. And so this is, we are discussing about the importance of biodiversity. We have most importance of biodiversity. Otherwise, so many species are now in the, they are going to be extinct. And with the awareness creating among the people, we may save some animals. And the biodiversity loss happening through various activities. And these are the key areas of human activities. We are, depleting the biodiversity. And what can we do to protect biodiversity? And we had to minimize the population growth and minimize the deforestation, as well as we had to giving awareness among the people about the importance of biodiversity. And as an individual, so as a group of people, we can create the awareness among the people, as well as an individual, we can protect the biodiversity. And individuals can make a difference. Some of the things we can, I can mention here: supporting political action committed by the government, supporting the institutions to promote the protection and restoration of biodiversity, support the local and regional projects, and buying fewer products, investing in ways of promote the biodiversity, reducing, recycling, and reusing of products, and educating the children when they are younger in years, when we educate the people about the biodiversity so that they can save the biodiversity. And at my conclusion, biodiversity has become an important issue on the global, and we have to save the biodiversity by minimizing the anthropogenic activities. So as I'm the lecturer in zoology, on behalf of Department of Zoology, we are conducting so many awareness programs on behalf of environment as well as biodiversity. And a few are mentioned here: the ozone layer day and Animal Day, and some other activities also we are conducting on behalf of our zoology department. So thank you, sir, for giving me this opportunity to share my review work on this international conference platform.
Thank you, ma'am. Thank you. Is there anyone to raise a question? Okay, thank you, ma'am.
Thank you, sir. Thank you, sir.
Next one is Sandhya Jagtap. Sandhya Jagtap.
In sir,
Abstract number 64. Mandar sir,
Yes. Abstract number 65. S.A. Funde. Abstract number 66. Payal Athrekar, Ambika Joshi.
Yes, sir. Sir, I'm there. I'm sharing.
Yeah. Please, ma'am. Start your presentation.
Yeah, Muskan ma'am, please mute yourself.
My screen, sir?
Yeah.
Yeah, it is loading.
Yeah. Okay, so may I start?
Yeah, please.
Yeah. Good afternoon, everyone. I'm Dr. Payal Asrikar from the Department of Botany, Jain College, Autonomous, Mumbai.
and I'll be speaking on vegetation cover for abatement of noise under the guidance of Dr. Ambika Joshi. The talk will be subdivided into the following headings such as basic introduction, materials and methodology, results and discussion, conclusion, future prospects, and references. Now, to start off with, why this topic? As you must be aware, with the increase in urbanization and population explosion, there is also an increase in the number of industries and vehicles that lead to release in harmful chemicals, which lead to air pollution. But at the same time, they also manage to release or generate a lot of excessive noise. For a very long time, noise was not even considered worthy of a pollutant, and nobody gave it any significance or importance. It was later discovered, thanks to the WHO reports, that a lot of health ailments were a result of the direct impact of noise on them. Now, it has been proven time and time again that trees play a major role in reducing air pollution, and there has also been ample evidence that there is effect of music on the growth of plants. So why not use these trees in abatement of noise? But will planting random trees just help? Well, another study here has already shown to us that the sound absorption capacities of individual species vary, and that they have some have more capacity and some have less capacity. We, on the other hand, are trying to emphasize on how the shape of a tree canopy can contribute other than the nature of the leaf, such as leaf texture and size. As observed in the image here, we majorly come across 4 different types of canopy shapes, such as triangular or conical, hemispherical, spherical, and ellipsoidal. Now let's proceed to see how they can contribute in noise abatement. To start off with, the major aim of this experiment was to make use of inexpensive and effective methods for the purpose of noise studies and at the same time noise abatement. Now, a similar thing could have easily been done by LAI or satellite system, but it is very, very expensive. The procedure is fairly simple. We need to select healthy and mature trees to get a full-fledged canopy. Then we need to record the noise values below and above the tree canopy. Now, as you see here on the top right-hand side corner, there is an instrument known as sound level meter or decibel meter or NSLM, and it is one of the most basic instruments that can help us record noise. Now, there are higher versions available as well, but I have used this basic model. The instrument consists of a mic, display screen, and a range of audible ranges from as low as 40 to 150. And 50 observations were made under the various tree species. Now, the next important step is the measurement of the tree canopy, which is further subdivided into a manual observation of the shape of the canopy, and we also need to calculate the volume of the canopy. Now, if you have a look at the bottom right, there is an instrument known as Abney's level. An amusing thing about this instrument is that it is used to measure the height of tall buildings in the field of engineering. We here are applying it and using it to measure the height of a tree canopy. As seen in the image on the left-hand side, the man is standing over there and you're using it to measure the height. Now, the height is further used to calculate the volume of the canopy. For example, it can be used as a radius or height depending on the formula that you may apply. The distance of the man standing there is determined by the visibility of a tiny bubble through the Abney's level. The bubble in the center, and that is, like if you can see the tip of the canopy, that is the appropriate distance. Coming to discussion and conclusion, on careful observation, we have noticed that the majority of the trees under the study were either hemispherical, which is, for example, Semania saman, or spherical, which is Peltophorum. Tree canopies which were other than this, that is conical canopy, which were only 16%, and ellipsoidal, which were only 11%. Now, going by the volume of the largest canopy, Kauropita shows the largest volume, but the best abatement results were seen in a triangular canopy shown by Polyalthia or Putranjiva. The reason for the same is that they are broad at the base and they go tapering or linear upwards, thus diffusing the sound waves and abating noise. Unfortunately, the worst is seen in ellipsoidal, as it is narrow at the base and broad in the center, and again narrow at the top. Going by the leaf texture, Ficus hispida shows the best result, and it is not so far in the canopy as well, which is spherical in nature. Future prospects, we wish to recommend the appropriate tree species which can not only help us in town planning, but at the same time help us in producing more oxygen, reducing air pollution, and abating noise. We also aim to continue research in this field with the help of our current study to produce better suggestions and recommendations. Now, these are my few references. Thank you very much.
Okay, thank you, ma'am. Thank you.
Thank you, sir.
Yeah.
Any
Is there anyone to
questions?
ask a question? Any question, please? Okay, thank you, ma'am.
Thank you, sir. Thank you for.
Next, Shweta Shinde, Snehal Goye, and Manoj Patil. Yes, ma'am, please.
Hello, sir.
Yeah.
Hello, sir.
Yeah. Start your presentation. And
Hello.
Please, yeah.
Hello, sir.
Please just discuss methodology
Hello,
and
sir.
findings. Hello, madam.
Hello.
Yeah, you are audible. Very much audible.
Hello, sir. I am Jagtap Sandhya. I send my video presentation.
Okay. Hello, Dr. Microarray.
Hello.
Hello. You have mailed it. Hello.
Hello.
Have you mailed it?
हाँ mailed it हो.
Okay. Next one is we will see it on later on. Okay. Next one is abstract number 68. Ajit Devle, Shubham Dakle, Sagar Sampad, etc. Abstract number 69.
Hello, sir. I'm audible.
Prakash Shinde. Yeah, you are audible. Hello. Okay, Shweta, go ahead. Start your presentation, please.
Okay, sir. Hello, good afternoon, everyone. I am Shweta Shinde. My friend Snehal Goye and Dr. Manoj Patil, sir, from PG Department of Botany, SNJP College of Stanford. We are here to present our work on phytochemical analysis of citrus isora fruits. Thank you for giving us this opportunity to present our work. So I may get attention for a few minutes. So let us move on the presentation. Phytochemical analysis of Helictrus isora fruits. We all know that medicinal plants are owed to nature and the pharmacological activities of
Hello. Hello, Shweta. You are not audible. Hello, Shweta, ma'am.
Sir,
You may be facing the connectivity problem.
Hello, sir.
Hello.
Hello, sir.
Ma'am, go ahead, please.
According to World Health Organization, about 8% of population relies on traditional medicines for their primary health needs. The phytochemical studies of plants is an important step to identify the bioactive compounds present in the medicinal plants. The bioactive compounds present in the medicinal plants have been evaluated for their
Hello, ma'am.
Yes, sir.
Just discuss your methodology and finding, please.
Yes, sir. Okay, sir. So these are the methodology. The fruits of Helictrus isora were collected from plateau of Sandward, from Sandward, Nasik district, which falls in Satmaya Hills, perpendicular to Western Ghats of Maharashtra. The fruits of Helictrus isora were washed with water to remove the dust particles. The shade-dried fruits were finely powdered auto-mix blender. The aqueous and alcoholic extract of powdered fruits were prepared for phytochemical screening. These methods we use for the screening chemicals.
For the proteins, Minas test were performed. White PPT represents the presence of proteins.
Hello.
Hello, sir.
Yeah, your screen is not
I am
visible.
audible.
You are audible, but screen is not visible. Okay, just tell your findings, please.
Okay, sir. So our results show that the primary screening of phytochemicals show that presence of proteins, flavonoids, alkaloids, steroids, carbohydrates, amino acid, fats and oils are found present, while the glycosides and tannins are absent. The table shows that the presence of proteins, flavonoids, alkaloids, steroids, carbohydrates, fats and oils and amino acids, while the glycosides and tannins are absent. So in our result,
Hello. Hello, ma'am.
hello, sir.
Please.
Sir, sorry, network issue. Experimental results reveal that Helichrus isora fruit is good potential source of secondary metabolites. The overall, it can be considered that the plant is highly medicinally potential. The work is going on microbial activities of Helichrus isora fruit. For the antimicrobial, powdered wash weight and extraction process is carried out by using 250 ml of chloroform in oxalate apparatus for 48 hours. Extract was concentrated by using rotary vacuum evaporator, and concentrated extract was stored in gel or semi-solid form at the room temperature and prior to phytochemical activity. That that we refer for our research. Thank you. Thank you so much.
Okay, thank you. Thank you. Please stop your sharing. Next, abstract number 70. Chandrasekhar Bhamre.
Hello, sir. Good afternoon. I'm here, Durge. Wait, sir, I'm sharing.
Yes. Please go ahead, sir.
Yeah. Nice. Okay, my screen is visible, sir. Hello. Hello. Hello, sir.
Yes, visible. It is visible. Please go.
Okay, sir. So I'm Dr. Durge. Okay, from the Patkarwardhe College, okay, Department of Physics. Okay, Mumbai, Goregaon West. Okay. That is the outline, introduction to the crystal growth method and the characterization and conclusion part. And this is the abstract topic. And name is study of structural and thermal properties of metal nitrate containing NLO semi-organic crystal, and how the crystal is growth based on the phase transformation process. And this crystal growth technique can be classified as a solid growth, vapor growth, and metal growth, and solution growth. So these are the and this is the constant temperature bath used for the to grow the bulk to grow the crystal from the solution. This is slow evaporation technique method, and the temperature is fixed. And the provision is made for the evaporation. And with a non-toxic solvent like a water, it is possible to allow the evaporation into the atmosphere. And typical growth condition involve the temperature stabilization about plus or minus 0.00 degrees Celsius, and rates of the evaporation few milliliter growth. And the evaporation technique of the crystal growth have the advantage and that crystal grow fixed temperature. And there is some advantage of the slow temperature solution growth. Simple growth apparatus, growth of strain and the dislocation of the free crystal permit the growth of plasmatic crystal by waiving the growth condition. This is only the method can be used for the substance that undergo the decomposition method, and this is the more convenient method also. And the growth substance could not be the react with a solvent, and this method is applicable for the substance fairly soluble in the solvent. And this method, the crystal growth of the good quality single crystal by slow evaporation requires for the optimize the condition, and the same may be achieved with the help of the following norms. First is material purification, second is solvent selection, third is a solubility, and fourth is solution preparation, and five is a seed preparation, and the seven is agitation, and the seventh is the crystal habit, and the last is the cooling rate. And this is we have grown this the crystal in the lab, and then characterization study is done. That is the confirmed is the chemical composition, structure defect, study of their optical properties, etc. And characterization are divided into this crystal structure analysis, investigation of the growth defects, non-linear optical characterization, third is measurement of electrical properties, and the fourth is a thermal property. And now this is the semi-organic analog crystal. This is the LM. LM. LM means L methionine and barium nitrate, and the second is a L-leucine plus magnesium nitrate, were successfully grown by the slow evaporation solution growth method at a constant temperature 40 degrees, and then XRD data of this both the crystal confirm the cubic and monoclinic crystal structure respectively, and FTR is carried out and confirm the functional group of the pure and open material are present in the spectrum. And then the TGA then the UV BIS NRN spectrum also. It is a found the good transference of the LM and BN crystal is observed, and the spectrum indicates that the crystal has broad optical window from 304 nm to the 1100 nm, as there is a lack of absorption in this spectral range. Then in the case of LL and MN crystal has a wide optical transfer window, and the spectrum indicates that the crystal has a broad optical window from 300 nm to 1100 nm, as there is a no absorptions in this spectral range. And then the TGA DTA data, TGA and DTA data in the case of LM and BN crystal written it a texture up to the 286 approximately degrees Celsius, and started to lose its lattice water molecule, and this process continued to 87 degrees Celsius. Around the 49.8% of weight of a sample is reduced, and beyond which another step decrease in the weight is observed, which is due to the melting and decomposition of remaining sample at 311.6 degrees Celsius with the total weight loss. In the case of this is also in the LMNM is up to 45 to 84.2 degrees Celsius, and the weight loss is found to be 3.5% at a 84.2 degrees Celsius and 29.2 degree percentage, okay, at a 21062 degrees Celsius, 17.8% at a 0.16 degrees Celsius. Here the endothermic peak in a DT above the, that is the 101, 0.23 degrees Celsius represents the melting point sample, and there is a small decomposition crystal material from the 39, approximately the 39 to 68 degrees Celsius. Further decomposition continues up to the 101 degree Celsius. And the conclusion part of my research, the XRT data of LM and MN is a confirmed, and it is a cubic and monoclinic crystal structure respectively. And thermal analysis of the LM and BN and LN plus MN crystal is thermally stable only up to the 286.5 degrees Celsius and 84.2 respectively. And the fruits Perry test confirmed LN, and this is and just exactly the same simple harmonic generation efficiency comparisons with India and KDF research. And these are my reference, and thank you very much, sir, by giving me the opportunity to share this.
Thank you, sir. Thank you, sir. Is there anyone to ask any question, please? Okay, thank you, sir. Next in the row, abstract number 71, K.B. Khairnar, P.P. Bhalekar, and R.P. Borare. Is there any of them? Next, 72, Manish Kumar Gupta, Sujit Nagre, Virendra Srivastava, Supriya Ham, and Ketaki Dhani. Abstract number 72. Is there anyone? 73. Sonali Patil.
Sir, yes, I have forwarded the presentation, sir.
Okay.
Pratap, sir, can you please?
Yeah, I'm playing.
Thank you, sir.
73.
Thank you.
Good afternoon, everyone. Myself, Dr. Sonali Patil. I would like to introduce to you the topic potential strategies for ensuring the health of the natural environment. As we know, environmental health is defined as those aspects of human health and disease that are determined by environmental variables. The definition is given by World Health Organization, which includes all physical, chemical, and biological factors which are external to a person, as well as any connected aspects affecting the behavior of the person. The environment has a significant impact on people's physical, mental, and social health. Despite tremendous progress, there are still significant disparities in environmental quality and human health equation. The complex links between environmental elements and human health is thus viewed in a broader spatial and social context, taking into consideration many past interactions. The environment, which is also known as ecosystem, is a natural unit that includes plants, microorganisms, animals, which are biotic factors, as well as all non-living physical variables, which are abiotic variables. The term ecosystem can also refer to human-made habitats, such as human ecosystems and human-influenced ecosystems, and can be used to describe any situation in which living organisms interact with their surroundings. There are various disciplines that can contribute to environmental health, like environmental epidemiology, toxicology, exposure science, environmental engineering, and environmental law. Environmental factors have a significant impact on human health. According to assessment by WHO in 2016, various environmental parameters are responsible for 24% of birth illness burden and roughly a third of the burden in children. So there are a variety of environmental concerns that can obstruct human health and well-being of the human. There are various factors
which can contribute to human health like chemical pollution, air pollution, climate change, disease causing bacteria, lack of access to healthcare, poor infrastructure, and poor water quality. The recent example is being the infection by COVID-19 microbe, because of which we are now facing the pandemic. To address ecosystem and health needs, as well as developing concerns, a broader approach is required, and numerous advances have been made to target initiatives to enhance environmental quality and to reduce the specific cost on human health. But all over the world, it's not possible, and it is only being used in developed countries. In conclusion, as people become increasingly conscious of multi-causality, complexity, and uncertainty, there have been the principles that have to be used, like prevalence and preventive. More acknowledgement of the limits of what we can know in time to prevent harm is required, as the need to act on sufficient rather than overwhelming evidence of potential health effects, given the benefits and drawbacks according to situation. Thank you.
Thank you, sir.
Okay. Is there anyone to ask question? Okay. Thank you, ma'am. Thank you. Next one is abstract number 74. Alpaq Patel. Abstract number 75. Kiran Gaikwad.
Mandar sir.
Yeah.
Yeah. Before proceeding to 75, I will play video of abstract number 74.
74. Okay.
Good afternoon to one and all. Myself, Dr. Alpaq Patel. I am assistant professor in SICS Degree College, Ambernath. And today's my topic is comparative study of chronic cognizance and appearances as per for the skin disorder. Now, in our college, as our college is situated at Chisquare, we are having 4 programs around the college, and as we know that medicinal plants are backbone of traditional medicine, and development.
So, volume is too low.
Yeah, it is the maximum one.
Okay.
Just campus drive and then combined for 2 days, and after the partial drive, there was separation, and the drive is not at over at 50 degrees Celsius for 24 hours. Now there are various extraction methods for both sample and safety body methods, where the drive of both the plants were powdered using water and calcium, and after storing like that, express steroid and dietary, which aspirin appearances as per remaining all the phytochemicals like tannin, alkaloid, phenol, flavonoid, saponin, and coumarin. All the phytochemicals are present in both the. So, as per my study, I came to know that certain phytochemicals are common in both the plants, and that is why both the plants are used by the people for storage food in these orders. And after my whole study, once again I came to know that it before previous study was shown that all dermatological diseases like skin rashes, wounds, skin infections were mostly treated by these 2 weeks, that is Friday, Topical and Appearance Aspera. So I think we can make some common drugs by using this powder of these 2 common things, and combination of various drugs. It's also after the participation in primary healthcare, the combination of various drugs is also done in developing countries. Both the Appearance Aspera and direct processes are having strong therapeutic damage, and especially for the skin
disorders, and therefore the silica has very common active constituents as so dry leaf of powder of these two plants can be used for the formulation of strong drugs, for instance, or the dermatological diseases, and it will be very economical as both the drugs are easily available, so it can be used with all the people. After this, my research, I came to know that definitely you can formulate a strong drug as both of these have strong therapeutic properties for the action. I was thankful to my principal teaching agnostic of my college for constant support and providing all the laboratory facilities to complete this research work. And
Yeah, yes, Mandar sir. So this was all about presentation of Alpha Patel, and then you can continue with abstract number 75.
Yeah. She may not be virtually available to answer the questions, isn't it?
Yeah.
So we'll go for the next presentation. Presentation number 75.
Yes, sir.
Kiran Gaikwad.
Yes, yes, sir. Yes, sir.
Yes, please, sir.
Okay. Can I audible?
Yeah. You are audible very much.
Okay.
Sir, please make your presentation precise and up to limited methodology and findings only, please.
Okay, sir. Okay. Can you see the presentation, sir?
You can start. That will be visible after some seconds.
Okay. Myself, Professor Kiran Gaikwad, Assistant Professor from KNMS Art, Commerce and Science College, Satana. My PhD guide is Kiran Gaikwad, sir. And my today's topic for a presentation is diversity of diatoms in the river Darna of Igatpuri Tehsil of Nasik district. Okay. Introduction. The present study deals with the diversity of diatoms in the river Darna of Igatpuri Tehsil of Nasik district. Different physiochemical parameters were analyzed from the water sample of from a different sites such as the pH, temperature, turbidity, conductivity, etc. The main aim of this presentation is to study and evaluate the biodiversity of diatoms and the impact of seasonal variation on diatoms diversity from which is water collected from the Darna river. Then the material and method. Darna river is a main tributary of Godavari in Nasik district, which is means rising north of the Kalsubai ranges. The investigation was conducted at monthly interval for a period of 2 years, from July 2019 to June 2021. 4 sampling stations were selected from the river through the Igatpuri Tehsil. Different physiochemical parameter analyzed from the different samples. Okay. Then the diatoms are identified by a standard literature, fish, and as well as the online literature algal base. Then result and discussion. Different physiochemical parameters studied from the sample, that is temperature, pH, DO, TDS, nitrate, silica, etc. Many parameters like dissolved oxygen, alkaline pH has affect the growth of different diatoms. Then increase the concentration of silica in the water. It favored the increase the population of diatoms. From all the stations, from all the 4 stations, the diatoms were dominated, followed by the blue-green algae and green algae. Then dominated diatoms species are Cynindra ulna, then the Cimbella aspera, Cimbella cistula, then Navicula minusculus, then Gomphonemma species, then the species, etc. Then there is a sample parameter which is analyzed from the different force side. First is the air temperature, which is analyzed through a thermometer, water temperature by a gradual Celsius thermometer, then the transparency by SH method, then the pH by digital pH meter, then the electric conductivity by digital conductivity meter, then the TDS gravimetric method, and the DO from Winkler's iodometric method, then the total alkalinity from titrimetry method, then the total hardness by a same method, then the fluoride by a arginometric titration method, then the nitrate nitrogen by the spectrophotometrically followed by the Then the dissolved silica from the spectrophotometry, same from the okay. Then conclusion of my research. The physiochemical parameter of river shows that the winter is unpolluted as the dissolved oxygen content is high and concentration of phosphate and nitrate is very low. Okay, then silica was present in considerable quantity, which is indicate that the dominance of diatom in the water, means presence of the silica and the presence of dissolved oxygen in high amount, then the low amount of phosphate and nitrate, which is useful for the dominance of diatoms. Then in the river Darna, the diatom constitute the main algal population from all the stations concerning with the blueberry algae and chlorophyce. The diatom is the dominance from all the station. So the river water can be safety used for the drinking as well as mainly for the irrigation purposes. Okay, this is the some photograph of different diatoms. Synandra ulna, then the Symbella aspera, Symbella cistula, then Navicula menisculus, then the Gomphonemus species, then the Pendularia adolosa. Okay, my acknowledgement for author are gratefully acknowledged to my research guide, Dr. Ken Gaikwad sir. Then,
thank you, sir.
Okay,
Thank
okay,
you.
okay,
Thank you.
okay. Thank you. This is the references.
Any question, please? Okay, thank you, sir.
Okay.
Thank you. Abstract number 76.
Hello, Mandar sir.
Yeah.
The abstract number 76 is video.
Vivek Kulkarni. Okay.
Okay, sir. I will place it.
Today in this presentation, I am presenting the topic that is the main title, it is a targeted analytical field.
Sir, it is audible.
Currently, I am
Volume is, try to increase the volume if possible.
at School of Pharmaceutical Sciences, National University.
Otherwise, let it go as usual.
So this is the abstract for this
volume is too low.
For this presentation. This is the type of nano drug delivery system. So this particular drug delivery system is generally preferred to not have any kind of toxicity. It avoids the proximity other regular formulation. The targeted delivery system for the efficient or for the effective use of these drugs. It will use a higher activity. From that, the transferosome is the second generation elastic lipid vesicle carrier, which having utilized for the hydrophilic drugs, hydrophobic drugs. They are biocompatible, they are biodegradable, they have high interface efficiency, they are more flexible, and they are in capacity. If some may feel hypersensitivity or skin irritation, and generally these are the chemically unstable due to the oxidative degradation. These are some disadvantages of transferosome. This is the structure of transferosome. So hydrophobic drug particles are there inside. Above that, lipid molecules are there. They are enclosed with the surfactant molecule. It is the age activator. Then the composition. So you can say in case of composition, the transferosome, it consists of phospholipids which going in hydration technique, and second one, modified handshaking or lipid switch. Then we can characterize transferosome, that is in case of entrapment efficiency, how much amount of will be entrapped in that transferosome, then those contain, then we can utilize for the division of different kind of drugs. And the main advantage is that due to the targeted, it can reach to the site of action. So if potent drug is there, it must show the proper effect. So we get the in case of regular or the conventional dosage form, what happens? They have to pass different barriers. First pass metabolism, then at the site of action, they have to be, they will show the activity. But in case of this transferosome, that is the nanomaterial biodegradable. Okay. then less amount of drug is required to drug into the transfer. So in case of anti-cancerogen, where we have to penetrate the skin, we have to give the better digestible. They have lots of advantages. That's why transferosome can be utilized, or it is the drug in the future where most of the drugs can be converted into the transferosome formulation. It's for me opportunity to.
Sir, you can call next.
Yeah. Next one is 77. Pranali Vasate and Navnath Kashid. '78. Nilesh Wagh, Abhijit Mirgal, Akanksha Rathod, Rohan Goi, Sumit Khara. '79. Ravina More, Gauri Kadam, Supriya Ghodke, Sujit Nagare. '80.
Hello, Mandar
Yeah.
sir.
Yeah.
78 is video, sir.
Okay, please.
I will
play
play.
that video. Please, sir, share the video.
Yes, sir. Yes, sir. It is visible, sir?
Yes, sir.
Hello, respected sir and ma'am. We are students from Indira Institute of Pharmacy, Karur, where we are studying Bachelor of Pharmacy. Today I will be presenting about extraction, formulation, and evaluation of novel and natural mosquito repellent liquid. Mosquitoes are among the most disturbing blood-sucking insects afflicting human beings. For that, we use marketed mosquito repellent in which hazardous chemical substances present that affect human beings. So we are formulated natural repellent from herbal dust. Now we will see aim and objective. In aim, the aim of work is related to extraction, formulation, and evaluation of novel and natural mosquito repellent liquid. Objective: to extract plant with simple extraction methods, to assess replicancy and larvicidal activity of selected plants with aim of developing the mosquito repellent preparation from plant. Perform acute thermoregulating temperature test according to the term procedure. Evaluate mosquito repellent activity selected plant, then assess larvicidal activity of selected plant, then develop a stable mosquito repellent from selected plant that is safe and then we use materials and computers. Material used for experimenting is powder, the powder of herbal drugs like clove powder, cinnamon powder, chili powder, and lemongrass powder. Then we use essential oils of natural drugs like clove oil, cinnamon oil, chili oil, lemongrass oil. Then chemicals we use methanol, hexane, in which we require apparatus, the RP of glass rod, ultrasonometer. In which instead of ultrasonometer we use plastic box. Then method. First method, plant extraction. Use plant materials of all four plants where water thoroughly to water and then air dry. Later, they were graded into powder using an electric grinder. The extraction of the plant is carried out using maceration method. We accurately 200 grams of powder of each plant and extracted for 2 weeks using 1000 ml of ethanol. The extract were filtered and concentrated for a few days and then stored at room temperature deep fertilizer. Then formulation of the mosquito, in which the mosquito liquid will be formulated by 60 ethanol extract and essential oils of cinnamon, clove, tulsi, and lemongrass, along with ethanol, hexane, and Mispart. Mixture water were added to the mixture until the final value is available. All the constituents were mixed using mechanical stirrer for an hour. Finally, the mixture was poured into an empty container of mosquito repellent, plug-in device, and labeled. Then evaluation of mosquito repellent liquid, in which we take a percentage of the repellent activity used for the number of mosquitoes controlled minus number of mosquitoes treatment upon number of mosquitoes controlled in which we see result that means percentage efficacy is 82.22%. The result obtained from behavior response of the the formulation plastic box introduced alternative introduced plastic box that had 10% concentration. The formulation consists of extract and essential oils of cinnamon, lemongrass, and clove exhibited. Significantly higher effect activity was demonstrable in stage out of 10 mosquitoes inserted. Only one mosquito percentage referral activity of 6.61%. Similarly, the second day, only one mosquito presented. However, the percentage referral activity was quite different of 90%. The highest percentage referral activity was shown during the third trial with no mosquito present in the treatment. Overall, based on the percentage referral activity of the formulation shown a greater referral activity of 82.20%. Then go to the conclusion. Initially, natural based mosquito referral was formulated successfully. The repellent properties of the liquid contain essential oil and plant extract was evaluated and determined. The test is carried outside in order to get the mean percentage. The mean was found to be 82.22%, which is more than half. Thereby, it shows that the formulation is very good. No any harmful effects reported, thus it is totally safe and economical and ecological product. There are references to take. Thank you.
Hello, sir.
Yes, sir.
Next 79 is also video, sir.
Video. Okay.
I will play sir. Video.
Go on, please.
Hello everyone, we the students of India Institute of Pharmacy, Sadabhi Dev presenting the poster for ICEDS 2022 conference. We are glad for the acceptance of the poster. So the topic of our poster is assessment of microbial bioburden on used mask. As we all know, COVID-19 pandemic has distorted the workings of the entire world, and although there is no valid cure for coronavirus yet, there are various ways you can prevent yourself as well as others from getting an infection. Using a mask is one of the most effective ways to avoid COVID-19 infection. In the past months, there has been a surge in the use of masks. However, what's more important is the correct usage of masks and not just the usage. Primarily, there are three types of masks used: cloth mask, surgical mask, and N95 respirator mask. No matter which mask you use, it is important to reuse and dispose of the mask appropriately and responsibly. Coming to our title, basically bioburden is defined as the number of microorganisms living on a surface that has not been sterilized. The aim of bioburden testing is to measure the total number of viable microorganisms prior to sterilization. So, this study was carried out for quantification of microbes on mask used by individuals during COVID-19 pandemic and to estimate the microbial population. Coming to methods of our project, first of all, we had prepared the media. The media used for quantification of microorganisms was the English media. Total 100 marks were distributed between faculty members and students and informed them to use the mask for 24 hours and then collected the mask the next day in a Ziploc bag. Then the surgical masks were tested by initially making an impression of the internal surface of the mask on sterile culture media and incubated for 24 hours in an aerobic humid atmosphere at 37 degrees Celsius. The growth of microorganisms was observed and the colony forming units were then counted on microbial bacterial colonies counter. In this study, all the 100 samples showed the presence of the microbes. The graph represents that 33% masks are found to be heavily contaminated with colonies more than 300 CFC and are considered as TNTC, that is, too numerous to count. 15% masks found to be having colonies in between range 1 to 50. 17% comes in range of 50 to 100. 15% masks found in the range of 100 to 150. 9% found to have faunes in the range of 150 to 200. 4% found to have faunes in the range of 250 CFU to 300 CFU respectively. Therefore, based on the results of the present study, we conclude that surgical masks are a potential source of bacterial shedding, and ideally, it is suggested to avoid reusing the mask after using them once, and should be used properly in proper manner. That is, it should always remove using its string, making sure that you don't touch the front of the mask. With the mask snugly around the nose and must be completely covering your nose and mouth and not just hanging loosely around the neck. Otherwise, the purpose of using the mask is defeated. And while discarding, wrap the mask in newspaper, put it in a polythene bag and discard it. Finally, I would like to end by saying that we should consider the mask as a part of our professional life and try to convince people that there is no way of considering the mask as an enemy, but consider it as a protection, like an umbrella against the rain, like a coat against the cold, and as a sign of civilization to protect our colleges and people around us. Thank you.
Hello, sir. Hello. Raki sir. Hello.
Yes, sir.
Sir, you can call now 82, 83.
Okay.
Okay, sir.
Next one is 80, 8080. Siraj Salim Bhatt, Suhail Ahmad, and Nazima Hamid Mir.
Good afternoon, sir.
Good afternoon, sir. Please go ahead.
Sir, is my screen visible?
Yeah.
Okay.
Please
Good
go ahead.
afternoon, Chairman, sir, and other participants of the conference. Today, in this conference, I shall be deliberating upon the temperate forage genetic resources, sustainable management of fodder deficit, especially during the lean period in northern Himalayas, that is the winter season. If we have a look at our fodder genetic resources and livestock status in India, there's a huge deficit in fodder production and availability for the livestock, which impacts the health of the livestock, which in turn has a profound effect on the socio-economic status and livelihood of the farmers and other entrepreneurs dealing with the industry. Land allocation for green fodder production is just 5% of our grass cropped area in our country, and there is no scope for this horizontal increase, horizontal improvement for fodder production. So, and the status in our northern Himalayan region, that is the Jammu Kashmir, Himachal Pradesh, and Uttarakhand. Here also we have a huge deficit in green and as well as dry fodder throughout the year, especially during the winter season. And the deficit in green and dry fodder in Jammu and Kashmir is 67% and 27% respectively. That is a huge deficit. And the same condition is in Himachal Pradesh. It is 54% and 34% for green and dry fodder respectively. And the concern is decreasing trend in the area of grasslands and pasture lands, because we are facing encroachments, conversions, developmental projects, and some other factors are also there. And at the same time, as we talk of sustainable production of fodder crops or forage production from our livestock, there has to be a shift from sustainable to progressive sustainable production, because keeping in view the increase in the livestock population on one hand and the decrease in the area for production of forage crops on the other hand. So we conducted one project toward this to explore and evaluate major underutilized forage genetic resources in the region, to frame out measures for sustaining the livestock during the winter months. And the third objective was to understand the major issues regarding these forage genetic resources and their solutions. What we did, we took explorations of forage crop genetic resources from different grasslands, pastures, local farmers, and we interacted with the farmers, and we evaluated the germplasm at our farm, and for both qualitative as well as quantitative traits. Germplasm exploration evaluation, it is the basic step in enhancing the forage productivity for meeting the huge demand of fodder for our livestock. There are two types of fodder these crops. There some are annual, some are perennial. It may be cereals, legumes, and grasses. Some perennial cereal legumes and grasses or legumes include red clover, white clover, alfalfa, sainfoin, Egyptian clover, that's berseem, birds, trefoil, crown vetch, and hairy vetch. And we have so many grasses like ryegrass, perennial ryegrass, tall fescue, red fescue, orchard grass, phalaris, brome grass, and timothy grass. These are major perennial grasses in our region. These are some photographs of these grasses. In brief, I wanted to, taking care of time, I can't deliberate on each and every grass, but in general, I can say these are major legumes, and the production of these is 200 quintals per hectare. This for this average, but in sainfoin it is more, because there are so many good things which we have from these legumes with respect to their nutritive quality, their impact on the soil health. They increase the sequester this atmospheric nitrogen and provide good nutrition to the animals. And these are some major grasses in our region. And if we see these legumes, their crude protein content is around 24%, 20 to 24%, while in grasses it is 15 to 17, 18% on an average for these grasses. And we have evaluated all these grasses and legumes at our farm, and we have this is the details of different accession of forage crops which we collected, and they are present at our station, and the same has been deposited for long-term module at ICR and National Bureau of Land Genetic Resources, New Delhi, and the accession numbers of these grass and legumes has been received. This is the evaluation of different temperate forage crops at our station we have done for their this fodder productivity, dry fodder yield, as well as we have done this quality evaluation of all these grass and legumes. So taking care of the second objective, as we see, we have observed that the hay silage making, fodder blocks, concentrated feed, and tree branch fodder, these are major active these sources which provide fodder for our livestock during the winter months. Hay is the most common, and we should take care that the moisture content should not be more than 20%, so that it is protected from fermentation. Silage is like in simple term, I can tell you it is like a pickle, but it is a green biomass rich in soluble carbohydrates, conserved having moisture content 65 to 70% or more for 40 to 45 days in anaerobic conditions. There should not be entry of air. So if it happens, there may be production of butyric acid instead of lactic acid, which is not good for good silage. This lactic acid acts as a preservative and a good source of readily fermented sugars for the rumen microbes. It helps in the this silage. Actually, if you talk of any dairy farmer, he is feeding his animals silage daily, while as in winter months, it is more preferable to feed silage, and in other months when we have green fodder availability, we can provide our animals the green fodder. And in when we have the green fodder availability at our farm, that is better than other the sources. And in a winter month, we need fodder blocks which contain straw, molasses, dry leaves of berseem or other leguminous plants, concentrated mixtures, and other things like that. We also need to do concentrated feeding, which is high in nutrients like mineral mixtures, feed chocolates to feed our animals. And if we see this concentrated feeding, we as a farmer, if we see, we have observed that most of the farmers, they don't do this concentrated feeding. And what happens, especially for the pregnant cattle or livestock, in the later months, when they deliver the calf or they are not able to stand up. So it has to be taken under consideration that this concentrated feeding is must, and at least 30, it depends on the size of the animal, how much concentrated feeding and how much mineral mixture we have to feed the animal daily. And tree fodder is also available in our region. Planting of trees is done before this leaf fall, and the same are dried, stacked, bundled, and for feeding during the winter months. These are some, if we talk of these fodder trees in our region, these include these Grevia optiva in subtropical and tropical areas, mulberry, elm, black locust, poplar, willow, and crude protein in this Grevia, if we say, I have observed that it is crude proteins up to 24%, which is as comparable as Lucina, Leucocephala, these are, and other leguminous. Even in alfalfa, which is a legume, the crude protein content is only up to 24%, and this Grevia optiva, which is locally called as buhl, the crude protein content in this leaf, in its leaves is as much. So, and one more thing is, most of these trees, they are if we take them under agroforestry models, they are ideal species for different agroforestry models and integrated farming systems. These are some photographs I am sharing, interaction awareness camps conducted by me and our station at different farmers fields and throughout the country, throughout the state. And this photograph, first one is at Gurez, the last village from our side. And in that village I have conducted programs. Major issues understood, what we understood, that conservation management of these fodder crops is required, and there has to be germplasm exchange and improvement. Quality seed production is important, which is lacking, and we most of the time we don't involve the locals. That has to be done through participatory management. Grazing management is important. Rotational grazing should be done, and fodder collection should be regulated for the locals, because what would they do? This improper management, what it happens that the animals graze these some selected plants, and there remain some unwanted weeds. They then encroach the whole grassland, and it becomes a menace. So grazing management is important, and we have to improve the condition of rangelands and grasslands. Threats to forest crop genetic resources, which lead to their genetic erosion, that must be checked. And we have to identify alternate source of livelihood and energy for the local, so that they don't depend on the livestock only, and thereby decreasing the pressure on the fodder availability as well as on the forests. So research and education regarding forest crops must be taken up. And these are like what we have observed, that these forest crops, they are mostly taken as orphan crops or underutilized crops. Much research is not done on these crops, and
thank you, sir.
And extension programs and demonstration regarding forest crops are required, and that we have been doing that. And
can you conclude?
Institution is okay, sir. And we should encourage mixed farming. I conclude that there is huge deficit of fodder in the region. These underutilized perennial grass and legumes have a huge potential to mitigate fodder shortage, especially during the lean period. Exploration and evaluation of these underutilized forest crop genetic sources has been done, and is further to be done. Increased productivity from sustainable to progressive sustainable fodder production is the need of the hour, and which has to be done through horizontal vertical improvement in interventions. For sustaining the livestock, hay, silage, fodder blocks, concentrated feeding mixture, and fodder from trees plays an important role. Regarding germplasm, there is need of broadening up genetic base. Introductions, threats to genetic erosion need to be addressed. Improvement programs and development of high-yielding varieties is the need of the hour, and there has to be germplasm exchange at national and international levels. Besides these, we have an immense need for extension programs regarding these forage crop technologies, which must be extended to the farmers level.
Yes.
Thank you, sir.
Thank you, sir. Thank you. Any question, please? Okay, thank you, sir.
Okay, thank you, sir. Thank you, sir, very much.
Thank you. Next one is presentation number 81, Praveen Sonavane and Changdev Pudnar. Then 82, Kaveri Devre, Shubham Kolle and Manoj Patil.
Yes, sir, present.
Please go on. Go ahead.
I'm audible, sir.
Yeah. Please go ahead.
Good afternoon, everyone. I am Kaveri Devre, my friend Shubham Kolle and Dr. Manoj Patil, sir, PG Department of Botany from SNJB Arts, Commerce and Science College, Sandwad, Nasik district. My presentation topic is effect of sodium azide on germination parameters of Vigna unguiculata. First, we see introduction. Vigna unguiculata is a herbaceous legume, belongs to family Leguminosae, commonly called as cowpea and chowli. Cowpea is an annual legume having nutritious component in human diet. Sodium azide is a chemical mutagen, which is most common mutagen in mutation breeding. Mutation breeding is one of the best alternative to traditional breeding methods for crop improvement. These are some objectives. First is to study the effect of sodium azide on germination percentage of cowpea, and second is to study the effect of sodium azide on seedling growth of cowpea. This material and methodology we use to our research. The seeds of coffee were selected. 60 seeds were taken which are pre-soaked in distilled water for 9 hours. After soaking in distilled water, all seeds were treated with freshly prepared chemical mutagen sodium azide in different concentration like 0.01%, 0.02%, 0.03%, 0.04%, and 0.05%. The seeds were kept in mutagenic solution for 5 hours at room temperature. An equal number of seeds soaked in distilled water, which serve as a control. This is the result. Experimental results observed showed that the germination root and shoot length of the seed, which are treated with sodium azide, first is control. Germination percentage is 100%. Average root length is 2.85 centimeter, and average shoot length is 4.5 centimeter. Total seedling length is 7.35 centimeter. Second concentration is 0.01%. Germination percentage is 90%. Average root length is 3.61 centimeter, and average shoot length is 4.42 centimeter. Total seedling length is 8.03 centimeter. Third concentration is 0.02 percentage. Germination percentage is 80%. Average root length is 3.07 centimeter, and average shoot length is 4.66 centimeter. Total seedling length is 7.33 centimeter. Next concentration is 0.03%. Germination percentage is 40%. Average root length is 2.42 centimeter, and average shoot length is 3.6 centimeter. Total seedling length is 6.02 centimeter. Next concentration is 0.04%. Germination percentage is 30%. Average root length is 1.73 centimeter, and average shoot length is 1.7 centimeter. Total seedling length is 3.43 centimeter. Next is 0.05% concentration. Germination percentage is 20%. Average shoot length is 1.5 centimeter, and average shoot length is 1.1 centimeter. Total seedling length is 2.6 centimeter. This is our result. Further work is going on the biochemical estimation like protein, carbohydrates, amino acid, etc.
This is conclusion. Sodium azide is the potential chemical mutagen which is affected on germination percentage as well as seedling growth. From the above result, it can be concluded that 0.01% solution of sodium azide treatment is more effective for plant growth and germination percentage, while 0.05% has low plant growth and germination percentage. As the concentration of sodium azide increases, the germination percentage and seedling growth is decreases. These are some references that we refer for our work. Thank you so much.
Sir, तुमचा माईक बंद आहे.
Hello, hello. Thank you, ma'am. Thank you. Any question, please? No one is asking question. Okay, next presentation is 83. Digambar Kulkarni, Joshi Vishwesh, Shetty Sneha, Rewale Nupur. Okay. Then next abstract number 84. Again Digambar Kulkarni, Mamtule Alfia. Who is presenting? Digambar Kulkarni. Okay.
Naikode sir. Please unmute yourself, sir.
Ma'am, please unmute yourself. You are muted. Hello, ma'am. Okay. It is video being played.
Naikode sir, unmute yourself.
Hello, Naikode sir.
Software use the amount of reflected light absorbance through the RGB background sheet. Laboratory spectrophotometer used in this research is to find the comparative result of the software. So the experimental setup of this experiment is as shown in figure number 3. The respective values shown in figure number 3 give the absorbance value of background sheet which is kept behind the solution. So, and the light used in this experiment is natural source. The light coming through natural source incident on the paper sheet and is reflected. It enters the solution and we take the respective readings and found that the readings can be taken at a particular distance and the distance is 79 centimeters. The readings for smartphone colorimeter software is shown in the observation table number 1 and graph is shown in
and graph is as shown. So as concentration increases, the absorbance will increase. And it is noticed that darker solution has more tendency to absorb reflected light because of its concentration. So, and then we perform the comparative experiment between the spectrophotometer laboratory device and the colorimeter software. As we coincidentally get a chance to come to college, we perform this through laboratory spectrophotometer, and the readings are as shown. So the result from this experiment is for constant concentration, the absorbance for red color is 0.0693. For green, it is 0.151, and similarly for yellow, it is 0.249. And we perform this experiment through spectrophotometer laboratory device and found that the very similar, and the readings for red from spectrophotometer is 0.069. For green, it is 0.156. Similarly for yellow, it is 0.200. Following conclusions are drawn from this experiment. So it can be attributed that the colorimeter software can be used as alternative way for learning laboratory spectrophotometry, as it is a free software available on Play Store, and it is very easy to handle. I'm very much thankful for the following dignitaries for giving me this opportunity to present this research paper with you.
Mandar sir, over to you.
Yes, sir. Yes, sir. Okay. Website number 85. Shweta Shinde, Snehal Gore, and Manoj Patil.
Good afternoon.
Excuse me, sir.
Yeah, please.
Sir, abstract number 84 is 83 is sent to you.
83 and 84.
Yes, both.
84.
Yeah, ma'am, what happened that we received your video.
84 is done, 83.
Yeah, I think 83, we did not have received your 83, abstract number 83 video, yeah. But I will see, I will check. So that was the reason that the video of abstract number 84 was played and not of 83.
Good afternoon, sir.
If it is available, then I will play.
Paper number 21.
What happened, Sridevi ma'am?
Sir, paper number 121. This is the same link for that presentation, sir.
It is the same link, yeah. You will be called afterwards, ma'am. Mandar sir, you can continue.
Okay, thank you, sir.
Abstract number
85.
Yeah.
Yeah. Shweta Shinde, Snehal Gore, and Manoj Patil. Is there any one of them? Okay. Then abstract number 86, Vivek Kulkarni.
I am playing video of abstract number 86. Sir.
86. Okay.
Yeah.
Myself, Vivek Kulkarni. Currently, I am working as assistant professor, Department of Pharmaceutical Chemistry in the Institute of Pharmacy, Sadoville. People are more prone to take the herbal medicine instead of the other medicine. What is the problem with the allopathy of other medicine? That they have a large kind of side effects. They will show you the quick effect or immediate effect, but it shows a large kind of side effects. For example, for the headache treatment of headache, and easily available plant you can see. So we have the different parts of this plant. It's the part, seed, spores, or seed. So different parts we can observe. So all these parts or the whole plant and to go to the extraction of these constituents of this plant it can be better, we can convert that extraction of this plant in the from the family Moringaceae. Okay, so this is the information, kingdom, subkingdom, superdivision, division, class, subclass, order, family, and species. Now, various types of plants have been used, but the Moringa oleifera has been used as a traditional medicinal source. In addition to that, it has been treated for the many of the treating as well as useful for the treatment of different diseases. So already we have seen it is also called as the miracle vegetable. So it is one of the valued plants in the traditional folk medicine. So it can utilize as an analgesic, antiseptic, antibiotic, anti-cancer, antioxidant, diuretic, local anesthetic, food healing, antimicrobial, immunomodulating, antidiuretic. Variety of activities we can observe here from Moringa oleifera. So it is said that, or it is present in most of the at least are responsible for the antioxidant activity, and along with the antioxidant activity or radical scavenging activity, they are also responsible for the treatment of tumors or different types of cancer. These are immunoconstituents. In pharmacological activity, so you can see it's a variety of activities you can observe in case of Moringa oleifera. So antioxidant, anti-inflammatory, these are the main activities. Then anti-tumor is there, anti-ulcer is there, appetite suppression. So generally in the food we are taking such type of the vegetables, but we don't anti-inflammatory. 18 amino acids, 9 essential amino acids. These are the chemical constituents which are present in the Moringa oleifera or the different parts of Moringa oleifera. How to extract? So it can be extracted by simple or complete extraction method. We using the different kinds of solvents, water, alcohol, aqua alcohol, combination of alcohol. There we can see these different solvents. But it is found that the ethanol extract use the large amount of sugars, the large amount of extraction of chemical constituents from the Moringa oleifera. Which are present in Moringa oleifera. These chemical constituents can be utilized for the treatment of different diseases, that is wide variety of disease as well as the case of nutrition. So these activities from food, stain, bark, leaf, flower, pod, and seed. These are only part of this plant having the nutritional as well as the medicinal properties. So that's why this Moringa oleifera offers the value which can form the basis of supplementation. It is considered for the treatment of different diseases as an alternative. In future, we can go for, or we can seek for the providing for the different types of activities. Then, for the better understanding and properties, the isolation of active compound, it is further required to attraction over heat and for the development of new drugs, which having this kind of plant, which having less or no side effects along with the maximum time.
Yeah, it was all about the presentation of abstract number 86.
It was 86, isn't it? Hello, sir.
Yes, sir. Yeah, Mandar sir.
That was 86, no? Just
Yeah, it was 86.
Okay. Then now 87. Sandeep Berde, Om Salunke, Gaurav Bhave, Lavina Fernandez, Salina Fernandez.
Yes, sir.
Okay, please go on present. Please.
My screen is visible. Hello, sir. So my screen is visible.
The screen is not yet visible. It may be later on, but you can start your talk.
Okay, sir. Myself, Ms. Elena Loki Fernandez, Assistant Professor at Indira Institute of Pharmacy, Sadoli.
So the research topic is antibacterial activity of the Pseudium guava leaf, that is guava leaves extracts on the bacterial pathogens. About 80% of individuals from developed countries use traditional medicines which has compound derived from the medicinal plants. Therefore, such plants should be investigated to better understand their properties, safety, and efficiency. So mainly in local tradition setting, plant parts such as the roots or the leaves are used without recourse to the photochemical isolate. So mainly the plant that we have selected, it is guava leaves, several chemical constituents such as coumarins, essential oils, flavonoids, triterpenes, and allicate tannins, which are known to have antimicrobial properties. The leaves of the Pseudium guava tree have a long history of medicinal use that are still employed today. So guava leaves and fruit juice have also been tested in treatment of the infantile diarrhea, and the results showed that those who were treated with guava than the control, and the study concluded the guava had good curative effect on the infantile rotaviral entities. So mainly the plant that is
I'm sorry
Pseudium
to interrupt you.
guava is a plant in the family
Hello, ma'am.
Mispart, that is Mispart along with the clove. Then it also having the similar property that of the eucalyptus.
Hello.
So screen is visible.
Hello. Yes, screen is not yet visible.
Okay, sir. Guava leaves, roots, and fruits have used for the prevention and treatment of diarrhea, and high level of antibacterial activity was detected in the guava leaves. So in several studies, guava showed significant antibacterial
Salinda
activity.
ma'am,
Yes, sir.
are you possible?
Yes,
Please
sir.
share your screen.
Sir, already shared.
But
It's
your
not
photo is
visible.
visible, madam, not the presentation.
Not visible, sir.
No, madam. First, you do your stop sharing, then you share your PPT.
Sir, it's visible now.
It is a blank screen. Okay, madam, continue talking.
Okay, sir. That's Pseudium guava or the guava in plant in the family of Misparaceae. So in this paper, attempt was made to study antibacterial properties of the leaves of the Pseudium guava leaf in various solvents extracts against human bacterial pathogens. Okay, so that plant extracts were tested for the antibacterial activity. From the result, it was concluded that leaves extracts of the Pseudium guava leaf plant was shown effective and efficient result against bacterial pathogens. The Pseudium guava leaves were collected, cleaned, and dried in the shade drying at room temperature for 2 weeks. So this is a main step, which is the first starting step. Then 5 grams of the powder were used for crude solvent extraction. So different solvent that we have used, that is chloroform, ethanol, petroleum ether, and the water. These are the different solvents we have selected. Then extracts were filtered out and filters with residues were used. The solvents was then evaporated to dryness under reduced pressure, and the extracted compound was used for the antibacterial assay. The bacterial strains used for the test were Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, and the Salmonella typhi. Then we have used the Muller-Hinton agar, and the nutrient broth was used for the media for the culturing the bacterial strains. Then lupul of the bacterial cultures were inoculated. in the nutrient broth and incubated at 37 degrees Celsius for 24 hours. Then this is an assessment of the antibacterial activity. That sterile filter paper dish, 50 microliter of each extracts air dried to the eliminates residual solvent and were placed on the surface of the inoculated plates. The plates were inoculated at 37 degrees Celsius for 24 hours. Then result and discussion. Plant leaves is used as a medicine against gastroenteritis, diarrhea, and the toothache by those who cannot afford, don't have access to the antibiotics. The present study screened the antibacterial effect of the leaves, food extracts of the cover. So these are the different results in the tabulated form. So this is a extractions in the chloroform, ethanol, then petroleum ether and water against E. coli, Staphylococcus aureus, then Salmonella typhi, and S. aureus, that is Staphylococcus aureus. Then this is the graphical representation for the chloroform extracts was shown maximum activity against Staphylococcus typhi and zone of the inhibition is 12 nanometer and lowest activity against Staphylococcus aureus and E. coli with zone of inhibition 10 mm. Okay. This is a graphical representation for E. coli, Staphylococcus aureus, Salmonella typhi, and Pseudomonas aeruginosa as well as Pseudomonas aureus. Then in this figure 2, it is shown that antibacterial activity of the leaves extracts of the chloroform ethanol of Cerium guajua against Staphylococcus aureus, Escherichia coli, and the Pseudomonas aeruginosa, as well as on the Salmonella typhi. So this is the antibacterial activity of the guajua leaf extracts have been associated with the flavonoids such as mucin glycosides as well as quercetin glycosides. However, the guajua leaf extracts showed minimum activity against the fungal pathogens investigated in the present study. The oil has antibacterial activity, particularly Bacillus subtilis and Staphylococcus aureus. So these are the different organisms, and this is a figure 2 that shows the antibacterial activity of the leaves, crude extracts of the Cerium guajua of the pathogen bacteria. Conclusion: the present study has helped in demonstrating the potential bioactive compound of the natural plant extracts that eco-friendly, economical, and available in bulk to farmers with easy preparation protocols. The Cerium guajua plant parts are used to develop for the various industrial pharmaceutical products. In this paper, attempts was made medicinal properties of the leaves Cerium guajua in various solvent extracts against human bacterial pathogens. From this the result, it was concluded that leaves extracts of the Cerium guajua plant was shown effective and efficient results against bacterial pathogens and it having the antibacterial activity. So these are the different parts. Thank you so much.
Thank you, ma'am. Thank you.
Mandar sir,
yes, sir.
The thing is that abstract number 64, Sandha Jagtap ma'am has already sent one video regarding her presentation, so I will play that one. Abstract number 64.
64.
Yeah, role of data science in modern world.
But that can be considered. Okay, no problem.
Yeah, I will play within.
Yeah.
Data science uses that data for the various that extract the knowledge from the structured and unstructured data. In data science is applied data uses and algorithms for the extract the knowledge from the data. Also for the finding the certain patterns. Various things are associated with the data science. First one is the artificial intelligence. Artificial intelligence exhibits the machine or on the basis of data and image classification. Machine learning. It automates the any machine. Learn the machines on the basis of the data. It is a subset of artificial intelligence. It creates the algorithm that operates on the
have a agency data science also in the industry. What is the data is very large. The help of data science technology. Data analyzing what data science also helps done by the data whatever you can surface on data science is all the new technologies the possible to find a microscopic standard also correlating and depending upon the part the doctor will use the doctor but diagnosis purpose all these predictions are declared before any match will be discussed as possible by using the data science, data visualization, and data mining. Data science also comes in the atom options, also the airline, also the finance, also like a CQ app. Let's take the scope and features of data science in the world. Data science will be ability to take a challenge.
Over to you, Mandar, sir.
Yes. Thank you very much. Abstract number 88. Manish Kumar Gupta, Ketaki Dhane. Virendra Srivastava, Supriya Ham, Sujit Nagare, and Abhinandan Patil.
Yes, sir.
Is there anyone of them to present?
Yes, sir. Ketki Dhane.
Okay. Abstract number 89. Kondamudi, Sai Sindhu.
Sir, I'm there. 88.
Hello.
Huh, sir. Sir, I'm there. 88.
88 are you there?
Huh.
Okay.
Can I present my slides, sir?
Yeah. Yes, sir.
Okay.
Please go ahead.
Thank you, sir. Can I start, sir? Is the screen is visible?
Yeah, screen is visible. Go ahead, please.
Good afternoon, everyone. Novel coronavirus took birth in 2019 in Wuhan city of China. Okay, so myself, Gauri Nilesh Thatte, Indira Institute of Pharmacy, Sadavli, under the guidance of Mrs. K.S. Dhane, ma'am. May I start, sir?
Please go ahead, ma'am.
Okay. Novel coronavirus took birth in 2019 in Wuhan city of China. In a very less span of time, it found its place worldwide with around 4 lakh cases till May 2020. The World Health Organization declared it to be a global pandemic by February 2020. The coronavirus attacks the human respiratory system and acts as a pathogen in the lungs. Its structure comprises of positive sense single-stranded RNA virus as seen in the picture. The coronavirus is a non-segmented RNA virus which is divided into 5 genera, namely alpha, beta, gamma, delta, and it is capable of infecting mammals as well as birds. The virus can be transmitted from bats via some unknown host and infect humans. Based on various genome sequencing results, bat has been suspected as the natural host of virus origin. The novel coronavirus contains various proteins like S protein, M protein, N protein, and
हमारा है ना पेन?
You can continue, ma'am.
These proteins are used for various purposes like determining host range, transport of the nutrients, interference with host immune response, and many more. The virus has a structure comprising of helical RNA protein nucleocapsid and various glycoproteins. These glycoproteins make up various characteristics of the novel coronavirus. The other group of the virus express another glycoprotein, hemagglutinin esterase, which forms spikes on the virion. The genome structure of coronaviruses. The virus encodes 5 structural proteins, namely spike, membrane, envelope glycoproteins, hemagglutinin esterase, and nucleocapsid protein. The various glycoproteins like S and M give the virion its shape and play key role in regenerating virions respectively. The pathogenesis of coronavirus includes various clinical manifestations which are common, such as fever, cough, myalgia, fatigue, and altered leukocyte counts. The coronavirus makes its entry into the host cell via the S protein. The infection is caused by membrane fusion between the virus and the plasma membrane. The virus germinates in the Golgi intermediate apparatus of the endoplasmic reticulum. The virus particles are finally released when the vesicles merge with the plasma membrane. The various diagnostic methods of COVID-19 include LFIA, NTS, ELISA, and RT-qPCR. The LFIA stands for lateral flow immunoassay, which detects the presence or absence of target annulate, that is the pathogen. NTS system method is nanopore target sequencing used to check nucleic acid mutations, and it gives result in almost 6 to 10 hours. ELISA is enzyme-linked immunosorbent assay, and the most common RT-qPCR stands for real-time quantitative polymeric chain reaction. The symptoms of the novel COVID-19 disease include fever, cough, fatigue, headache, diarrhea, dyspnea, lymphopenia, and some abnormal features like acute cardiac injury and respiratory distress syndrome. As such, there is no special antiviral agent available for the treatment of SARS-CoV-19 virus, but it can be treated supportively with oxygen therapy, conservation fluid management, and use of broad spectrum antibiotics. The antibody and plasma therapy is also very helpful in this pandemic. Many convalescent patients are willingly donating plasma against the virus, and also it has acquired good results in acute and severe patients. It can be used as an alternative for the treatment of the virus. We can have some future directions to control the spread of disease, such as reduction in person-to-person transmission, improving of one's immune system, provision of decontaminating reagents, use of sanitizers, masks, etc. And we need to screen while traveling, such as checking the body temperature, oxygen level of the people around. We can see visually how to protect ourselves from COVID-19 and help this pandemic reduce. Thank you.
Thank you, ma'am. Thank you. Next presentation will be abstract number 89. Kondamudi Sai Sindhu. Is it there?
That's it.
Then abstract number 90.
Mandar sir.
Yes, sir.
Yeah, so abstract number 83, Digambar Kulkarni, Joshi Shetty Shweta, Revai Nupur, and Mohite Sejal. They have also sent video, so I am playing the video.
Good evening, all of you. Myself, Sneha Narendra Shetty, one of the participants. It gives me immense pleasure to share the research paper here. The title of our research paper is Synthesis and Characterization of Cobalt Zinc Ferrites by Soil Gel Method. Without wasting time, I will start introduction. In this research paper, we study the cobalt ferrite nanopowder obtained by soil gel autocompression method using citric acid as a fuel material. The source materials we used are zinc nitrate, cobalt nitrate, and ferric nitrate. The metal nitrates to citric acid composition was taken as ratio 1:1. pH maintained at 7. The prepared powder of the size and the properties of spinel ferrite greatly depend upon pH, fuel, stirring time, and speed, as well as metal nitrate to fuel ratio. Now we will see the preparation of ferrites. The source materials we use are cobalt nitrate, zinc nitrate, ferric nitrate, ammonia, ethylene glycol, and citric acid. The compound was stirred with distilled water to reduce the impurities found in the final product. The pH of the compound is maintained at 7 by dropwise addition of ammonia. Then heat the compound and stir it. Then we get the final product. Now the characterization of final product has been done, that is XRD and IR. The table shows the HKL planes for zinc cobalt ferrite. XRD characterization was done to understand the structural properties of cobalt ferrite. These are the diffractograms of XRD characterization of zinc cobalt ferrite with X is equal to 0.1, 0.2, 0.5, and 0.3. We conclude from the XRD characterization that we understand the formation of spinal structure ferrite. Then reflections observed at 220, 311, 400, 422, and 511 are having highest intensity. The strongest reflection comes from 311 plane and it denotes spinal phase. Broad XRD lines show that particles are of nano-size range. We see the similar results in the given research paper. Then we done IR characterization. The table shows the frequency range of zinc cobalt ferrite. These are the graphs of IR characterization with X is equal to 0.4 and 0.8. From our characterization, we conclude that, and from figure, we get that the formation of metal-oxygen bonds has been done, and the transition waveband mu1 to mu4 in the range of 3495.49 to 602.31 corresponds to formation of oxygen bonds. FTIR characterization has done to understand bond formation. It is a rapid method of characterization and used to and help us to understand electrical and magnetic properties of ferrites.
Hello, Zaki sir.
Yes, sir. Yes, sir. Yes, sir.
There is a
There was a
problem.
connectivity problem in between.
Yes.
I could not.
Sir, you can call the, if it is over, the first participant, you can call the next.
Okay, 81 and 90 is not available for presentation.
90 is not available. So 90, I have video of 90.
Okay, okay. Are you going to play it?
Okay, sir, I will place a video.
Okay.
Video. Sir, it is visible?
Yeah, yeah, visible.
Microbial bioburden testing is normally defined as the number of bacteria living on a surface that has not been studied. The purpose of testing is to enumerate the quantity of viable microorganisms on various surfaces. Smartphones are considered to be the main route for transmitting microbes and infections to the plant. The purpose of the experiment is to study microbial count on the mobile phones of a college student and to aware the people about the importance of cleanliness and hygiene of equipment for daily life. We can address students about maintenance of personal gadgets. We can study various preventive measures for the microbial contamination. We use DNA media for the growth of bacteria. Sample collection from mobile phones. We took 100 samples of two mobiles from college students. The sample was collected from the smartphone by the help of earbuds. The earbuds were previously sterilized to ensure that there is no presence of microorganisms. Then we took that earbud and kept it in sterile Ziploc bag. Preparation of a meal. Suspend 39 grams of de-English neutralizing drugs in 1000 ml of a distilled water. Then we boil it for the proper dissolution. Then we sterilize it by autoclaving under 15 Celsius pressure at 91 degrees Celsius for 15 minutes. Sampling process. All the prepared plates were sterile in nature. We remove earbud from sterile Ziploc bags aseptically and spread on de-English media by gently spreading. Remove earbud after gentle spreading and ensure that there should not be heavy pressure applied to the spread. Then we took that earbud and packed in Ziploc bag and sent temperature in incubator. After 24 hours of incubation, we examine cells under colony counter and count colony, record the result. But we can see in the pie chart, it represents that 35% of mobiles are found to be the heavily contaminated with colonies more than TNT, that is too numerous to count. 1% mobile found to be heavily colonies in between range 250 CFU to 300 CFU and 200 CFU to 250 CFU and 150 CFU to 200 CFU. 20% found to have colonies range 50 CFU to 100 CFU. 30% found to have colonies in range 1 CFU to 50 CFU. The study revealed that the almost third of the smartphones were heavily contaminated. There was no significant difference in source of bacteria between the mobile phones of the participants with the gender specificity and other factors. 57% smartphones found to be less contaminated with count less than 100 CFU per smartphone. However, every individual must ensure that there should be proper care of smartphones from being contaminated by microarray. As we can see, mobiles may lead to the spread of bacteria. So we have to find the preventive measures for it. We can sanitize smartphones by using non-alcoholic solution. We must avoid frequent contact with hygienic surfaces. We can use sterilized wipes and microfiber cloths to clean mobile phones. We must keep the phone out of the path. Thank you.
Yes, sir. Yes, sir. Yes.
Mandar sir.
Yes.
Is there any date?
It is over now. Yeah, the first session is over, isn't it? The presentation which are expected are over, I think.
Okay. So thank you, dear participants.
Hello.
Hello. Hello. Good afternoon, sir.
Yes, ma'am.
Sir, I am Dr. Bharti Mahajan. My abstract number is 109.
109. Yeah, that will be. The new session will start within a minute, and you can present in the sequence whenever the announcement will be there of your number.
Okay, sir. Is this the same link?
Yeah, same. Here only.
Okay. Thank you.
Hello, Microarray sir. Hello.
Hello.
Hello, Pansode sir.
Hello.
Sir. We are unable to hear the voice of Microarray sir. He is not audible now.
Can you hear?
Hello, this is Deepali Varange. Hello.
Hello, Microarray sir.
Yes, sir. Yes, sir. Yes, sir. Sir, you continue, sir.
The presentation is over for the first session. The second session of the presentation will start immediately now. Before that, 40 presentations were expected at the outset, but out of those, 27 researchers have presented their research.
Sir.
Yes, ma'am.
Sir, myself, Dr. Kokate from Chikhallara, Amravati district.
Yeah.
Sir, I had to present my paper, sir.
Just ma'am. There are 2 sessions, ma'am.
Okay, sir. My number is 102.
Yeah. The people.
Next session.
People
Okay,
after
sir.
91.
Okay.
People after 91 will present now. Okay. Before that, I have to give a comment on the presentations, earlier presentations. Okay. So,
Okay, sir, continue.
okay. So 27 researchers or professors have presented their research. After going through these presentations, I'm very much happy to congratulate all the people or all the presenters that I could acknowledge the hard work they have taken in doing the research and bringing the virgin knowledge to contribute in the body of the knowledge of our environment and society. I would like to congratulate all and would like to give best wishes for them for their future endeavor. And I would like to hand over all the things with the organizing committee, Dr. Microarray sir as well as Ranjit Panchole sir. Thank you very much for giving the opportunity to work as a chairperson of this session. Thank you all of you and thank you all the presenters. Thank you.
Okay, thank you, sir.
Panchole sir, you can continue.
Okay, okay, thank you, sir. Thank you, sir. The next presentation session is starting now. I'm going to play the abstract number 91. I think he has shared a video of his lecture. I'm going to start his video.
Hello. Here we are presenting the review on cell-based phenotypic approaches in drug discovery. Now, let us just get to the point that will be discussed in this presentation, and the points are: introduction, aim, objective, drug discovery and development by cell-based phenotypic approaches, identification and quantification of cell phenotypes, that is cell profiler, opportunities and challenges of phenotypic drug discovery, conclusion and references. Introduction. Phenotypic drug discovery is used for finding out new medicines. Peptides, RNA, are used which can be alter phenotypes of the cell in the phenotypic screening. It has much scope in biological research and drug discovery. And to introduce target-oriented drug discovery system and to fulfill medical needs in phenotypic drug approaches. Drug discovery and development by cell-based phenotypic approach. First, pharmaceutical modeling and artificial intelligence. Second, repurposing and repositioning of existing drug molecules. First, pharmaceutical modeling and artificial intelligence. Here, advantages using software simulation of drug receptor binding process is observed. It is virtual screening and also called as computer-aided drug design. Second, repurposing and repositioning of existing drug molecules. Repurposing is the new therapeutic effect of old drug observed. And for repurposing, repositioning of the primary molecular structure should be done. Key advantages are faster development time, higher approval rates, and lower overall development cost. Cell profiler first load the image processed through image processing module. Then processed image is passed through object identification module. Then identified objects were processed through measurement module, and the measurement of the small molecule is completed. Opportunities are compound sources, that is lack of data which will be provided precursor of medicine. Mechanism of action and the target ID. The identification and validation of the target is necessary. Safety issues. The mechanism of action is unknown. There will be safety issues. Feature. First, data collected all over the year connect the bridge between drugs. And second, by adopting phenotypic assays and platforms enhances the understanding mechanisms. Hence, it makes drug more safe. Conclusion is the phenotypic drug discovery as a powerful setup for drug discovery. The cell-based under various in vitro and in vivo models offer the possibility to model disease phenotypes and conduct therapeutic screen in large numbers and accepted that condition and accelerated drug discovery.
Okay, the next paper is 92. Formulation and comparative anti-ulcer activity of developed formulation and marketed formulation. Gupta, MK, Khade, MA. Is there anyone? Is there anyone from this? Okay,
Hello, sir.
the next. Yeah.
Sir, this is Deepali Warange. Serial numbers 9 and 10. Sir, I have two papers.
Yeah, madam. Number is your number, Deepali?
9 and 10, sir.
Madam, I will go to the ascending order. I will call. Okay, madam. No problem. I will call. 93, paper number 93, pediatric, Shivaji Andres, sir.
Yes, sir. Are you showing my screen, sir?
Yes, sir. Yes, sir. Yes, sir. You can continue, sir. You are audible and your screen is visible, sir.
Okay, sir. Thank you. Respected Dr. Ranjit Bansal, sir, today's conference chairperson. I will present here a few pieces of this pediastrum. My topic is pediastrum, a little star in a Nandur Madmeshwar Wildlife Sanctuary. Nandur Madmeshwar Wildlife Sanctuary is located in a Niphad Tahsil of Nashik district in Maharashtra, popularly known as a Bharatpur of Maharashtra. Why it is called as a Bharatpur? Because so many species of migratory bird are come here, and it is available of huge amount of food material in this Nandur Madmeshwar dam. It is considered as one of the important waterfall habitat by the International Union of Conservation of Nature. There are the few pictures of this Nandur Madmeshwar. During this winter, and millions of birds, more than 213 different types of the species of the bird visited this water body, and 18 different of the species of migratory bird are come here. About 25 to 28% of this area of the water body is a shallow. Due to this shallow water body, there is a huge amount of algal species can be grow there. As well as the algal species not in, but also in angiospermic species, and there are the certain of this migratory bird, certain of the herbivorous, certain are carnivorous, certain of the omnivorous. That herbivorous bird is depending on this algal species. Their depth is a very less of this that region is 1 meter. This region is biologically active, having a large number of the flora and fauna. A study of the vegetation of the water body was undertaken during the year in 2018 to 2020. During the study, a large number of planktonic algae like Pediastrum, Hydrodictyon, Clostridium, Phacus, Euglena, Cosmerium, and Diatoms were observed. The present paper describes some of the species of the Pediastrum only. There are these first species of this Pediastrum simplex. This Pediastrum simplex, this colony is a oval or rounded structure, and inner side of this marginal cell there is a straight, and outer of this produced into their gradually tapering to them, and the cell wall without their intercellular space are present in this species. Then in the second species of this Pediastrum simplex variety is a Diodranum. These are the colony. There is a large intercellular space and acute their apex in this marginal cell, and they are the concave in structure. They are prolonged and into their single. There is a delicating tapering their process. Acute their tapering process of this Diodranum basis. Then another species of this Pediastrum simplex. This is the new variety. I will first identify in that location. This variety has a circular colonies. Why these are the new variety of the simplex species? There is a perforated terminal part of the cell they're drawn into a single process and curve. Only the species, these are looking like the curve in the apex of this pediastrum simplex species. Then another species of this pediastrum is the pediastrum angulosum, variety is a levigatum. In this species, the colony is 8 or also in a 16 their cell number or 32 cell number, they are present to them, and the cell are very compactly arranged to each other without any intercellular space. And there is the interior cell, they are irregularly hexagonal like structure. This is the specific their identification. Then marginal cell is transferably elongated. Another species of this pediastrum, pediastrum boreanium. They are the colony is circular to oval in structure. Usually these colony they can be formed about 16 to 30 cells. They can be developing to them. Cell arranged in concentric ring without their intercellular space. Outer space of marginal cell with their two short their process. They are present in each of this cell. And another of the species of this pediastrum, they are looking at this pediastrum boreanium, variety is the brevicorni. In this cell, it is also without their intercellular space, and this cell they can be developing in their oval or rounded their structure, and outer cell with their very short their process, and inner cell is that prolonged. There is a hexagonal also in their structure they can be developing to them.
Andreas sir.
Yes sir.
Sorry for disturb, but you go with your slides, sir.
Okay.
Your presentation is not playing, sir.
Okay.
Sorry for disturb, sir.
Then another species of pediastrum boreanium, and is another variety is a longicorni. And this species is also rounded in the structure. They can be also operate to them. There is a two projection in each of the cell. Then another species of pediastrum duplex. In this cell, this pediastrum duplex, there is a certain of the few of the intercellular space are present, and two projection they are this short. They are present to them processes. Then another species of this pediastrum duplex variety is the clathratum. This cell there is a very larger and very larger intercellular space, and colony form the 8 to 64 their cell they can be developing to them. Another cell of the pediastrum duplex variety is the genianum, and this species about 4, 8 or 60 or 32 cell formation. There is also two projection are present in each of the cell. Then another species of the pediastrum, there is a pediastrum duplex variety is the gracillium. And these species also very larger intercellular space, and each of the cell there is a two projection are present to them. Then these are the another species, this is the pediastrum glanduliform perum. Why this glanduliform? Each of the projection there is presence of the gland like structure appearance they can be present to them without their intercellular space. Then another species is a pediastrum integrum. Each of the cell having a two projections very shortly and without their intercellular space. Then another species is the pediastrum ovatum. They are the formation of the colony about 4 or 8, 16 their cell formation, and these cells there is a arranged in the acute their apex and larger intercellular space of this each of the cell of the pediastrum ovatum. Another species of this pediastrum tetras, variety is a tetradron. These species there is a very compactly arranged, and there is a looking a hexagonal structure and very short their processes. They are just few of the references of the used for the identification of the species. Thank you so much. Have you any questions, sir?
No, sir. Thank you very much.
Thank you, sir. I would like to thank to Dr. Pratap Naikwadi, sir, coordinator of this conference, and I will thank to respected chairperson Ranjit Bansal, sir. I would also like thank to our principal. Thank you so much, sir.
The next abstract is 94. Impact of Russia and Ukraine war on Indian economy. Rupali Ode. Rupali ma'am, are you there? Okay, no problem. The next abstract is 95. Morphology and anatomical study of Thania somnifera. Sabiha Visayat. Sabiha madam, are you there? Okay, the next 96, abstract number 96, impact of nickel prices on electric vehicle industry. Piyusha Telam. पियुष येतील? Okay. Next, the abstract number 97. Kinetic studies of adsorption of ferrous metal ions from its aqueous solution with natural adsorbent. Chaudhary SV. Chaudhary SV. Okay, the abstract number 98. I have the video of abstract number 98. I will play that video.
Good morning, everyone. We are the students of the Bachelor of Pharmacy studying the Intrinsic of Pharmacy Saturday. Today I am going to present challenges to the natural products. Content of my PPT is aim, objective, introduction, the role of natural product in the drug discovery with challenges in the natural product research, the importance of standardization, challenges in the configuration assessment of natural product conclusion and reference. Now, introduction. Natural product contain some chemical substance produced by the living organisms like microbes, which are found in a nature, or it may be prepared by the chemical synthesis. Challenges to the drug discovery in such a technical barrier to the screening of isolation. Aim and objective. Challenges to the natural product, bringing the studies on all aspects of natural product, including plant source, animal source, microorganism source, marine source, the standardization and bioactive natural product. Collection of information regarding the different challenges to the natural products. Role of natural product in drug discovery. First is the plant source. Most of the biological active natural products are made up of the plant source. For example, morphine. Common challenges in the production of plant-based natural products. Current challenges to the use of natural products and difficulty in accepting their therapeutic efficacy. First is the lack of standardization procedures. Second, a lack of isolation of pure chemical products or compounds. Third is lack of elucidation of biological mechanism and rarely undergoing so-called controlling, and fourth, documented clinical trial according to standards. Animals of the Ecuadorian poison crop. The common failure of mouse models, the human inflammatory diseases, potential interpotent source of the drug A, for example, penicillin. Marine source. Marine natural product can be defined as a biological active products as a secondary metabolite as well as enzymes, lipid, and heteropolysaccharides. In a recent year, there has been a great interest in finding a pharmacological active natural approaches, taxonomic expertise, biodiversity access, supply and technical challenges, variability identification of the MNP producer, sustainable supply, rate is critical to ensure the consistent pharmacological and clinical results. CV technology has been made a great strides in the resolving these challenges through the patented technology, chemobioprin. During the early chemobioprin products development, the optimal active component of a natural extract are identified and characterized chemically. And continues to be a cases. Conclusion. Presentation of the selected natural product areas offer a different challenges to the natural product community in its various forms to address some pertinent issues in the development of the natural products for the enhanced global health. It also presents a brief discussion on the impact of plant animal microarray-based challenges, the use of challenges to overcome the problem associated with the manufacturing, storage, stability, standardization of the natural products, and some references are. Thank you.
Okay, the next presenter is abstract number 99. Is there anyone from 99? Varad Nalaude.
Namaste. I'm Dr. Sujata Ajbarkare, Assistant Professor of Medical Campus College. Today I present my topic, water harvesting system in Chandrapur, region of Undan.
Is there anyone from 99? Okay. 100, abstract number 100.
Yes, sir.
Aruna, C and Asha.
Yes, sir.
Okay, madam, you can present. You can share your screen now.
Is my screen visible, sir? Sir, is my screen visible? Sir, is my screen visible?
Yes, it is visible.
Yeah, okay, sir. Thank you, sir. Myself, C. Aruna, doing my research work in the Department of Civil Engineering, Annamalai University. My co-authors are Sheila and Asha. My title is Pursuant Study of Hybrid Bioreactor During Startup for Treating Industrial Wastewater. Environmental pollution is the greatest danger that the whole civilization is facing right now. Pollution is the addition to the ecosystem of something which has a detrimental effect on it. So it is important to treat the wastewater and make reuse of it. Main objective is to evaluate the feasibility of the laboratory scale hybrid bioreactor model to treat the industrial wastewater. Experimental methodology, this model has fabricated by plexiglass with a working volume of 13.3 liters. This reactor was accomplished with both suspended and attached growth process, and oxygen was applied in the entire process of operation. This is a photographic view of an experimental setup. Startup process. The reactor was operated under mesophilic temperature range 25 to 35 degrees Celsius. The startup stage was being continuously feeding of the reactor with an influent COD concentration from around 544 mg per liter to 600 mg per liter. In the initial stage, the COD removal rate was around 7 to 13% because of the adaptation of biomass to the new environment. And during the startup period, the pH and DO plays an important role. And the graph between time and days with pH. The red one shows the influent pH and the blue shows the effluent pH. Time and days with a percentage COD removal efficiency. The COD reduction was attained 7% in the initial stage, and it was incremental up to 25th day, and suddenly decreases from 25th to 27th day, and then it attains a steady state from 28 to 32nd day. Results and discussion. The influent and effluent samples from the reactor were collected once in 3 days, were analyzed immediately. Initially, it was collected from the municipality Chidambaram. Sorry, here I mentioned wrongly. And feed to the reactor. Then the low initial loading rate was recommended for the successful startup of bioreactor. Prompt startup is essential for the highly efficient operation of bioreactor due to slow growth rates of microorganisms. The result showed that the HBR attained a steady state from 28th day to 32nd day, and the maximum COD removal efficiency was achieved at 96.45%. Conclusion. Startup is often considered to be the most unstable and difficult phase in aerobic digestion. Aerobic treatment plays an important role in the treatment efficiency. Generally, any aerobic treatment process attains a steady state for long duration, like around 40 to 50 days. But in this HPR system, we attained a short duration due to the implementation of both suspended as well as attached group process in a single reactor. The result showed us COD removal efficiency was maximum is 96.45%. These are some of the references. Thank you.
Okay, madam. It's a kind suggestion is that next time you will use the good to see font.
Sorry, sir. I'm presenting in my mobile, so I'm extremely sorry, sir.
But next time you will take care of
Yeah,
this.
sure, sir. Sure, sir.
Okay,
Thank you, sir.
The abstract number is next abstract is 101. 100.
Yes, sir. Yes, sir. I'm there. Dr. Archana Chaudhary.
Madam.
Yes, yes. I'm
You can
there.
start the presentation. Madam.
Yes, yes, yes. Sir, is my screen is visible?
Yeah, madam. Yeah.
Yes, sir.
Your screen is visible. Your sound is also.
Yeah. Yes, sir. Respected Chairperson, Organizing Committee, and dear participants, I am Dr. Archana Chaudhary, Associate Professor from SSVPS Gogure Science College, Dhule. My title of the presentation is Algal Taxa from the Water Filtration Plants in Dhule District. So this is the water filtration site. The algal growth that causes problem in the water treatment plants. Algal troubles are quite considerable because the cost and efficiency of the working plant really having the effect on it. The algal growth cause some nuisance in this filtration plant. For example, they can clog the filters and they also add the cost of the filtration plant. Actually, these algae are coming from the water reservoirs which are nearby to this filtration plant. These water reservoirs, their available surface area, their stagnancy and new environmental factor can promote the algal communities in this water. And from this water, these spores can carried out in this filtration plant and they cause some kind of a nuisance in this particular working of the filtration plant. So the present study includes the report on the algal taxa present in this water treatment plant of the Dhule district. Objective is study of algal taxa from Dhule water filtration plant. Actually, the problem is associated with the algae in water treatment plant in having both direct and indirect nature. The algae can having the influence on the water quality due to their metabolic activity. Some mucilaginous algae can clog the water filters. So it is having some economic value, and this again increases the cost of the treatment. So working efficiency and execution of any treatment plant mostly depend on the nature of the raw water supply, which comes ultimately from the resources like river, lake, reservoirs nearby to it, which are rich in algal spores. The review of literature then in this field, Tarar and Mazumdar, then Mahapatra have work in this site. Palmer having much more concentration on this particular problem. Material method, that study area, the Tapi River, that water purification plant is selected from this North Maharashtra region, Dhule district. Then the field work is done that the algal samples were taken from this treatment plant time to time, 15 days and within a month twicely. Then microscopic observations were done. Then the algal taxa were observed with the standard monograph from the Desikachari, Sarude Kamath and Philippos. These are the site of the water filtration plant. And then this discussion. Interference of the algae most of the time cause the problems in water treatment plant. Algal growth on the basin and filter wall not only are unsightly, but also give very bad impression of the plant sanitation. Presence of the algae creates many problems with respect to the maintenance and working of the plant. It increases the challenges for its operation, efficiency and cost of the drinking water treatment. Summary, raw water of this Tapi filtration plant harbor many algal forms belonging to the Cyanophyceae, Chlorophyceae and Bacillariophyceae. About 22 algal taxa were reported. Cyanophyceae of 9, Chlorophyceae of 3 and Bacillariophyceae of the 10. This is the list of the algal taxa, and these are some microscopic photographs we have taken. These are particularly Synedesmus, Crucoccus, this diatom, then Clostridium green alga, then Spirulina, this Fragillaria. These are the references. Thank you, sir. If you have any question, please ask. Hello, sir.
Okay, thank you, madam. Thank you.
Okay.
Okay,
Thank you, sir.
Next abstract is 102. Ujjwala Kokate, ma'am.
Yes, sir. Yes, sir. I am here, sir.
Oh, madam, you can start your presentation.
Okay, sir. 1 minute. 1 second. Sir, can you see?
Yeah, madam. Yeah.
Okay, okay. Good afternoon to all the respected colleagues. Myself, Dr. Ujjwala Kokate, Associate Professor and Head from Department of Botany, Arts Science Commerce College, Chikhallara. My topic is study of some aromatic medicinal plants of Melghat forest, which comes under the Amravati district in Maharashtra. We all know Melghat is the forest which is of the dry deciduous type of forest, and it is the paradise of Vidarbha. This Melghat means the meeting of God. In this Melghat, there are Goan, Korku peoples living, and all these people uses the medicinal plant for their daily uses as well as for the cure of their disease also. They are totally depend on the forest. This Melghat, it is having the much more biodiversity for the flora and fauna also. This Melghat forest contains all the medicinal plants which is usable to our cure the diseases. The people from the Ayurveda as well as the botany, forest departments all comes here and takes the benefits of these medicinal plants. So this is my attempt to show all the some of these medicinal plants because there is a lesser time we have. So I will show you these medicinal plants to you. Datta. These are some medicinal plants. These are, sir, I know there is a less time, so I don't want to take your time. So I will explain in the short period. Some medicinal and aromatic plants are the Adathola vesica. Much more plants of this area, they are usable on the medicines. So I take few plants, Adathola vesica, which is comes under the family useful in the cough skin diseases. Likewise, Cassia fistula. It is also from the Scissalpinoideae family, which is useful in piles, digestion problems. This Cassia fistula is used in the digestion problem in the human being, as well as this plant used by the beer also when they have caused some digestion problems. Likewise, the Vidania somnifera, which comes from the family Solanaceae, it is useful in the joint pain. It is also energetic because the men who have some sexual problem, they are use the powder of these roots on their problem. Then Agel marvelous, which comes from the Rutaceae, it is useful in the stomach pain and dysentery also. Terminalia Arjuna and Terminalia Bellerica. These both plants come from the Combretaceae and useful in the heart problem, as well as it is one of the ingredients in the Triphala Churna. Plumbago geolanica. It is from the Plumbaginaceae family, which is useful in the stomach problem and skin diseases. Next is the Symbopogon martini, which is from the Poaceae family. The oil of this plant or oil leaves is useful in the paralysis, as well as it is useful in the cosmetic industry, soap industry, perfumery industries, and also for the massage. The oil is useful. Then the Scissors quadrangularis, which comes from the family Vitaceae, it is useful in the joint pains. Then Erythrina variegata comes from the Papilionoideae. It is useful in the eye troubles, and when there is a toothache, the people of this area uses the leaves of this plant. Then the Ocimum canum, which is from the family Lamiaceae. It is useful in the cough, cold, and asthma. Vidania tomentosa from the Apocynaceae family, useful in the skin diseases and stomach problems. Vitex nigundo comes from the Verbinaceae. They are useful in the arthritis and joint pain also. Solanum album, which is from the Santalaceae family. It is the aromatic plant and useful on the skin diseases, cosmetic industries, and perfumeries. Andropogon mauriculus, which is from the Ranunculaceae family, useful in the perfumery industries. Clematis triloba, generally known as the Ranchafa. It is from the family Ranunculaceae and used because, as the name suggests, that it is useful in the perfumery industry. Ruta graveolens from the Rutaceae, useful in temperature, worms, and cough and cold. Mellitus philippinensis, which is from the Euphorbiaceae family. Generally, these people called as the kunku tree. The seeds of these tree useful in the stomach pain, as well as these people uses these seeds as a natural color also or natural dye. Bixa oleracea, which is from the family Bixaceae, and it is useful for making the natural colors, because all these peoples dependent for whole life on the Melgard forest and uses this in the large number. Thank you, sir.
Okay, thank you, madam. The next abstract is abstract number 103. Okay, next 104. Synthesis of biological active compounds. Deepak R. Tope. Okay. Next abstract is 105. Recent dates in English literature. Yevita Ajay Patil. Is there anyone from this? Okay. Abstract number 106. Internet of Things, Shivani Bajaj. Okay. Next, abstract number 107. Rahul Gaike. Is there anyone? Rahul Gaike and K.N. Gaikwad. Okay. Abstract number 108.
Rahul Gaike is present, sir.
Sir, you can share your screen and start your presentation, sir.
I will inform you, sir.
You are not going to present, sir?
He is my friend, sir.
Okay.
Okay, sir. Shall I present? 108.
108.
Professor Indy.
108 Shedke.
Yeah.
Yes, sir. Yes, sir. You can present, sir.
Okay, sir.
Share your presentation, sir.
Okay, thank you. Thank you, sir. Is my screen visible?
Yes, sir. It is visible, sir.
Okay, okay. Sir.
Screen is visible and you are audible, sir. You start, sir.
Okay. So, respected Dr. Ranjit Bansod, sir, and my colleagues, myself, Professor Yendi Shedke, working as an assistant professor at Government of Maharashtra Ismail Yusuf College, Mumbai, and a PhD student of Ramakrishna Gore College, Akurdi, Pune. Myself, going to present on a topic, ethnomedicinal herbaceous plants, diversity of Surgana and Kalvan Tahsil of Nasik district, Maharashtra. So, here I am going to deal with these topics. Actually, my core topic is herbaceous diversity. But as I was working on the herbaceous diversity, simultaneously I found some certain economical importance as well as the medicinal importance. So I have chosen this topic. So introduction of this, that is, Surgana is a tribal and the remote tehsil of Nasik district. In this tehsil, Konkana, then Madhavpuri, Warli are the major population tribes of this talukas, that is Surgana and Kalvan. Then the vegetation in this area is most diversely spread, and it. Then the important thing why I have chosen this topic, because in case of this tehsils, that is Surgana and Kalvan, the unawareness of the modern medicinal facility, poverty, then the superstitions and the less availability of vehicle. Nowadays also, the tribal peoples are used the traditional knowledge and they utilize this traditional herbal medicine. Okay, so as they use these medicines, it's our duty as a botanicians that we should have to conserve this knowledge. So the indigenous knowledge is transferring from one generation to the next generation, and nowadays as the old generation are going to vanish, this knowledge is also going to vanish. So we have to conserve this. So here we have the review of literature. I have done this all study from 2015 to 2019. Previously this area, that is the Nashik district, is covered under the taxonomical works by the numbers of workers, that is the tea cook. Already we know all the data about tea cook. Okay, then recently till 2016, Mr. Patil has worked on this area. Phytosociological sociological studies is done by Dr. Jadhav and Dr. Parag. Then the wild edible plants are studied by Gavit, Wable and Thurdesers in 2017. As the important work is done by P. Lakshmi Narasimhan sir by BSI, he reported that about 61.2% of vegetation of this district is reported as a herbaceous plant, and overall vegetation, from the overall vegetation, about 5.14% of the plants are reported as a medicinal plants. So I have taken this topic. So purpose of this topic or selection of topic, that is to restore the indigenous knowledge, to maintain the record of the medicinally important herbs, then to find out the impact of human interference on these vegetations, then to find out the impact of the changing environment. Okay, these are the certain purposes of my topic. The study area of the my research, the Nashik district, which is distributed or located in longitude about 19.33 degree to 20 degree 50, longitude about 73 degrees, then 6 to 74 degrees. Okay, and it having overall area about 15,582 square kilometers. From this overall this Nashik, we have the area, that is I have selected the area of Surgana and Kalwan. That is Surgana has the geographical area about 4,314 square kilometers. From this, about 6,614 square kilometers is acquired by forest area, and it having the range about average rainfall is about 1808 millimeters. That is the highest rainfall in that Nashik district. Why? The nearest district which is present here, that Kalwan Tehsil, in this Kalwan Tehsil, it having the area about 1,195 square kilometers. From that area, about 393 square kilometers is covered by this forest. And here we have the rainfall about 625 millimeters. That is here, these both areas, that is the Nashik and Kalwan, which are located at the northwest of this Nashik district. At this region, at west of this, the Surgana and Kalwan, at both of this, we have here the Gujarat area or the boundary of the Gujarat area. While here the both tehsils are covered by Pet, Dindori, Chanwan, Devla, and the Bagla. So materials and methods for this, I have done this area. I have done this work from 2015 to 2019. Here I have made visit repeatedly to that area and collect the information from that area for the herbaceous diversity. Simultaneously, I have visited the numbers of persons like the Vaidyas, Katims, then price, local practitioners, and the farmers, who gave me the important information regarding the ethnomedicinal importance of these plants. I have also prepared questionnaires, and through that questionnaires I have visited to different area and different peoples and collected the information. Okay, so the herbaceous plants which are present here, the medicinal use I have collected here. In this I have noted my observation about 22. This one is the first slide for the herbaceous plant. Here I have reported the name of the plant, then common name, family, plant particles, then the order or disorder on which it applied. Then here we have the second slide, third slide. Actually, these are the actual observations. In this case, we have the families. About 21 families are reported as a medicinal. From this, Asteraceae is the most important family. After that, we have the Amaranthaceae and the Solanaceae family, which is medicinally more important. And then we have here we have the Asteraceae. Here we have. Then we have the next family, that is the Labiataceae and the Biaceae. These are the certain families which are reported as the most important family. The plant particles here In case of this, about 42% of the plant part which is utilized here is the leaves, which is the most abundant, and here the inflorescence is utilized very less. Here we have the disorder which is covered here about. In case of this, we have these are the certain disorder, about 26 disorders which are covered here. The plant utilized on the disorders are the fever and the malaria. This one is the important disease on which the most of the plants are utilized. Then also they are utilized in the skin disease problems. Then they are utilized mostly in the piles and features. Then we have the stomach problem. These are the certain importance problem on which the numbers of plants are utilized. Now the important part of this presentation, that is the result and discussion. Okay. So in this present study, about 42 herbaceous plants are reported, from that about 37 genera and 42 species are reported, which are from about 21 families. The Polypodaceae is the only family belongs to Pteridophyte family. Communaceae, Poaceae, Liliaceae are the families of the monocotyledons. Then the family Asteraceae, Amaranthaceae, and the Solanaceae are used more frequently and abundantly as compared to the other family. The plants are used here about 26 different disorder. The important thing, that is the herbaceous diversity, is specifically used for curing the skin disease, fever, mouth, and the stomach disease. Okay, these are the certain important results I have found here. The conclusion: the indigenous knowledge is getting vanished with the old generation. It is need to restore the indigenous knowledge before completely lost. As the generation are going to lose, that is old generation are going to lose simultaneously with that old generation, the knowledge is also getting to lose. So we have to conserve this. The herbaceous flora should be conserved before destruction due to the human interference and the environmental problems. Then the study opens a new door for the further phytochemical study of the herbaceous plant. Here I have noted numbers of herbaceous plants which having the economical importance, also then they are having the certain ecological importance. So I think that also on the medicinal plant, the phytochemical study should be done on that. We can done this. These are the certain references which I have used for the paper. And lastly, thanks to all of you. This one is the actual new generation, or we can say that the upcoming generation of the indigenous people, or we can say that the tribal people. As they have lost of hopes from us, we also should have to work for this. Thanks to all of you for listening.
Thank you, Sir. Thank you very much.
Good afternoon, sir. 107 is here.
Yeah. Rahul Gaike.
Yes, Gaike.
Sir, you start your presentation, sir.
Yes.
Shall I move, sir?
Yes, sir.
Okay, thank you.
So stop your sharing, sir.
Good afternoon, all of you. Myself, Rahul Chandrakant Gaike, PG student, and my research center is KRT Arts, Commerce, Science College, Nasik, under guidance of Professor Dr. K.M. Gaikwad, sir. And my oral presentation topic is study on nitrogen fixing ability of blue green algae from Tambakeshwar Tehsil. So, moving to introduction. Cyanobacteria are the prokaryotic photosynthetic microorganisms among which are able to fix atmospheric nitrogens. Nitrogen is one of the major limiting nutrient in plants, and cyanobacteria with nitrogen fixing ability were used as a biofertilizers. It plays major role to build up soil fertility under the existing socio-economic conditions of the farmers of Trambakeshwar region. The use of biofertilizers shall partially fulfill the nitrogen demands of crop. The study was carried out to know the nitrogen fixing ability of some selected cyanobacterial species. So this is a methodology where is used by me. So the collection of soil and water samples from the paddy field of some villages from study area. Identifications of the species was done by measuring the cell diameter with the help of ocular micrometry. So isolated cyanobacteria are grown on BG11, that is a blue-green algae 11 culture media. Geldhal method is used to determine the amount of nitrogen from the cyanobacteria. That for that digestion, distillation, and titration of sample was done. And there is a standard formula to calculate the nitrogen content. Okay, so this is formula is used. And these are the results of from where the 4 species were identified from the selected samples such as the Calothrix, Nostoc, Annapana, and the Oscillatoria. And the identified species are grown on the BG11. You can see the picture on the right side. So these are the picture where the algae are grown on the BG11 broth. And by using the standard formula, the nitrogen is calculated in milligram per liter, and the data indicates that the Annapana species efficiently fix atmospheric nitrogen than the Nostoc, Oscillatoria, and the Calothrix species. So this line graph was easily indicate that the Annapana has high nitrogen fixing ability than the other species. And the conclusion is that the cyanobacteria with nitrogen fixation ability were used as a biofertilizer. Present study was conducted with the aim to know the nitrogen fixing ability of the different cyanobacteria. From the village of Trambakeshwar Tehsil, some different species were identified: Calothrix, Nostoc, Annapana, and Oscillatoria. From this study, it observed that the Annapana species can efficiently fix the atmospheric nitrogen in soil than the Nostoc, Oscillatoria, and Calothrix. So the present work indicates that the such nitrogen fixing cyanobacteria species are the alternative to the harmful chemical fertilizers. So, and this is the references which are you referred by me for this work. And thank you to all the organizing organizers to give such opportunity for us. Thank you.
Thank you, Vaike sir. Thank you very much. The next abstract is 109.
Yes, sir. Yes, sir. Good afternoon, sir.
Ma'am, you can share your screen now.
Yes, sir. Sir, is it visible?
Not yet, ma'am. Yeah, ma'am.
Okay, sir.
Ma'am, you just
to every...
screen sharing, but you select the PPT. Yeah, ma'am. Yeah. It is visible, ma'am.
Okay, thank you. Okay. Good afternoon to everyone. I am Dr. Bharti Mahajan, Assistant Professor, Department of Botany, SBM Government PG College, Barwani. Today I am presenting the topic anti-diabetic effect of probiotic lactic acid bacteria in high fructose fat type 2 diabetic rats. These are the contents. Type 2 diabetes mellitus is a prevalent metabolic disorder which is emerging as an epidemic and generally affects the adult population. It is a global public health problem that affects and threatens the economics of all nations, predominantly developing countries. According to the recent report of WHO, 382 million cases were found with diabetes in 2013, worldwide, which will rise to about 592 million by 2035. In India, the prevalence of type 2 diabetes in some rural and urban areas was found to be maximum 13%. This is revealed by ICMR supported research. What are probiotics? That can be an alternative supportive theory for type 2 diabetes mellitus. They are defined as live microorganism when administered in adequate amount exerts the several health benefits to the host. Most of the probiotics belong to the lactic acid bacteria and bifidobacterium species. They are generally recognized as safe for human consumption with various probiotic properties. They are commonly associated with dairy food products and involved in the prevention and treatment of various metabolic disorders. The aim and objective. The aim of this study to assess the anti-diabetic effect of probiotic lactic acid bacteria in high fructose type 2 diabetic rats. This is the material methods to check the anti-diabetic activity. I have used 2 bacterial culture. First is Lactobacillus plantarum LPL-D6 strain, Lactobacillus fermentum CM1H strain. These are the methods I have been used in my study. Oral glucose test. It was performed and the physiological parameter was fasting the glucose level, estimation of glycosylated hemoglobin that was measured by using urodiagnosis system kit, and then estimated the insulin. It is determined by insulin rate ELISA kit, and the estimation of profile was calculated according to the Friedwald formula. The statistical analysis were analyzed by one-way ANOVA followed by one-flank test using sigma 32. This is the result and discussion. Here in this graph, you can see that after the 4 weeks of experimental period, both the groups LPFG and LFG have the better control the glucose as compared to diabetic control group and normal control group. Then here you can see that the fasting blood glucose at 8th week of experimental period, high fructose fed diet induced that the diabetic with the highest blood glucose level as compared to other metformin drug treated group or lactobacillus plantain treated group and lactose fermentated group. These two groups were controlled very well. Then after 8 weeks of experiment, we have checked the glycosylated hemoglobin that was also significantly lower in the lactobacillus treated group. Then the plasma insulin also were betterly, it is lower in the lactobacillus plantain treated group and lactose fermented treated group as compared to diabetic control group. Then the total cholesterol was checked at the 8th week. The highest cholesterol was found in the diabetic control group as compared to lactobacillus plantarum and lactobacillus fermented treated group. So the conclusion based on this study is it indicates that lactobacillus plantarum, LPL-D6 and lactobacillus fermented CM1 are potential probiotic strain which delayed the onset of hyperglycemia, dyslipidemia, and oxidative stress condition through immunomodulation and valuable for the management of type 2 diabetes and complication. Furthermore, investigation are required in other diabetic models of probiotic lactic acid bacteria supplementation in the prevention and progression of type 2 diabetes mellitus. These are the references. These are few publications. I am highly acknowledged to the Department of Microbiology, Barkatullah University, Bhopal and SBN Government PG College Barwani to carry out this research work. Thank you.
Okay, thank you, madam. Thank you very much.
Thank you, sir.
The next abstract is abstract number 110.
Yes, sir.
Arti Damle.
Yes, sir. Yes, sir. Am I audible?
You share your screen.
Yes, sir. Sir, is my screen visible?
Yeah, madam. Yeah.
Okay.
Your screen is visible and you are audible. You start, madam.
Okay. Thank you, sir. Okay. So, good evening to all. Today, I'm going to present topic preliminary study on insect diversity of Bhanduk Pumping Station, Mumbai, Maharashtra. So, to introduce about the topic, basically insects are most versatile and adaptive creatures. As I said, they are adaptive, so they are omnipresent, which means they are found almost in all kinds of habitat, and they help to maintain the proper functioning and stability of different ecosystems. In spite of such huge diversity, urbanization and anthropogenic activities affect the diversity of insects in various ecosystems directly or indirectly. Now, I'll be telling you something about Bhanduk Pumping Station, okay, which is situated in the Mumbai, Maharashtra. Basically, it is a sewage treatment plant where the sewage water is treated and reused. Actually, it's not potable, but we can use it for different uses like washing, gardening, etc. Now, BPS is home to several species of birds, insects, reptiles. Regular studies and surveys of birds have led to recognition of this area as vulnerable, and hence it is converted into sanctuary nowadays for better protection. A lot of avifaunal biodiversity of BPS has been studied, but no preliminary data on insect diversity of Bhanduk Pumping Station has been studied yet. So therefore, the present research work was carried out to study primary data of insect diversity in BPS. Now, I would like to tell you about the methodology. Before that, I will explain you something about the study area. Now, as you can see, this is the Bhanduk pumping area. Okay, you can see many buildings on both the sides. There is a national highway also. And this BPS is actually a heterogeneous habitat because it shows various kinds of wetlands, like there's a pond, there are salt pans, there is a creek, and mudflats. So this diverse habitat of BPS offers a suitable environment for rich flora and fauna. Now moving towards the methodology part, the study period was from July 2018 to November 2019. The field visits were conducted twice a week. We use sampling methods like line transact, random sampling, search sampling methods. The photographic documentation was done with the help of camera, Canon camera and mobile camera. Photographs were taken from several angles to acquire morphological details essential for identification. And for identification, we used different field guides, keys, and already published data. The identification was done up to family level. Okay, now I would like to share the results and discussion with all of you. So during the research period, we the number of insects were around 129. Belonging to 9 different orders and 58 families. As you can see in the pie chart, the Lepidoptera, which includes all the butterflies and moths, it is the dominant one with around 13 families and 34 species. Okay, and the least one is Blattodea. That is only one insect was recorded in this particular order, that is Blattodea. Now, I'll present the same data with a different bar diagram. So in this bar diagram, you can see there are all 9 orders mentioned, and you can see the families also and respective number of species count also. So here also it is clearly visible that the Lepidoptera is dominant with 13 families and 34 insects, followed by Diptera, Hymenoptera, and Hymenoptera. So these 3 having 10 families. In Hymenoptera, 17 insects were found. And Blattodea, as you can see, only 1 insect and only 1 family was recorded. Now, I have included a table here just to make you understand about the orders and families and the plate. Okay, so we have prepared altogether 12 plates. I am showing only 2 just due to time constraint. Here you can see all the insects are mentioned, and the order and families are also mentioned here. So the first one is cockroach. Similarly, we have also included all the photos of butterflies belonging to order Lepidoptera. So conclusion of this research work, the present study reveals family level insect diversity of BPS for the very first time. The survey has shown that BPS is rich in insect diversity. The heterogeneity of habitat is the main reason for the species richness. But now the measured threats for habitat destructions like high siltation, encroachment, overexploitation of resources, discharge of effluents. So therefore, the present study indicates that the conservation of natural habitat of BPS is of utmost importance for the sustainable growth and diversity of insects. Thank you. These are a few selected references.
Thank you, Aarti ma'am. Thank you.
Thank you so much, sir.
The next presenter is abstract number 111. Trees
Yes,
of
sir.
Ratnagiri. Trees of Ratnagiri. Sharad Apte, sir.
Yes, sir.
Is there anyone from this group? Trees of Ratnagiri.
I think Prashansa was there.
Prashansa, okay.
Prashansa, are you there?
Hello.
Yes, sir.
Sahil, yeah, you can be.
For
Okay,
a
Sahil.
second, sir, we are just sharing the screen.
Okay, okay. Yeah, ma'am. Screen is visible, ma'am.
Yes, sir. Even are we audible?
Yeah, ma'am. Yeah, continue.
So good afternoon, good evening, one and also, sir, we will be discussing about the trees of Ratnagiri, which we had an overview and conducted a study. So why we particularly have conducted studies on these trees, because the increasing urbanization and civilization has led to loss of many flora and fauna, as well as the diversity found in Ratnagiri, as well as on the various villages in Ratnagiri. So here Ratnagiri is one of the coastal district of Maharashtra state, which is situated at 16.30 degree to 18.4 north and 73.2 degree to 73.53 east, and surrounded by Kolhapur, Sangli and Satara district. So if we observe about Ratnagiri is a city which has been blessed with animals as well as the plant diversity, which we basically say flora and fauna. Ratnagiri is one of the rich village or district in case of such things. So Ratnagiri has a wide coast on western side and is surrounded by Satara, Kolhapur, Raigad, Sindhudurga on eastern, northern and southern respectively. Many medicinal, ecological as well as economically plants grow in this region. So the study area which we we visited random places in Ratnagiri and then we selected few of those plants. So these are the study area. The location of Ratnagiri district is 16.30 to 18.4 latitude and 73.2 to 75.53 east latitude. It is divided into 3 major zones and covers about 201 villages. West to it is urban sea and is surrounded by Kolhapur, Sangli and Satara. So methodology, we basically visited to different locations. We studied those plant, observation of the tree species, identification of the species using flora, collection of data, and final is the preparation of result. So the first plant which we visited was Gorak Chimsa, which is also known as Adansonia digitata or African Babua tree. This is one of the most important ethnobotanical plant. It is also known as Gorakshana. It also has traditional importance, that is Gorakshnath sage from Navta gave his valuable knowledge to his followers under this tree. These seeds are medicinal as well as large amount of vitamin C is found in this fruit pulp. Also, it helps in relieving constipation, effectiveness in leukorrhea and treating various diseases. So second one is the word which is also called as Vatavruksha. As we all know that word has a very significant importance in the Hindu rituals as it is worshipped in Hindu religion during the Vat Purnima. So basically this tree is used in treatment of white discharge in females. It also increases sperm motility, pile treatments, diabetic treatment and strengths are teeth and gums. So the next tree is pimple, which is also called as Ficus religosa, as well as it also has an importance in Hindu culture as well as in the Buddhist region, as it is said that Lord Buddha, he acquired his sage power sitting under this tree. So this tree is also known as paradise of God as well as mythological importance in Hindu and Buddhist region. It is grown as an avenue. It is used in diabetics, remedy on toothaches, treat various and human diseases. So next is the Saptaparni, which is scientifically called as Allenstonia sclerosis. So its cultural importance is what we got information from the people in that area is it is also known as devil's tree, as well as there are many myths about this tree. It uses our latex provides good quality for chewing gums, essential oils, gunpowder, malarial fever, and increases milk in lactating mother. So the last plant we will be discussing is about the surmad, which is also called as Caryota urens, which also known as fishtail palm. Basically, sir, this tree has a very significant importance in mythology of Ratnagiri, as basically during Holi, this plant is used on a very large scale. It uses are making of baskets, beer drinking, making to cure seminal weakness and urinary disorder, to treat gastric ulcers and snake bite, as well as it also shows anti-diabetic property. So why we choose this plants? Because these plants are now at high risk as we found these plants grown on the highway area. So many a times it happens that such old plants are destroyed due to such activities. As like I said, that increasing urbanization and civilization has led to many disturbance in the flora and fauna of our district, as well as sir, there are many other ecosystem dependence on this plant, whether it might be about the nitrogen, azola, or as well as there are many birds, animals which are been dependent on this trees. So sir, and basically they show medicinal important as well as they have religious important, and every plant they are very beneficial for we human beings. So it is very necessary to preserve and conserve these trees. So, sir, we have chosen this, and basically we will conclude that conservation of tree is necessary for us so that they will be available for us in future. Thank you, sir.
Okay, thank you, madam. Thank you for your nice presentation. The next presenter is 112, Miyawaki Forestation and Overall Development, Prashant Kanade.
Yes, sir.
Yes, sir. Yes, sir.
Yeah.
Sir, you share your presentation, sir.
Yes, is my screen visible, sir?
Yes, sir. Yes, sir.
Okay, I will start my presentation.
Yes, sir. Yes.
Okay, so I am not a environmentalist or I am not expert in environmental, but I have inclination towards the environment. That's why I am here to present my topic. Miyawaki forest is now largely adopted forestation technique because Miyawaki forests are grown within 2 to 3 years and with minimal cost. The cost is below 1 lakh rupees. That is the concept of Miyawaki forest. And you can create a small forest and then you can create cluster of forest. So Miyawaki forest technique is globally accepted, and states like Uttar Pradesh, Maharashtra, Kerala have adopted this scheme and they have released orders to develop Miyawaki forest in their particular region. So Miyawaki forest is definitely a backbone for maintaining the environmental sustainability. So why this topic was selected? Because environmental balance is need of the time, and environmental sustainability is now included in the fundamental rights. It is now taken for it is a human right that humans should get a clean air, clean atmosphere, clean water, and all natural resources. So there are some concepts of reforestation, afforestation, and forestation. So reforestation is nothing but the forest was previously existing, but it is destroyed due to some reason. Afforestation means you are creating a forest where there was no forest at all. And forestation is a normal foresting technique. Now, we know that cutting down the carbon content and then noise pollution, there was a nice paper previously discussed in this conference that noise pollution is also a major requirement and how trees can help in that. So my suggestion is that everyone should plant at least 10 trees every year. Do not plant only one tree and grow it. Plant 10 trees every year. So when you become at the age of 50, you will have 500 trees in your credit. So, and there are various new techniques that should be adopted because conventional technique may take a longer duration. And my another observation is this technique can also be helpful in commercial farming. Means you are conserving the environment and you are also generating some income, and that will be that is another part of organic farming and kind of thing. So can Miyawaki forest technique will be helpful in commercial farming or generating the economy for the youth or the farmers in that particular region. Now, before that, I studied various types of forest that we have in our country. So there are various types of forest like tropical evergreen forest. We normally call it as Sadaharit Vane. Then tropical deciduous forest means they are green for particular duration. Then there are temperate evergreen forest, temperate deciduous forest, Mediterranean vegetation forest, and coniferous forest. We will quickly have a look of what do you mean by tropical evergreen forest. These are the forests normally found in India and in the states of Andaman Nicobar Islands, Western Ghats of Maharashtra, then which are near to Arabian Sea, then peninsula. It is normally in the south, west and southeast region of India, and also in the northeast region of India. Then tropical deciduous forests are available in Madhya Pradesh, Uttar Pradesh, Bihar, Jharkhand, Chhattisgarh, and some parts of Maharashtra. Then there are temperate evergreen forests. These are also available in Western Ghats. Some part of Western Ghats are evergreen forest, and some part of Western Ghats are temperate evergreen forest. Then deciduous forest, these are also found in various plateaus of India. Then Mediterranean forest, Mediterranean vegetation forest, or what we normally call as Kurti Jungla. And there are also alpine forest. They are having around 3,000 meters of height, and they are excellent. And these are normally found in Mysore region or in Himalaya. or in the hilly regions of Tamil Nadu, Karnataka, and Maharashtra. So these are the types of forest. But if we decide, okay, we will have a evergreen forest and we will have plantation, so we need to have a cautious observation, because what happens if people are starting plantation, some another fine day, all the real estate agencies or real estate companies will purchase that particular land and they will destroy the forest. So that should not happen. So whatever land we have available, should we have green cover, we should have protection of soil, we should, then that will also protect the environment. So for this thing, the Miyawaki forest may be the excellent solution. So why we need, why we should grow the forest? No need to explain. Oxygen supply, then restoring the ecosystem, tree support or creating the habitat for birds and creatures. Then we can also get some supplements like medicines, fruits, vegetables. We also can have water management, and we can protect the heat islands. So these are the things, and we also are saving the energy and carbon sequestration. So now, what is the method? So in this method, actually, it is something called as planting the trees near to each other. Hardly, we are planting 2 to 3 trees per square meter. So if you have 1 square meter land, you can plant 2 to 3 or 2 to 4 trees. So that will definitely help the environment. And why these trees are growing faster? Because there is a competition among the trees. They fight for oxygen, they fight for sunlight, and they fight for all the resources. And apart from that, they are also creating habitat for the various species. So he is the person who is the pioneer of Miyawaki method. He is Dr. Akira Miyawaki. He is from Japan, and his successful stories are there, and that is the reason this method is adopted for planting the trees or creating the dense forest. It is also called as urban forest, or they are also called as seedling forest. So, and another suggestion given by Dr. Akira Miyawaki is that you have to, no need to strive for any special trees. You plant the trees which are locally available. Means you can plant. Yes, sir.
Yeah, sorry to interrupt you. Please conclude within a minute.
Yes.
Yeah.
So this is the method, and my major observation is we should think beyond the forestation. So some species are endangered, we can create a natural habitat from them, and feasible environmental conditions to create the environmental conditions that are disturbed. It is small contribution to provide the species near natural conditions. And what farmers can do? Farmers can grow medicinal plants. We can motivate farmers to grow medicinal plants. We can also motivate farmers to keep beehive boxes so that there will be honeybee production. Then it will also motivate bees through beehive boxes. Then we can create artificial lakes and we can have fish farming there, and we can create a habitat for endangered species. Another thing is agrotourism, and vegetable and organic farming is another outcome, and Miyawaki method will be definitely helpful. So poor farmer who is having very small land can have combination of plants. We can plant like hibiscus, marigold, stomp, that is we call it as body soap. Then we can have mint farming, we can have adulsa, which are medicinal plants. Then some spices also can be planted, and some big trees like neem, jackfruit, bamboo, and vrikshabandi can also be, and that will definitely benefit a lot for conservation of the nature. So we can have a creative ecology with the help of Miyawaki method, and we also should actively participate in supporting our environment and creating the benefit for the everybody. Okay, so thank you everyone, and thank you all the organizers and all the committee members of this conference, and providing me opportunity to share my views related to environmental sustainability. Thank you.
Okay, thank you, sir. Thank you, sir.
Yes, sir.
It's a kind suggestion to all the participants that you should stick the 5-minute time. Don't take the others time. The next presenter is 130. Virendra Srivastava. Is there anyone from this? Formulation and evaluation. No one is there? Okay. The next is 114. So I have the video of 114. So I'm going to share.
Dynasty during 13th to 19th century. You know, water is the most essential part of human life, and that's why water is the need of the human. Water is available to human beings from different sources, which comprise of lake, ponds, sand, rivers, wells, etc. And that's need of careful management of water and water resources is an integral part of water conservation. So the water management practice adopted by Gond rulers in 13th to 19th century in the Chandrapur district of Maharashtra state. And that Gond rulers, when started the improvement of that water management system, they decided, especially drinking water purpose for human being and horses, interlinking of lakes. In addition to his big dug well were also constructed throughout the empire. Thus, it can be argued that Gond rulers had set an example of water system which is still relevant in today's situation. Water, especially that's why people use too much quantity of water tanker, and that's why today's need. I just explained that what was the system of water management in the Gond dynasty. Especially Gond rulers ruled in Chandrapur district in 12th to 17th century. 500 years ago, they had contributed significantly towards water system in the district. Water system was not restricted only for drinking purpose, but extended towards construction and conservation of lakes, irrigation, creations of separate lakes for horses and state big wells. And after that, the system also very innovative, like Gond period, that's the Gond dynasty rulers, innovative system was adopted, and that system was called Tukum system. And this Tukum system started by Kohli community, and that Kohli community belonged to UP.
Good afternoon, one and all. Respected chairman of oral presentation session and all the dignitaries. I, Mr. Praveen Nilika, from Chisquare Department of Water.
The next presenter is 115. I think he had shared a video. Okay.
Good afternoon, one and all. Respected chairman of oral presentation session and all the dignitaries. I present a research paper, some gift of the lecture that provides all the basic requirements for and line functions which serve as the useful tool for calculation of lectures. The present work explain how plant powers are used by the people of the area for to worship various gods, goddesses, customs, and traditions related. For the present investigations of a survey of the religious and medicinal plants of Davhadi region of Nasik was carried out during the October 2020 to March 2020 to find out important plants in the religion, culture, and healthcare system of the local. Information regarding the uses of plants in this Torah and standard. For the present investigation, the area chosen was Davhadi region of Nasik district And fourth question we have selected Accidental system. The leaves are used to of the base of the plant in the front of the plants and roots. The real part of the plant is given as food for increasing milk gathers. Every coating of and it is having major synthesizer property. Oscillum science. It is also used in the Havan oil often from the test media for worships to various gods and goddesses. On conclusion, I would say that the present work explains how plant parts and sequence for their religious as well as medicinal purposes. The study on the sacred and religious plants used for various worship show their importance in human life. So the religious activities acting as a conserving tool for biodiversity, so it is necessary to promote this activity, aesthetic value to conserve the biodiversity and nature, which will surely play the role for the betterment of the human beings and the society and nature. So, thank you, sir. Thank you so much for giving time.
Okay, the next presenter is 116. Environmental.
Yes, sir.
Yeah.
Yeah. Am I audible, sir?
Yeah, madam. Yeah.
Yeah. Thank you, sir. I am going to share my screen. Is it visible, sir?
Yeah, madam. Yeah, it's visible and you are audible, ma'am.
Yeah. Thank you, sir. Good evening, everyone. Today my topic is environmental effects of waste to energy plant and energy generation from municipal solid waste in India. I am Dr. Gargi from CPCB Delhi, and I think that environmental sustainability is a need of the hour. So we should let's discuss about that increasing industrialization and urbanization and the changes of our lifestyles causes the increasing quantities of the waste, and now there is a necessary is the segregation of the waste is a major challenges. But now there are several types of waste generated in every sector, and we are here discussing about the municipal solid waste, that is MSW. So there are different technologies already developed and available in our system that to generate some energies in reducing these quantities of waste, and we can easily dispose of the remaining part that is not the too much hazardous for the environment. So we just the objective is to reduce the very available technology to reduce this type of huge amount of waste, because we all know that in our country there are many landfill lands are there which are like the big heaps. So the waste energy plants is the most beneficial way to recover the energy in the form of biogas, bio-CNG, electricity from the agriculture, industrial, and also the MSW. Now we see that what is the basic compositions of the MSW, that the 52% is the mostly organic matter, which comes from the domestic food waste, landscape tree trimmings, and another was the recyclable material is 23%, from where we can easily recycle the part, or we can use that section in another byproducts productions. And the remaining parts are the inert matter. Inert matter we can usually the CND waste, stones, other inorganic materials. So if we can segregate these three sections, and then we easily use it several ways. So basically organic matter is very much fruitful for the energy generation part, and also the recyclable part. I also say that there are several byproducts productions are there. So the present overview, the Indian scenario of the solid waste generation. So mostly that last 2020-2021 financial year, we can say that there are likely to be the 550 million tons per annum. This is the generation, and we can usually we interpret it as a TPD, that is ton per day. So the total, the overall generations is among the generations, there are processing at the 63%. Now we are processing 63% of the total generation, and the remaining which cannot do any byproducts or do any other processing purposes, then that is goes to a landfill, that is the 8%. Still, there are a gap of approximately 29 to 30% of the total waste and generation and collections. So we are in coming days, we should reduce that thing, that also benefit for ourselves, for our environmental sustenance. So now this is the representation that where as per our all over the states of the Indian scenario, the percentage of the treatment of the solid waste, municipal solid waste. So there are several states are there who are usually who are doing well in this sector. They are treated, most of them, solid waste and the cleaning the heap of the old municipal landfills. And some other states are also doing going to follow them and doing the same in near future. So here is the why waste to energy is so much important. I already briefed it. So we actually in our pattern of the lifestyle changing, we should go for this. The fuel, this renewable energy is our one of the necessary part. So we can say that this waste we can already use as a fuel part. So gas, fuel and electricity all are very much important for our necessary regular lifestyle. So if we can treat this waste and we can get some benefit, so then we should go for this as the basic sustainable development. Now this is the basic process flow chart. That is the how waste to energy is going on. I already showed that in municipal solid waste also there are the CND and inerts are the non-biodegradable, and remaining is the combustible part. So and all of the specifically the biodegradable and the combustible sections, we are going to get the biogas from the biodegradable part, and the electric generations from the combustible thermal through the thermal process, like gasification, incineration, and pyrolysis. Now there are some environmental issues are also there. Let's see what are the incomplete combustions of this large number of toxic compounds, which the waste, maybe the productions of the dioxin and furans, that is the most hazardous air pollutant. And inadequate flue gas treatment system may also cause the emissions of large volume of flue gas, which may sometimes cause the human health error that due to their organic and inorganic compound. Improper utilizations of the bottom fly ash may also cause the fine air particulate matter, and generation of leachate may cause the, if it is not properly disposed of, if it is not properly managed, so then this is causes the traces of water pollution, both surface water and groundwater, and sometimes it may also causes the soil pollution. So we should take care of these issues, and we should make in a better line. So now to manage everything, there are solid waste management rules where we can carve, we can try. There are three criteria which we can follow for this waste to energy process, for which this environmental issue we can minimize these environmental challenges. Firstly, non-recycling of 1500 kilocalories, so we should take care of that. Supply of waste should be stable. Incineration and chlorinated plastic should be avoided. Requires high temperature for the sufficient air and mixing gas stream. The furnace air temperature should be proper to minimize the prevention emissions of the dioxin and furans. Fly ash should be managed in proper way, and all of the yes hello
ma'am, sorry for interrupt, but come to the conclusion, ma'am.
Yeah, well, I have just only 3 slides more, so I can conclude. So we should go for that. Yeah, sure, sir. Emission standard we should follow. Now the simple case study that in Delhi, this Wakla and Timarpur waste energy plants there was, and it was established in 2007 and started as a operational facility 2012. Initially, there was a problem with with this particulate matter and the dioxin and furans, but gradually they are also minimizing the issues. So we can now this is the present scenario of our waste energy plants in India. So in most of the 12 states contains the 32 waste energy plants, and approximately 5,619 megawatt are generated annually in the India as a part of a renewable energy sources. Here are a few references, and thank you.
Okay,
Thank you, sir. Thank you, the organizing committee.
The next presenter is 170. Development of sonic rich Shatavari candy.
Sir, can you hear?
Yes, sir. Your screen is visible, sir, and you are audible. You start, sir.
Okay, sir. Sir, here I am presenting my research topic on the development of phenolic-rich Shatavari candy. So, I am Dr. Kiran Agujana, assistant professor working as a assistant professor in HF Devi College. These are the points covered in the seminar. And the thing is that that Shatavari, that is the scientific name is Asparagus racemosus. It's an important medicinal plant of Liliaceae family. In Ayurveda, this asparagus species is used to treat the more than 100 diseases. That's why it is called as a Shatavari or Shatavara in the name. The Shatavari root is referred as a queen of all the herbs. It's also a because of this importance that is called as a queen of herbs. These are the already existing work conducted on the Shatavari of the asparagus family. It's what we have taken this work as the asparagus racemosus is a species of common throughout India, but it's a very hard, bitter in a taste to direct consumption. What we have did, we have added the two, it's a minor fruit. It is having the highest phenolic content that we have blended and added to the to make the candy. So that ultimately we have added the micronutrient of the product. Yes, sir. Can you hear?
Please complete it within 5 minutes, sir.
Oh, sir, surely.
Sorry, but it is.
I will complete it. I will complete it.
Okay.
Because of that, we have added that minor fruit, and we have made it as a candy that directly anybody can consume it, this Shatavari, as a candy without appearing that bitter taste. That's one. These are the objective. It's improving the. These are the materials and methods what we have taken. It's in the first objective we have taken. That's in a 3 treatment we have taken. The how best the peeling we can achieve from the Shatavari we have done. From this, by that, it's in a total phenolic content which is having the best by that treatment. That treatment we have taken for the first objective. In that, it's in a first, it's in a 0.5% NaOH solution is given the best treatment to get the best quality phenolic content rich Shatavari candy. That treatment we have used to make the candies. That next, these are the gallic acid standard curve what we have developed. It's for the first peeling conclusion that T1 treatment has given the best result. These are the flow diagrams how we have prepared the Shatavari candy. This is the formulation what we have taken. It's in the 5 treatments we have taken with a different ratio with the cocoa syrup and the sugar syrup. These are the working flow process. These are the analysis we have done. These are the results. The moisture content, it's in the same with all the treatment. It's not at all changed. But when it comes to the color value, when we added the cocoa, the color is changed to a deep red color. Deep red color. That's what we can see here. It's an L value is increased. The total color chart. It's a mass transfer calculation of the candy also we have done. It's in a we can. It's in a reducing sugar standard curve. It's a sensory. It's an among sensory. It's an when it comes to the overall acceptability, the S3 treatment with a 50% concentration solution has given the best result. These are the conclusion for the first objective. The T1 treatment, that one is in a 0.5% NaOH solution treatment, best peeling of the Shatavari roots, and S3 sample having the highest overall acceptability. Then finally, the addition of the kokum adds a minor fruit is help to reduction of the bitterness of the shatavari candy. Thank you, sir.
Thank you, Kiran sir.
Sir, I have one more presentation.
Next paper.
Can you see this presentation, sir? It's a poster.
Yes, sir. Yes, sir.
It's this is a poster I wanted to present. That one is the ice content during freezing of a kulfi. It's here we have prepared the kulfi with a different concentration. But the thing is that it's very difficult to estimate the percentage of the ice formation in the ice cream. But the same concept we have taken, it's in a kulfi as we have considered it's without overrun ice cream. Here we have measured how much percent of the water is forming as ice, and that is the total work. Here it's in a total ice content water. The things is there. That's these all are methods, existing methods we have used to estimate the ice mass fraction with respect to temperature. Here we have taken that as we estimated that when the freezing point of the kulfi is started with minus 2.5. That's what we have estimated the ice mass fraction from, which is started from the minus 2.56 up to minus 30. We have estimated how much percentage of the water is converted into ice. We can see these all are the existing models, Rhodes models, Rotova model, and Tichova model. Among all the models, you can see here it's at the initial stage, the free water, whatever the free water is there, it is the percentage of the ice formation is highest at the initial stage. Then after the 80% of the ice formation, that is declined here. You can see here it's in almost all the curves, it is having the same values, because it's when it reaches to the bond water is never, it is forming an ice crystal. That's what you can see here. It's an only maximum 80% of the mass of the ice has formed as a ice crystals. From the total study, we can conclude that initial stage it is having the highest ice mass fraction. At finals, it's 100% of the free water we can't convert it into ice. It's remaining 10% is remaining 10 to 12% of the free water with bond water is not at all forming as an ice crystal. So thank you. This is my any questions?
No questions, sir.
Okay. Thank you.
Thank you very much, sir. The next presenter is 119. One pot synthesis. Jyoti Agashe. Jyoti ma'am, you are here? Okay. The next presenter is 120. Is there anyone from 120? Okay, the next presenter is 121.
Yes, sir, I am here.
Yeah, madam. Yeah. You start your
Shall I
screen share.
share my screen, sir?
Yeah, ma'am. You share your screen. Ma'am, screen is not visible, ma'am.
1 minute, sir.
Okay, ma'am. Okay, ma'am. Okay.
Is it visible, sir?
Not yet, ma'am.
Okay, sir. But I couldn't open, I think so. Shall I present my paper as such, sir, without PowerPoint?
Yeah, madam. Yeah, you can present.
Okay, sir. My presentation is evaluation of different seed priming agents, that is organic agents, on germination and seedling vigor of rice. Okay. So I am Dr. V. Sridevi. I am working as assistant professor at Pondicherry University, Karaikal. Okay. So my research is on rice. Rice is the most important staple food crops of world. It supplies calories for more than 45% of world population. So seed act as the basic unit for crop establishment. So rapid germination and emergence of seedling is the prime criteria for any establishment of any crop. So seed priming helps in rapid and uniform germination of any type of crops. So for inducing the rapid and uniform germination, seed priming helps. What is seed priming? Seed priming is a controlled hydration of seed to induce induce metabolic activity that induce germination, but it prevents the emergence of radicle. Okay, so in this keeping all those things in view, this study is undertaken to evaluate the different organic priming agents to improve the germination and the seedling vigor of rice. So for this, the experiment is conducted in the seed testing lab of our institute with 6 treatments in completely randomized design with 3 replications. The treatment... Am I audible, sir?
Yeah, ma'am. Yeah. Atul sir, please mute yourself.
Sir?
Yeah, ma'am. You continue, ma'am.
Okay, sir. So totally, we tried 7 treatments. First treatment is control, that is untreated seeds. There is no priming. Then we have 6 priming treatments. One is water. We prime the seeds just with the distilled water. Then 1% coconut water and 1% cow urine. Then moringa leaf extract at 3%. Then panchakavya at 3%. Arapu buttermilk solution at 3%. Okay, among the priming treatments we tried, cow urine excel in all the seed quality parameters such as germination percentage, seed speed of germination, then shoot length, root length, and seedling dry matter, then vigor index. One, that means vigor index mass and vigor index length. So all the priming treatments, that is 6 priming treatments, are out yielded than the control. So from the present investigation, we concluded that all the organic priming agents are used to improve the germination and the seed vigor, even by these small farmers, because they are cheap, they are easily available, so the farmer can afford all the organic priming agents we have tested, that is coconut water, then cow urine, panchakavya, then buttermilk solution, then moringa leaf extract. So the reason behind that is the growth regulators present, the growth regulators and nutrients, then trace element present in the cow urine is the main factor to increase or improve the germination and seed vigor. That's all. Thank you, sir. Thank you for your patience.
Okay.
I have throat infection, that's why I couldn't speak.
Okay, no problem. Okay. Next, absent number is 122.
Thank you, sir.
Okay. Effect of syrup concentration. Is there anyone?
Yeah, good afternoon, sir. Good evening, sir. This is Dr. Tepan here. Just give me a second.
Yes.
I'll just. Sorry, this is something else. Just I will. Am I audible?
Yes, sir. Audible and visible. But yes, you have to conclude, sir, within 5 minutes, sir.
Yeah, sure, sir. This is the Effect of syrup concentration and the drying methods for the quality of sapota community. All we know that sapota, different dry fruits are available in the market, but now sapota is the farmers are selling in the throwaway fresh. For that, we need to develop some shelf stable product. For that, very cheapest and easy method is to development of candy or osmotic dehydrated sapota. In this experiment, we conducted with this. What do you mean by osmosolar dehydration? Osmotic dehydration means we have to steep the sapota fruits or slices in concentration of 50. Through that, what will going to happen? Because of osmotic pressure, water from the fruit is coming out and some sugar from the syrup is getting into the fruit. That will going to give a very good body or texture to the product. After that, these are some of the information. We are using raw material like sapota and other things, and we are using different drying method like solar tunnel drying as well as ray drying. And the objective is to develop osmotic dehydrated sapota candy under different level of bricks and selected drying techniques to assess the overall acceptability of sapota candy through sensory evaluation. Yeah, this is a flowchart: selection, washing, slicing, preparation of syrup, steeping in different concentration of bricks, then keeping it for 18 hours in the room temperature. Then they have to take these slices from the syrup, they have to dry it and go for drying in two different methods and packaging and storage. This is the here we have a total 8 treatment. One is 50 degrees, 60, 70 with the solar treatment, and same with the tray dryer. And also we are controlled where exactly we are not using any kind of syrup, just drying the candy. This one slices. These are some observation we taken. That is a mass transfer kinetic, like some moisture loss, solid gain, and weight reduction. Yeah, with respect to the moisture and all these things, moisture and solid gain, the maximum loss of moisture as well as a weight reduction was observed in the treatment 6. They were exactly 70 degrees on the solar tunnel dryer, where exactly we are steeped these things in a highest concentration of syrup. Because of that reason, highest moisture loss as well as low at the same time the solid gain is optimum in that condition. And also it's exhibit the maximum the treatment T3, that is where exactly 70 degrees so this solar tunnel dryer has given a very good total solids and lowest moisture content. The same thing with respect to the moisture content is observed with the treatment. Wherever we use the maximum concentration of sugar syrup, we got a very good result with both solids as well as the weight reduction because of high osmotic pressure. Now we move to sensory properties. The treatment T3, where exactly solar tunnel dehydrated sapota candy with 70 degrees has given a very good, has got a very good sensory score up to 8, where exactly we use the 9-point Adani scale. This is a very good acceptable by the sensory panel. And totally, finally, we conclude that the fruits or slices we treated with the solar 70 degrees and dried at a solar tunnel dryer as gave a very good result with respect to all master circuit physical parameter as well as a sensory evaluation sensory properties. Yeah, thank you.
Okay, thank you, sir. Thank you very much. Thank you very much. The next presenter is 123. Swareli Naik.
Yes, sir. Yes, sir.
Yeah, ma'am, you start.
Sir, am I audible and visible?
Yeah, ma'am, you are audible and visible also.
Okay. My paper title is Sustainable Techniques for Selected Live Feed Culture on the Survival and Growth Rate of Draft Gurami. Okay, I am from Muljijetha College from Jalgaon. This image shows the status of India in global market. As we can see, in this global world, we export only 0.4%, and hence our rank is 31st in exporting countries. And there, Maharashtra's rank is 6th in India. Our object, availability of food for growth and survival, purity of food, food cost and maintenance is very low, high nutritional value and easy to acceptable by fish, providing live feed for better growth and survival rate. Classification of the species, we are using, I'm coming to directly genus, Trichogaster, and the species is tuna, commonly known as gurami. Materials using in this paper, commercial food which is locally available in market, and mixed diet food which is cyclopoids and Calodocerae. Methods, time and place of research is November to February 2021 to 2022, and the place of research is Jalgaon. Experimental design, this research was conducted using a completely randomized design trials with 2 treatments and 5 replication. Treatment A is commercial food as a control, and treatment B is mixed diet feed. These are formulations which is used for result and discussions. This is our results. You can see directly last 3 rows, the specific growth rate, survival rate, and mortality. You can see the mixed diet feed. It shows 100% survival rate of the fish. This investigation shows that the growth performance of mixed diet, including the protein, is superior to the activity control volume. Provides live food at time play a major role in achieving maximum growth and survival of the young ones of the fish. In terms of growth performance, the current experiment shows that the efficiency of the mixed diet integrated meals was greater than the controlled diet. Then the conclusion, the availability of on-growing live food would not only offer farmers and exporters a better alternative option for feeding to their fish, but more important to possibility of enhancing the fish performance and quality through gurami, ornamental fish culture. Thank you.
Okay, thank you, madam. Thank you very much. The next presenter is 124. Is there anyone from this? Dhanraj Balbin Bure. Okay, no problem. Next, 125. Sujit Chandar. Okay, no problem. The next one is 126. 127. Atul sir. Atul sir, can you hear me? Hello. Atul sir.
I am available. Yes, sir.
Yes, sir. Yes, sir. You can share your screen and start presentation.
Yes, sir. Thank you, sir.
Okay, sir.
Sir, is it visible to you?
Sir, your presentation is not visible, sir.
Yes, sir.
Sir, if you have any difficulty, you can talk.
Okay, sir. Thank you. Good afternoon, one and all. This is Atul Wak from Research Center in Botany from LUH College, Nasik. Today, I would like to present my paper in our International Conference on Environment, Development and Sustainability. The topic is utilization of some exotic plants in ethnobotany of Malegaon Tehsil from Nasik district, Maharashtra. As we know that the exotic plants means the plants which are non-native, foreign species, or we can say the introduced species by anthropogenic activities, or we can say with other mediums. These alien species, or we can say the non-indigenous species, they acclimatize in our region and start their reproduction. So where these exotic species, they are mostly showy in nature. They are purposefully cultivated, or we can say planted for ornamental purposes. So these exotic plants may show adverse effect or may be beneficial to the local system. And this local system, or the peoples are very clever in the plant utilization. So the tribal peoples or the peoples from the area, we can say the local peoples, they using these plants for food, fodder, medicine, and timber and other purposes. So when we started the survey field visits for this utilization of exotic plants, we gone through structured, semi-structured, and oral interviews. And the plants were identified by a few articles, monographs, or we can say the senior plant taxonomist. Because these species are not present in our floras. Now few much additions are there, but most of the plants are not in the floras. That's why senior plant taxonomist were for the help. So the plants were arranged alphabetically with their scientific names, local names, habit, and all these things. When we try to find out the result and discussion, that time we got 29 exotic plant species from 22 families. When we classify them in dicot and monocot, 18 are dicot and 4 were monocots. In this one, the dominant families, they were Amaranthaceae and Euphorbiaceae, in which the 15 plant species are herbs, 2 were shrubs, 10 plant species are trees, and 2 species were climbers. So in this exact survey, we find out that purposes as well as we can say in ethnobotanical purposes. Later on, root is there. The number of root uses of root is at the second place. Stem, flower, seeds, latex, bark, fruit, sap is there. So when we try to find out the exact findings of this plant, so we can say that each and every plant is medicinal. Each and every plant can be introduced in ethnobotany. Just we have to know the knowledge of the plants. Few plants were on trial and error basis, but the local people get result, and because of these results, they are successfully in the results, and now finally they are using these plants for various purposes. At the time of COVID situation, coronavirus is there. Few plants were taken on trial and error basis, but they got the results. So we can say the exotic plants, they are also be useful for local peoples, or we can say the local system. But the one thing is that Gliricidia is the plant that is exotic. This plant is showing adverse effect on the ecosystem. Nowadays, we can see in the summer season, they have only flowers, no leaves, nothing is there. No one living organism can depend on this plant because it has an allelopathic nature. So few are the examples which are adverse, which shows the adverse effects on the environment. So this is the finding. So I would like to conclude and thank you, sir, for the opportunity.
Okay, thank you, sir. Thank you very much. The next presenter is 130. Is there anyone from 130? Sandeep Shinde. Okay. The next presenter is 133. Effect of drying methods on quality.
Yes, sir. Yes, sir. Yes, sir. Good evening, sir. Good evening,
Yes,
sir.
sir. Yes, sir. Yes, sir. You will start sharing, sir.
Yeah, it is shared, sir. Are you visible? Is
Yes,
it
sir. Yes,
visible,
sir.
sir?
Yeah, it's visible, sir.
Okay, sir. Thank you, sir. Good evening, sir. Myself, Dr. Shithin Goda Edchi, working as an assistant professor of agri engineering at College of Horticulture Engineering and Food Technology, Devusur. It is a new college in Karnataka. My topic of this research paper is effects of drying methods on quality characteristics of chili powder. Basically, the chili, as we all know, chili is a fruit of plants belong to the Solanaceae family and genus of capsicum. In India, with respect to chili, it is cultivated in the area of 7.33 lakh hectares with a production of 17.64 lakh tons with a productivity of 2,400 kg per hectares. Most importantly, the Andhra Pradesh stands first rank with a 26% of production to the total production, followed by Karnataka, Orissa, and West Bengal and Maharashtra. Why we have selected this study? The justification is, so there were some speculations and concerns from the chili growing farmers that Drying of chili at higher temperature in different drying methods like tunnel dryer and tray dryers were affecting the quality of seeds, because the seed is a very important input resource for the agriculture as well as farmers. So that these two drying methods are affecting the seed quality of the chili as well as powder quality also. And in order to study the effect of different drying methods on powder as well as seed quality, so we have taken this study with these two specific objectives. To first, to know the effects of drying methods on quality of chili powder, and finally to know the effects of drying methods on quality of seed. So these are the materials and methodologies we have followed in this study. We have considered two varieties of chili. One is Kaddi and Dabbi. So these two varieties are extensive. Badgi chili is very famous in Karnataka, so it has got a GI tag, and so these two varieties have been considered in under this study, Kaddi and Dabbi varieties. So these are the lab requirements, and these are other requirements with respect to chemicals. So drying methods considered under the study were drying on polythene tarpaulin sheet, and drying on concrete cement floor, and drying in solar tunnel dryer, and drying in tray dryer. So this is the flow chart of what the protocol we have followed for this research work. So first we harvested the chili of around 2 to 5 kgs for each treatment, to conduct each treatment. Then we cleaned and sorted the odd ones. So then we weighed around 500 grams of chili for each treatment. So then we dried those selected 500 grams of chili samples under 4 drying methods, and then we separated for the purpose of carrying out the seed viability studies in the laboratory as well as field emergence. We separated the seeds from other lots of 4 treatments and stock. Then we ground the chili fruits. Finally, the safe storage moisture content. When the fruits reach this safe storage moisture content, we ground this one powder made into the powder. Then it was sieved for a fine powder. Then we sorted the powder, and we carry out the different quality assessment of the powder. So these were the different properties assessed under the quality characteristics of powder. So moisture analysis, we made moisture analysis, bulk density, tap density, flowability, cohesiveness, and color attributes, and dispensability, then wettability, oleoresin content, germination percentage, and field emergence. Both these two properties were analyzed with respect to seed viability. So this is the data with respect to the moisture analysis of chili fruit. You can see as compared to T1 and T2, T3, that is drying the fruits under solar tunnel dryer, reduced almost 3 times the time required for the drying. Means, so if the drying under open sun drying is taking 10 days, in tunnel dryer as well as tray dryer, it is taking 4 to 6 days. So we are reducing the drying time required for the drying of the fruits by following the so these advanced techniques like tray drying and the solar tunnel drying. So these are the value data required with respect to bulk density and tap density of the fruits. So we have recorded under tray drying as well as solar tunnel drying, higher tap density was observed for all the treatments and all the varieties. And with respect to the color values, with respect to oleoresin content, so received, we have observed, we have recorded the highest oleoresin content in treatment 1 and 2, that is open sun drying under polyethylene sheet, tarpaulin sheet as well as concrete surface. Because in case of tray drying as well as solar tunnel drying, so because of there we will not be having control over the temperature. So because of that, so there may be this oleoresin content is an highly volatile compound which might have evaporated. That's why these values are very less. So we have retained the oleoresin content in these two open sun drying treatments for both the varieties. So these are the color values with respect to chili powder, red chili powder. So the A star value is very important because the brick red color used to be preferred by the food processors. So that's why we have to consider the A star value. So we have received for treatment 1 and 2, we have received for both the varieties A star value, higher A star values. So these are the samples of chili powders prepared, dried and prepared under the different treatments of drying for both the varieties. So with respect to seed viability assessment under as influenced by the drying treatments for both the varieties. So you can see for T4 there is no germination, because means the drying of chili fruits under tray drying has resulted in loss of germination. So it has not resulted any germination, because that's why, because we cannot have control over the temperature. This is a high temperature drying, so viability has been lost. Okay, as compared to that, so in T1 and T2 we have got a good germination as well as field emergence. So farmers should practice these open sun drying if their intention is to produce a seed material. If their intention is to go for the powder, then they have to go for the solar tunnel drying. So you can see this is the table which represents the outcome of the study. With respect to outcome, see these are the different treatments. With respect to drying under tarpaulin, we have got the very good color attributes and oleoresin content, seed quality, and disadvantage is contamination. Contamination will be there, and this requires a long period. So then drying under a concrete surface, this has got a best result, but only disadvantage is this is also taking a long drying period. Under solar tunnel drying, you can see most of the benefits you can harness from solar tunnel drying. You can save the time, and in trail drying also you can save time, but you cannot have more benefits under this trail drying, as it is not resulting in a good seed viability properties. So based on this, we can conclude that the overall quality of the chili powder was better under solar tunnel dryer. The tunnel and the tray drying affected the germination and emergence of the chili seeds, and drying of bagging, dabbi and kaddi varieties of chili fruits on polyethylene tarpaulin sheet didn't affect the seed quality. Hence, the tunnel drying may be preferred for retaining maximum quality of chili powder, and the tarpaulin sheet and concrete surface drying may be followed for getting the good seed material. Thank you, sir. Thank you for providing.
Okay, thank you, sir. Thank you. The next, we have received some abstract after the completion of this list. There are 135 abstract number. Antibacterial activity of Calotropis procera. B.D. Pandit.
Sir,
Is there anyone?
Hello. Yes, sir.
Yeah,
Sir. Sir, this is Deepali Varange. I have two papers.
Abstract number?
Serial number 9 and 10. Yeah, sir.
But your presentation was yesterday, ma'am.
Medicinal applications of Adathoda vesica, and one more, that is the hello. Uses of indoor plant. Hello.
Ma'am, बोलिए. Ma'am, abstract number your.
Uses of indoor plants, and one we respond positively to them.
Ma'am, please tell your abstract number.
9 and 10.
9 and 10.
Okay.
Your presentation was yesterday, ma'am. Yeah, ma'am.
Hello.
Ma'am. Ma'am, share your screen fastly, ma'am. Within 5 minutes, you complete your presentation, ma'am.
Okay, sir. Is my screen is visible, sir?
Screen is visible, but ma'am, your presentation is not visible.
Okay, sir, without, can I present?
Yeah, ma'am. You can start.
Hello.
Yes, ma'am.
Okay,
Yes,
okay.
ma'am. You can start.
Yes. Okay, thank you, sir. Yes. Okay. I'm Mrs. Deepali Samir Varange. I'm the PhD students under the guidance of Dr. Vijay Chavan from the Gokhale Institute, Srivardhan. So my first paper is
ma'am, your sound is breaking.
Of the Adathoda vesica. So first one is the abstract. We know very well that is the 75% to 85% of the world population. Hello. Hi,
Your
yes,
sound
sir.
is breaking.
Sir, I am audible.
You are audible, but your sound is breaking, ma'am.
Okay. Just 1 minute, sir.
Okay.
Sir, now I am audible.
Yeah, ma'am. Yes, ma'am.
Okay, sir. So for the primary healthcare because of the better culture acceptability, better compatibility with the human's body and the laser side effects. Okay, that's why the Adathoda have the great uses. Mostly in the Ayurveda, Vasica have the great role in cure the specific disease. Vasica is popular for its beneficial effect, particularly in the bronchitis. Mostly a warm decoction of its leaves is useful in treating the scabies and any other skin diseases. Then here how the juice from the leaves should be given in dose for 2 to 4 grams in treating the diarrhea and the dysentery. Then the introductions that the name of Vasica is based on the Sanskrit name, that is a Vasaka, which is indigenous to the India. It's grown all over the India and the lower Himalaya ranges. Vasaka also called as a Malabar tree, the Malabar nut, through the all over the India. It is tall with the several branches, dense and the evergreen shrub. Adathoda vasica is very well known remedy available everywhere, and it's especially popular in the rural areas. Now the methods and materials here have the 3 methods. That first is the traditional bolus methods, that 100 grams of the fresh leaves crushed with the mortar and pestles made into the bolus, and it was covered with the fresh leaves of the Chisquare. It was then covered with the layers of paste of wheat flours. Then the second method is was a steaming, which is carried out using the 2 different methods. In the first one, 100 grams of the fresh leaves were crushed using the mortar and pestles, which is placed in the steel vessels and heated at 121 degrees centigrade for the 30 minutes. Then the third one was methods employ, which is 100 grams of the fresh leaves whereas the treated to the fine paste in the stone mortars. It was taken in the 4 layers of the muslin cloth and squeezed by the hand to take out the juice. Then the chemical compositions, the principal constituents of the Vasaka are the several alkaloids, and the chief one is the vaccines. Then the other 2 major alkaloids which presents in the leaves, that is the vaccines and vaccition 2,3. Now the conclusion that plant is a well-known herb in the indigenous systems of the medicines for its beneficial effects, particularly in the bronchitis. The leaves of the plants contain alkaloids, vaccines, which is the responsible for the small but the persistent bronchitis, and the essentials oil, which is chiefly responsible for the extracorporeal actions such as the unique herb supports the bronchital functions with the expectorant and the mucolytic properties. Now, thank you, sir. It's my first paper. Now I will present my second papers. That is
madam.
uses of indoor plants and why. Hello.
5 minutes you conclude.
Hello, sir. Sir.
Within 5 minutes you have to conclude.
Okay, within 5 minutes. Yes, sir. Okay. Just okay. So the second, uses of indoor plants and why we respond positively to them. First, abstract. For the survival of living plants are the essentials because peoples have been bringing plants into their homes for the thousands of years. They provided food, fibers, buildings like that. So research study associated with the indoor plants, which are discussed, of increasing interest to many people in the questions why the plants have intangible positive effects. Now the effects of the indoor plants, that is bifurcates into 2, that is indoors air quality. In the indoor means they helps to increase the indoor air quality, affects the air pollutants, then the relative humidity, particulate matters, acousticity. Then the well-beings in we feeling well in the indoor plants as well as the outdoor, that stress reductions, productivity, in the health improvements, that they helps to pain reductions. Then the another, that is general discomforts and the behavioral ecology. Then in the colors, right, means we like different types of the colors of the plants, like the flowers. Then the specific diversity fractals. Then the children's mostly in the cities. I'm explaining in very shortly because of the timings. Last one is the conclusions, that there are now many study documenting a wide range of positively effects on the plants, on the peoples. The importance plants is not limited to their role in meeting our physical and economic needs, that plants contribute positively to our mental health, improve our physical health, make our communities safer. Then, hello.
Ma'am, continue.
Hello.
Ma'am, continue.
Yes, yes. Many of the research studies documenting beneficial effects of the plants on peoples have focused on the plant outdoors of the nature. Research has shown that the interior plants in individual containers can also produce the same benefit. Research has confirmed the stress reducing benefits of passively weaving plants. It has documented that the people's impressions of a room and their mental well-being can be significantly the productivity and the mental functionings, and research on the effects of plants on people has shown in essence that the plants are essential for people to at their best, and plants are needed in our lives all around us every day. They have a civilizing effect, they humanize our surroundings. Thank you, sir. Thank you. I would like to thank the principals for ours. Thank you. Thank you very much, sir.
Is there anyone?
Dear participants, is there Ratna in the participant? Sangaratna Taide.
Yes, sir. Yes.
Yeah. So you can start your presentation, Taide.
Okay. Am I visible, sir?
Yeah.
Good afternoon, everyone. So my project is about, my work is about the aeroalgal diversity in the publicity. I am Sangaratna Taide. I am working at the Rayat Shikshan Samsthat Art Science and Commerce College, Mukhada. So I have worked at Dhule city. As we all know, the air is full of bio. As we all know, air is all about the full of ingredients which contain all the biological products also, and the algae is one of them. The algae is ubiquitous in nature, so all the possible habitat algae has found. So air is one of the components where algae is also found. So we have studied the aeroalgal flora of the Dhule city. So we analyzed the algal flora and various parts of the Dhule city. So to ensure the aerophycology, to know the dominant algae at present in Dhule city, so we analyze the algae present in the atmosphere of Dhule city. So for that, we use the method the culture media preparation and open air exposing petri plates method. For that, we prepared two cultures, agarified base medium and gold basal medium. And then after we expose the petri plates for about 2 hours interval. And then we incubate the petri plates and illuminate culture rack with a 40-watt fluorescent. After that, we found the observed the culture and found there is something growth in that culture medium. And after then we observe under microscope. For that we use about 25 to 30 plates which sealed and labeled as according to the sessions, like morning session, afternoon session, evening session, and for the night session. And we expose them for about 2 hours interval. Okay, then after observing the growth, we seal the petri plates and incubated the petri plates at the 40-watt fluorescent tube lights. Okay, giving the light intensity about the 2000 to 2500 lux continuously for about 20 hours. Okay, then we
continuously add medium for to avoid the drying of this petri plates. These are the petri plates we prepared. Okay. After result, we conclude that we found about 20 genera or 20 taxa of various types of algae are found in the air of Thule city, among which there are the certain classes like Chlorophyceae, Bacillariophyceae, and Cyanophyceae. Which of this Cyanophyceae is most dominant among them due to the nature of Thule city. Thule city is generally contain the high temperature, and the Cyanophyceae are the members which can grow at the high temperature. So among them, the Cyanophyceae has the most of members. Okay. After that, the chlorophyceal member like unicellular algas like Anchistromus, Chlorella, Chlorococcum are also found in the Thule city. And the members from Cyanophyceae like Anabina fertilissima, Anabina iangaria, Microcystis, Nostoc, which are the also dominant parts due to the moisture contained near the Panjra river. Okay, these are the some algal. These are some algae according to the graph, and these are the growth which we found at the culture plates. Okay, these are some members. These are the reference we use. Okay, thank you, sir. Any question to anyone?
Thank you, that is, sir.
Okay.
Okay, this is the last session. Naikode, sir.
So is there anyone who want to present his or her research work and has not presented yet?
Is there anyone?
Okay, then very nice. So I would like to invite Dr. Ranjit Bansode sir for the remarks of chairman.
Okay, thank you, sir. Thank you very much. I firstly congratulate all the researchers who have presented the paper in this particular oral presentation session. We have expected that in this particular session, the total presentation will be 52. But in particular this session, the researcher presented only 25 presentation. It is not bad, particularly in this hot season. So I must congratulate all the researchers once again, and thank you very much for giving me this opportunity to act as a chairman for this particular oral presentation session. Thank you very much, sir.
Thank you, Ranjit sir. Thank you very much for being very valuable as well as very resourceful chairman for the session. As well as thank you very much our technical team, Tanmay Mandrekar and Srushti Dhyan, as well as all dear participants for your encouraging support as well as very active participation in the conference. Definitely these 2 days, they are very fruitful us, but yeah, due to time constraint, it is not possible to have valedictory function, that official valedictory function at the end of the conference. But definitely you all will have enjoyed as well as you have learned very much from the conference, I think. And definitely we all are going to miss you, all dear participants. So maybe in future we will organize such conference, and your cooperation as well as support is always needed. So if anyone want to talk, then he can give the feedback about the conference, and then we will talk. So anyone from the participant who want to talk? Okay, so dear participants, I will be sharing feedback form as well as another form regarding the paper presentation. So it will be shared in the group and it will be open for few hours. So you are requested to submit the information as well as the feedback form. Thank you one and all. Thank you. See you soon.
Thank you, sir. Thank you, sir. Thank you.